Okay. Okay.
So
Start there, then go here.
good morning, SPF friends. Welcome to Tampa, and welcome to the 2024 SPF Annual Convention and Conference. We're so glad that you're joining us virtually. Normally we did this last year. It seemed to work well, so we're taking this shot again. We appreciate our team here that helps put this together. This is going to be a great conference. As you guys know, last year we talked about momentum. And as we ended that conference, we kicked off the Spastic Paraplegia Foundation Centers of Excellence Research Network. And one of the big things we'll be hearing about at this conference is the progress that group has made in our first year as we get ready to move into our second year. So a lot of good things happening. You guys help us get the word out about the SPF and all that we're doing, all that we continue to work toward doing in terms of finding treatments and cures. But we got a lot of information in these next 2 days. And again, we're so thankful that you've joined us virtually. And with me, this lady, all of you know, you've talked to her, you've worked with her, and of course I work with her every day. Norma Pruitt, who is our executive director and who has put together this program and this agenda. Norma, great job, but share with our folks your thoughts about this weekend.
Well, we are so excited that everyone is able to join us in person. But if you can't join us in person, this is the next best thing, right? We're all here to have a great time and enjoy each other socially, but we're also here to enjoy all the information that's there. And there is a lot of information. So I want you to take all that in. I want you to look at and listen to and research what these doctors and investigators and physicians are talking about. You got questions, write all of those questions out in an email. Send all that information to us. You've got all of our email addresses, and then we're going to try to compile all that information and at some point have a bigger, broader conversation about what did that mean? What did they say? And we want to make sure that everybody is all on the same page. So thank you all for joining us. I want to give a big shout out to those folks that have provided sponsorships to the conference. As you guys know, this is not a fundraiser. We're trying to cover the costs and be able to, meet whatever research proposals that we've received. And we've got a list of things to go over as far as the research proposals. But I cannot do it without all of our sponsors, all of the contributors and donators. So thank you so much for letting me sit here this morning and talk with you. And I'll be running around, so you may see me zip in on a live feed. So wave at me, and I'll come back to you sometime later while we're here. Thank you, Greg.
Nora makes a great point. We could not do this without the sponsors, and I think you can access those in our on our website, sp-foundation.org, or you may have gotten those in the email that Norma has sent you about the conference. But look at those and be aware of who those sponsors are. We could not do this without them. And one last point that Norma makes, it's important for you guys to remember. This annual conference is just part of the annual ongoing work of the foundation. We'll continue with information we learn here as we talk about future SPF talks, as we talk about future emails that we do to you guys. So
e-blasts.
So take notes, and we will be talking to you about more about these issues in the near future and through this next year. We're getting about ready to get started. We'll kick things off in a few moments by welcoming folks here, and then our MC, Tim Krogan, will join us. Our first speaker this morning will be Dr. Hande Ozdinler, who's our keynote speaker. So we're looking forward to kicking this off. Hang with us, and we'll be back with you in just a few minutes. God bless you, and thank you for being with us.
Thank you, guys.
go. It's all right. It's all good. Yeah, they can see you. Come down here. Lay on you. Oh. There you go.
Are these open?
Good morning. It's great to see you here this morning. I hope you've had a good trip in and you're well rested to get started at 8 o'clock in the morning. And I know that we've got some folks still registering out there, so we're a few minutes late in getting started to try to give those folks an opportunity to get registered and get in here. But just so you know, we've got a little bit of padding in our timing this morning, knowing that it might take a moment to get things going. So we'll be back on schedule very quickly because there's a lot of information for you to hear, a lot of people for you to meet. We want to welcome you to the 2024 SPF Annual Conference. I'm Greg Pruitt, the president of SPF, and on behalf of our board, we want to thank you for being here. We're glad that you're here. There's so many. You know, I remember the first convention that I went to was in St. Louis in 2009. And you guys can probably remember, whether you're HSP or PLS, you can probably remember this stage in your dealing with your condition. It was 2009 and Norma was still working and she couldn't go with me. So I'm in St. Louis by myself, and in those days the conference was a Friday evening meal and speaker, and then all day Saturday. And so I get there and I get in my room and I get settled in, and it's time on Friday evening to go down and register. And so I go down the elevator and I walk off, and I don't know anyone there at all at this point. So I'm beginning to meet people, and we go through the process Friday night, and then Saturday we finish our main speaking, and down a hallway are some breakout sessions. And so I take off down the hall to go to a breakout session. Now, 2009, Greg thought that he walked normal, okay? So I go around the corner and I get to a turn in the hallway, and as I do, someone said, "We really want to thank you for being here, and how long have you been dealing with HSP?" And I kind of looked at him. I thought I was sneaking around here, you know. But there are different stages in our progressions and in our diseases. And at that very first conference, I met some people who are still friends today. I met some doctors and some researchers who are friends today that I try to follow and keep up with what they are doing. So if you're a first timer in particular, we want to welcome you. About 60% of you are first time attendees. And we hope that next year, that figure will be less, because we hope more of you first-timers come back for a second time and a third time. We hope that you find value in what you hear and what you learn, the professional researchers that you meet and hear from and get a chance to talk to, but also the friends that you meet here. Again, it's strange. I've seen a number of people already that I only see one time a year, but they feel so much like family because through the year we work and think and talk about the foundation, but more importantly about our personal lives and the things that we're dealing with. And all of us from time to time, I'm included in this, we have difficult days. There are days when I get really irritated that life is the way that it is. But as I do that, I look around me and I'm very thankful for
so many blessings, so many opportunities. You know, if I had my choice like you, I would choose it. I'd never had this. But I can tell you, I have met a lot of great people. I have a lot of great friends. And I hope that as we do this together and we support one another, that makes a meaningful lifetime difference. So I'm going to try to deal with it in that vein, and I want to encourage you to be as positive as you can, even on the tough days. The foundation, over the past few years, has worked to expand the number of people who are involved with the foundation, who know about the foundation. And let me say some things to you. Probably all of you who are here have done some of the things I'm going to mention, but if you have not become a member of the foundation through registering on our website, on our platform, we want you to do that. We need you to do that so that we can communicate with you about a lot of things during the year. I'll give you, for instance, last year was the first year we did the virtual feed that we do, and you'll see me and others back here at this setup interviewing folks and talking about things for our folks who couldn't be here but who registered to do this virtually. Norma and others have sent out probably half a dozen emails, email blasts, had discussions on SPF talks about the convention and about the fact that you could do this virtually. But just in the last 2 or 3 days, we've had a number of folks emailing us saying, didn't know we could do it virtually. How do we do that? How do we pay for it? And so we need folks to register. We need folks to look at those email blasts when you get them, when you can participate in an SPF talk. Those are the ways we get information out to you about the important things going on that we want you to know about and we want you to be involved in, because this foundation is only as strong as we are all together. I want to stop at the moment and encourage you in your program. On page 3 is a large listing of sponsors. And we're so thankful. I'm not going to read those to you. They are in the program. But that sponsorship list is growing every year, and we appreciate them. Some of them will have booths out here where they'll talk to you about various topics and various things associated with the conference and what they're doing. So please stop and visit with them when you can. Thank them for being here. And anytime you get a chance to say thank you to any of these sponsors, we appreciate that because they are important to us. I want to recognize our board members who are in the room. Frank Davis. Frank, are you here? And I'm missing you. I can't find Frank. Frank is here. We had a board of directors meeting yesterday, and I know Frank is here. He may be working on someone, but Frank Davis, I'll try to introduce him again later. Jim Sheehan. Jim, raise your hand, please. Here's Jim. John Cobb. Thank you, John. Tina Krogan in the very back. Thank you, Tina. So these are your board members who are here, and some of our other board members are participating virtually. We, as a board, want to thank you. Our word for the past couple of years has been momentum. We as an organization trying to build more momentum toward those treatments and cures. You know, we talked about yesterday in our board meeting, and at the end of the day, we had a 2-hour session with our researchers saying, okay, here's where we are. How do we get to the next step? How do we get focused in our research? How do we get focused organizationally and with you and finding those treatments and cures that are so important? We had a great discussion with him, and if you were at last year's meeting, you recall that last year we kicked off the Spastic Paraplegia Foundation Centers of Excellence Research Network. And that's a group right now of 10 clinics across the country who are working together to do things consistently, to build platforms and databases that can be used to diagnose people quicker, because I hear stories from many of you about how long it took to get properly diagnosed. So we want to have ways to get people diagnosed more accurately, quicker. We want to have opportunities to be able to share that information collaterally among the researchers so that what one researcher is learning and doing, others know about. Because it's with that working together, that collaborativeness, that we hopefully find some treatments. And you're going to hear people talk about some of those things quicker from this podium this week. How we maybe are beginning to focus on more clinic-based things that that result in some clinical trials and treatments to test some things about making our conditions better. You're an important part of all that in a lot of ways. Dealing with your physicians and your researchers is important. But I want to also thank you at this point. For the past 3 years, this foundation, with your help and the help of so many others, has raised about $1.3 million to go toward research. And you probably know from the information that at this point, 98% of that money goes directly to research. And that's important that we funnel in and we focus on the things that, what have we learned from the research we've done, and how can we take the next step toward a treatment or cure that makes your life. And if you've got HSP, as I do, and I'm SPG4, what does that mean? Well, 13 people in my family have had it. The worst part for me is my 36-year-old daughter has it. And beyond her, 5 grandchildren who have a 50% chance of having it. Those are the reasons that I still do what I do, and Norma does what she does with me with the foundation. We believe, we hope, and we're going to work to do all we can with you and with our board to find a day that we have a treatment that makes life better for us and for those who come after us. So I want to thank you for all that you do to help make that happen. The contributions that you make, the people you talk to about contributing, it all matters. Every penny adds up to a very important part of the process. But momentum has been a key word. We want to continue that momentum as we go forward in this meeting and in this year. A couple of other things you're going to hear about that maybe you haven't heard these kind of presentations before. We want you to know that your board is looking as broadly and as widely as we can, not just at research, but are there efforts out there to take prescription drugs, compounds, chemicals that might work for other medical problems that might work for some of us. There are organizations out there that are looking at those kind of process. Now, that's like research. It costs money. But we want you to know we're looking at those and whether we might make some progress in that regard. You're going to hear from a company today called Rarebase. And that's what they do. We just want you to know that we're looking at those things and for you to understand some of those things. The young lady who'll be presenting and talking to you about Rarebase is Natalie Downs. Another presenter you're going to hear today is from an organization called CPATH, a gentleman named Thom Hart. We all know that major dollars for research in our areas come from the National Institute of Health. We need to have the Federal Drug Administration involved. There are a number of Washington components. If we make it over the top, we're going to have their involvement, their help in many ways. So we're looking at organizations like CPATH who exist to help organizations like ours better network, better work with those networks that can help us reach those folks in places like Washington, D.C. So you're going to hear from Thom about our work in that area. So you're going to hear a wider range of discussions about the things your board is doing. We don't want that to be boring. We want it to be educational because we need for you to understand what we're trying to accomplish on your behalf. And we welcome your input. We appreciate your input all the time. You guys that were at our conference last year know that I'm a book reader, and I like to share sometimes things from books. And as I thought about this conference and where we are, I looked over at my bookcase and I saw this book that I read probably 15 or 20 years ago. It's just called Wooden. It's a book about John Wooden, the former basketball coach at UCLA. And if you're an old college basketball fan like I am, you know that he's probably the greatest college basketball coach that ever coached. Those of us who are from Kentucky thought for a while Calipari might be it, but he's gone to Arkansas, so we gave up. But anyway, John Wooden, a great coach, won 7 national championships in a row and 10 overall. And he was born in 1910 in Indiana, grew up in kind of a poor situation, but was focused and trained and worked hard all of his life. And as you all know, 10 national championships is quite a feat. But this book is an easy book to read, and it just has a lot of old stories, a lot of principles that John Wooden lived by. When we were in Pittsburgh, our theme was, and we did some video stuff about it, it's time. It's time. It's time for success in treatments. It's time for success in cures. It's time for us to grow more in that direction. Well, one story I want to tell you two real quickly. One in this book, in 1975, John Wooden won his last national championship. And being an old guy from Kentucky, I remember that year well because I was 16 years old, and John Wooden and the UCLA Bruins on semifinal night beat the University of Louisville. Well, if you're a UK alum and fan like I am, that was a great night. We were rooting for UCLA. But that meant the University of Kentucky now was going to play UCLA for the national title on Monday night. Well, John Wooden writes in this book that all of his years, when the game was over, he knew he had to head to a press conference, and he looked forward to talking about the game, meeting with his players. He looked forward to the post-game work that he did. But that evening after he beat the University of Louisville, as he walked off the floor for the first time ever, he didn't look forward to that. He dreaded that. And in the steps from the court to that press conference or to his dressing room, he made the decision, it's time. It's time for me to retire after tomorrow night's game. Now, the story is that he hadn't told anyone but his wife. His players didn't know, the media didn't know, but he made that decision on his own at one time based on the circumstances. That's where we are. It's time based on where we are, how we are growing, how research money is growing. It's time for us to take that next step toward finding those treatments and cures. And every one of you, every one of us is a very important part of that. You may be here saying, I'm here for the first time and I'm learning and I don't have a role. Yes, you do. You may be here and your disease may have progressed to the point that you feel like I can't be very involved. I can't be helpful. I can't make a difference. Yes, you can. I want to read this to you. It's entitled, Why Teams Fail. I'm getting older in my eyes too. He writes, no matter how great your product, if your sales department doesn't produce, you won't get the results you want. Different departments must all function well for the company to succeed. Different individuals must also function well for the departments to succeed. It takes all doing their best. I told players at UCLA that we, as a team, are like a powerful car. Maybe a Bill Walton or Kareem Abdul-Jabbar or Michael Jordan is the big engine, but if one wheel is flat, we're going no place. And if we have brand new tires, but the lug nuts are missing, the wheels come off. What good is the powerful engine? It's no good at all. A lug nut may seem like a little thing, but it's not. There's a role that each and every one of us must play. We may aspire to what we consider to be a larger role or a more important role, but we cannot achieve that until we show that we are able to fulfill the role we are assigned. It's these little things that make the big things happen. So no matter what seat you sit in today, no matter your age, no matter where you're from, no matter where you are in the progression of this condition, or you may be a spouse or caretaker or supporter, whoever you are, wherever you are in this process, you are extremely important, and we need you, we need each other. So thank all of you for being here. I look forward to meeting as many of you as we can over the weekend, because when I leave here every year, well, we have some great new friends. So take time to meet everyone that you can, and we appreciate you being here. Now, I'm looking around the room. Tim Krogan, who most of you know, is our MC, and Tim does a great job. But Tim, anybody know where Tim is?
Hey, I'm coming. Give me a minute.
Would you get in here, Tim? Come on.
Look, man, I've been on this journey for the last half an hour to get all the way from the 6th floor down to this ballroom. I am on my way, but I'm taking it a little bit easy because I got me a little bit of potatoes in my pants. and I'm on my way to the stage. So everybody, just hang on. Hey, by the way, who put the stage all the way over there? I mean, come on. I'm getting there. Don't worry. Don't worry. We'll still be done by Sunday at midnight. Don't worry about it. I'm almost there. Uh-oh. Got a little sideways. And, of course, I come to the side of the stage with the stairs. Uh-oh. All righty, here we go. All righty, everybody. I made it. Just barely. As you can see, I'm wearing the latest fashion accessory, and they are called a potato pants. And these came from an idea from one of our members, Lori Renna-Linton, and she lives in Austria, and it's a great story. We've got some information in the booklet about that, and this is really a great way for caregivers and members of families to understand what it's like for their family members to go through an upper motor neuron rare disease and kind of understand what they have to go through. My wife, Tina, has HSP SPF 7. I don't know what she's got. Now I screwed up all the adjectives, but who cares? But anyway, so she has SPG 7, and it's tough for her to move around and whatever, and this really is a way for me to understand what she's doing and what she feels. And I deal with her every day, and I love her, and it's our way to at least be compassionate and understanding of what she goes through during the day. This is also a way to do some fundraising. You guys can get together and do fundraisers with this concept. It's really kind of neat. We have some information about that in the booklet. Also, we have 4 pairs of these that we're going to have available for you to try on out in the lobby today. Okay? So anybody wants to try on a pair and feel the sensation I'm having right now, you're welcome to do that. So we're going to have those out there for you guys to deal with and run around with today. You know, this hotel is kind of done in a circle. Maybe we'll time you taking laps around the building, okay? So anyway, much ado about nothing there, but here we go. We do want to show you a quick little video that was produced with Lori Renna-Linton, and it tells her story and about the potato pants. And what we want to do, I have to open my phone here for a quick second and read this intro. We want to have a special thanks to Daniel DeFabio. He's the person responsible for the documentary and helping to organize the Rare Advocacy Summit. And this documentary is with Global Genes and the Disorder Channel. Okay, here we go.
My nickname is Runner, a play on my last name, Renna, but mainly because I'm a pretty fast actual runner. I was diagnosed with hereditary spastic paraplegia, or HSP for short. My uncle's head. HSP is a communication problem. The muscles in the lower limbs get the signal to move either too late, too weakly, or not at all. Hence the difficulty in moving. It is also progressive. Let me give you a picture of what I mean. Here I am in 2014 at my house, attempting to run. This is a therapy clinic in 2018, walking with the help of hiking sticks. This is at my house with my physical therapist in 2021, testing my speed without hiking sticks. And this is not walking, but rather getting up from the ground in 2023. So you see, it gets worse over time to eventually being in a wheelchair, which I currently use whenever I'm outside of my house. I am no longer a runner, and there is currently no cure. I am physically disabled, but instead of my world diminishing with this diagnosis, it has only opened up possibilities. To be clear, I did not go from getting the diagnosis to immediately being okay with it. The way you thought your life was going to play out has been turned upside down. You imagine what you will no longer be able to do. But if you flip the script, it's not what you can no longer do. It's what you must now do differently and what you can do in addition. I co-created Potato Pants with Constanza, my friend and colleague. It is my answer to turning an idea into reality. In August of 2014, I went hiking with my 10-year-old daughter and her best friend. Along the way, I had to stop every few minutes. Mama, why do you keep stopping? Honestly, it's like walking with my pants full of potatoes. The next day, I told Constanza that I went hiking, that I had to stop often, and that I told the girls that it was like walking with my pants full of potatoes. She laughed and said, "Really? That's a great image. Let's figure out what we can do with that." We are both teachers at a school in Austria and have access to a huge community. Here is the Potato Pants idea that we pitched to them.
Wir werden ein Rennen veranstalten, bei dem wir Hosen mit Erdäpfel füllen, bis zu 10 Kilo, bei dem die Schülerinnen und Schüler, die Lehrer, die Eltern, die Politiker, alle, die kommen, um die Wette laufen werden, wobei es nicht ums Gewinnen geht, sondern darum, dass jeder einmal fühlen kann, wie das ist, wenn man im Alltag mit so schweren Beinen durchs Leben geht.
They loved it. And then they went to work. The sewing teacher made the pants. The phys ed teachers set up the racetrack. The French teacher volunteered to be the DJ. The parents made food. The head of the PTA organized the donation boxes. Students volunteered. And my student filmed the entire event, an excerpt of which you were about to see. That is how you harness the power of community to do big things. And that is how you turn an idea into reality. From that event, we raised 6,000 euros for research, but I thought the idea was so good that it needed to be revived. In 2016, my students took up the mantle. They wrote, filmed, and edited a movie to raise awareness of HSP. They created a huge stand at the Vienna Sports World, and they organized over 80 volunteers for the Vienna City Marathon. To date, Potato Pants has raised over 50,000 euros for research, and from our school alone, 15,000 euros. That is impressive, but just a drop in the bucket of what is needed. Potato Pants relates to the experience of HSPers. No one is saying that if you don't have HSP, you can ever know what it feels like. But waddling around in awkward, bulky, heavy pants where they're slow, off balance, and get tied quickly will hopefully make people stop and think that it must be pretty tough going for HSPers. And with that awareness, develop empathy and appreciation and do things like fundraising. Some of the biggest insights come when they take the pants off, when they realize that HSPers are stuck in their potato pants permanently. My world has certainly been enhanced by this awareness of my body, of having to figure out how to do things differently. However, I have had this for 15 years. I got the lesson. I understand. It is now time to find a cure. The money we raise for Potato Pants will go specifically towards a project that is aimed at finding a cure. But that can only happen if we all work together. We need you. Together, we can do big things. Let's turn the idea of being cured into the reality of being cured.
Okay, everybody. Thank you. Thank you very much for enjoying that presentation and sort of understanding why I came in this way. And another thing is, if you notice, I sort of look like a zebra, don't I? Got a lot of zebra stripes on over the place. Well, that is sort of, we've got a nice little notation in our program on page 28 of why zebras. It's sort of the, what's the word we want to call it? The zebra is commonly known as the rare disease community as the mascot of rare diseases, hence the look of what I have today. So we want to tell you guys this is a really great idea, kind of fun, kind of nice to do, and you can learn and tell people all about what it's like to live daily with HSP or PLS. I got a couple of quick notes before we get on with the show. First of all, Dr. Fink is doing his gait assessment, so if you guys signed up for that, make sure you remember the times and where you have to go. He's over here in this corner of the ballroom space outside. Also, Dr. Fakhari is doing his child assessments today and stuff, so make sure everybody gets with him at the right time. One big note, it says in your program, during lunch at 12 o'clock, it's supposed to be in the private dining area on the first floor. That's not where it's gonna be. It's gonna be in here, okay? It's a little too hard to lift and shift everybody to another space. And since we got everybody tucked in here, all the meals are gonna be done in the main ballroom. Okay, which it's going to be a little goofy. We got to be able to set the tables. And you guys who've been around a while have been through this before. It happens. The staff here is really good, and they'll be able to get us set up for lunch. Okay, the main thing is we just want to get going. We've got a lot of information for you. We're going to start off this morning with our keynote speaker, Dr. Ozdinler. So she's going to come up in a minute. We're going to set up her program's ready to go. Now she has to make an entrance like I just did. There she is. Here we go. Come on up. Oh, thank you very much. Thank you. You're all good to go.
Thank you so much. What a great conference, and I'm happy and honored to be here with you today. And I really like the idea of these potato pens, because it gives an image on people, and then people can actually take it with them. And it's hard to talk to people if this hasn't touched them personally. And I think having images like this is very important for creating awareness. And for rare diseases, I think we have to create awareness to get everyone involved. But more important, or maybe equally important than awareness, is the science behind the research and how the research takes us forward. So I'm going to talk to you a little bit about, I don't know if I can move this, building treatments for upper motor neurons, and I'm going to team up with you. Yes, and I'll tell you next slide because I don't have my computer with me. I want to start by thanking everyone who made the research possible. And of course, the Spastic Paraplegia Foundation is one of the resources that we look up to when it comes to research. And our lab has been gifted by many other foundations, especially NIH as well and NINDS. So we thank them for the support that we receive, and it's a lot of grant writing, believe me. And the next slide, please. But more important than the money is actually the spirit behind the work. So we don't do research just to get the money. I think it is important that we help understand and bring a solution to an unmet need. And many people are suffering a condition that is so rare, and it came to them, and they don't know why, and they don't know how, and they don't know how to find a solution. But then when they come together, they create a community, and they create a momentum, and that makes the change. So I'm very thankful to Hank, and I don't know if he's watching us. He introduced me to this Spastic Paraplegia Foundation. I attended their meetings in Chicago, and I realized how passionate people were. And that passion is actually the fuel. And I'm very happy to be part of this community. So I thank all of you to be here and to create that momentum. I think you are the ones that makes us going every day. Next slide, please. So as you know, our overall goal is to have effective treatment strategies for upper motor neuron diseases. And I don't say HSP or PLS, I say upper motor neuron diseases. Because there are many diseases which originate when the upper motor neurons become vulnerable and progressively degenerate. And because we're already so rare, and then we divide among ourselves, I'm HSP, I'm PLS, I'm this mutation, that mutation, I think it defeats the purpose. I think we need to unite and say we are upper motor neuron diseases. And that is like an umbrella, starting point is an umbrella. So our overall goal is to have effective and long-term treatment strategies, right? But what are the roadblocks? Like, why can't we get there? What is the problem? There are actually numerous problems. One of them is the genetics of the disease, and the other one is the underlying mechanisms. Even though patients display similar clinical outcomes, the underlying cause which resulted in that clinical symptom may be different. And in that case, one drug may not work for everyone. And even though it's rare and then one drug doesn't cure everyone, so that's a problem. And then there were really no good preclinical screening platforms. Let's say there are companies out there who has a drug, but how will they test if this works in the upper motor neuron diseases, right? We did not really have preclinical screening methods. And then even if you start a clinical trial, what will be your biomarker? What will be the outcome measure? Whom are you going to include? So there are clinical trial readiness programs that were not ready. And I'm going to touch back on that because Dr. Mitsumoto has made immense progress in that area. But then once you realize what the problem is, then you can begin to bring solutions. So then the solutions could be that we generate knowledge about genetics and the mechanism of the disease. So we begin to understand the genetics of the disease, the mechanism of the disease. And then we can develop screening platforms for drug discovery. Then if it's so rare, maybe we can have gene therapy approaches for patients, especially with known mutations. And then maybe there could be drug repurposing, that we don't need to reinvent the wheel, that some drugs may actually work for some HSP or PLS patients. And we can identify new drugs, right? So I want to keep this as a thinking point throughout the symposium, because during the opening talk, you heard the idea that we are getting ready, we want therapies. I think we are at a point where we can begin to talk about how do we get there? Are we there yet? What needs to be done? What is the priority? Let's think about those things. Let's talk about those things. So next slide, please. All right, so let's talk about a little bit about the genetics of HSP and PLS. So HSP has, again, various types, and there are more than 80 genetics type of HSP, 80, imagine that. And then even though this is a rare disease, 80 different genetics. So next slide, please. Some of them could be autosomal dominant, which means even if you have one mutation, that is enough to cause the damage. And there are numerous genes, and I took them from the website of the foundation and also the clinicaltrials.gov HSP-PLS site. So you can go and search for those genes as well. I'm just giving a snapshot of some examples. But those are the mutations that are detected in HSP patients. And even if you have one copy of the mutation, that is strong enough to cause the disease. So whatever these genes are coding for, the proteins, imagine that. That protein must be so critically important for the function of the upper motor neuron that one protein not working can make the upper motor neuron decide to degenerate. So then the proteins that are coded by those genes are extremely important for the health and the function of the upper motor neurons. Next slide, please. And some of them could be autosomal recessive, which means you need to have 2 copies of the gene to be mutated. These are usually in the familial cases because the mother and the father may have the gene and they don't show phenotype because they're heterozygote, but then it may appear in the child. So there are numerous genes that are linked to HSP. Next slide, please. And also in PLS. PLS is actually a little bit different because they are also considering PLS within the spectrum of ALS, or the genes that are mutated in the PLS patients may not be the same as HSP patients, but some mutations that you see in the ALS patients are also in the PLS patients. So I chose this recent paper. Please go read that, and I think that's one of the most recent ones. So it gave a list of the mutations that are detected in the PLS patients. And I don't think we still understand the full spectrum of the genetics component of the disease. Maybe 10%, 15%, but there is a very large unknown sporadic component. But then what is the advantage of knowing the gene mutation, right? Why would we care about knowing the gene mutation? And what do those genes mean? Because if you look at them, one gene is a kinase, the other one is a actin skeleton protein, one of them is a receptor, they're all different proteins. How come mutations in different proteins lead to the same problem? So what do these genes mean? What do they do? And how come different gene mutations lead to the same disease? And if you know your mutation, how can this help you? Would this help you? And can there be personalized treatments because you know the mutation? And I want to pause here and tell you that knowing the mutation is actually critically important because we know from other diseases like SMA and recently ALS with the SOD1 mutations, there could be antisense oligonucleotides that are designed specifically for that mutation. And that could be personalized treatments or treatments just for that mutation. So the technology is actually there. There are antisense oligonucleotides, there are viruses, virus-mediated gene delivery, there are even small RNAs, so the technology is coming. And I think that's why knowing your gene mutation is very important. But of course, there are many questions. What is the mode of delivery? Is it toxic? Can we do clinical trials and so forth? But knowing the gene mutation is critically important. Next slide, please. All right, so let's say I told you that there are many genes that are related to the disease, right, in the HSP-PLS, and we call them the upper motor neuron diseases. And what do those genes tell us? We did an experiment trying to understand why different mutations in different genes all boil down to one clinical phenotype that is characterized as the upper motor neuron disease. We took all those genes that are mutated in patients, and we looked at their protein-protein interactions, and we identified their protein landscape, meaning with all their binding partners. Every gene that is mutated codes for a protein, and that protein binds to numerous other proteins to carry on a cellular function. So all these proteins that they bind to, we took them together as one group, and then we said, proteins, what do you do? What is your function? What's important for you? And they begin to tell us. They say, oh, the cytoarchitectural dynamics is very important for us. And we say, all right, axon transport and maintaining the long axon. And then they said lipid homeostasis is very important for us. And then they said, oh, if we have any neuroimmune modulation or if there's any attack, like an immune attack, we become very fragile. And especially for after these canonical pathways that are important for them, we said, all right, who interacts with whom? Where are you located? What do you boil down to? Are there any druggable targets? So we let those gene mutations speak to us about the disease mechanism. So I suggested you read this paper. The next slide, please. And then we went ahead and tried to understand, especially within spastin and alsin, and those are very unique genes that are mutated in upper motor neuron disease patients. And then we said, would the mutations help us understand disease mechanisms and also potential druggable targets? Now, what do I mean by that? Each gene has domains in them. And those domains are the regions where they interact with either different messenger RNAs or proteins or receptors, but there are different domains in each protein. And some patients have a mutation in one domain, some patients have a mutation in another domain. So when you have a mutation in one domain, I would imagine that whatever the protein is doing in that domain, the protein won't be able to do because there's a mutation. But other domains may still be working. So then we looked at the domain-specific interaction partner analysis. And by doing so, we made a heat map of the interaction domains in each partners in each domain, and then did canonical pathway analysis in these regions to see what is really important for that particular domain. So now it's not only the gene mutation, it's also where is your mutation? Which domain is your mutation in? Do you see we go very precise into the location? So when we do this for the alsin and spastin, we realize that they are very different. They are very different, but there are also some commonalities. Next slide, please. So when we looked for all those interactions and which canonical pathways are affected, then we said, all right, there may be some FDA drugs out there which have been either approved or in clinical trials, and their mechanism of action is particularly to mitigate or to improve that problem, like that canonical pathway. We searched, and we found many. So we found many FDA-approved drugs either in, or approved or still in clinical trial, and that are actually working on distinct canonical pathways that we identified based on our protein interaction domains for the alsin and spastin. Then we said, okay, is there any compound out there that may work for alsin and spastin based on the canonical pathways that are affected? And we found three, SMI71, and hopefully we'll be able to do research on that one. And there are two more, and they seem to be acting on the canonical pathways that are affected by the mutations in the alsin and spastin. So why do I tell you all this? I tell you because when we know the mutations in the gene, we can begin to understand what is the mechanism of the disease, what is the underlying cause of the disease, and also are there any potential druggable targets, and are there any drugs that have been approved using that mechanism of action, using that specific target. That, I think, opens the doors for repurposing of drugs. Maybe we don't need to reinvent the wheel. Maybe there is a potential out there. Maybe the drug is already out there that we don't know which one to try on the HSP and PLS patients. So that's why the mutations, gene mutations, are extremely important, because they begin to tell us about the disease mechanism, and they begin to tell us potential druggable targets and potential compounds to test. Okay, next slide, please. All right, so this summarizes what I just said. It's very important to know the gene mutation. So if there is a way that you can understand your genetic cause, please do, because it tells us about the mechanism and targets, druggable targets, and so forth. Next, please. So the other advantage of knowing your mutation, and your disease is a rare disease, I want to bring this paper to your attention, and our friend Darius is also an author on that paper, is to develop gene therapy approaches for patients with a known mutation. I mean, that is a very new concept, but it's not out of reach. Because if you know your mutation, if you know how this mutation causes the disease, and I told you with the mechanistic insights that we bring, then you can actually develop AAV-mediated gene delivery approaches specifically for those patients who have that particular mutation. And I think this is a seminal paper along that direction, that if you know the mutation, you can have the virus delivered, and this was an intrathecal delivery, and that they used a APA for M1, and that's one of the genes also related to the disease. And they have seen some improvement in the preclinical animal models. So that will also bring us to the importance of having animal models, importance of preclinical studies. But this is, I think, the way forward, the gene therapy approaches. Next slide, please. So this is one of our work that we actually published in Nature Gene Therapy, and then they made the cover of our publication, because they really thought that this was important. And the reason it was important was because, especially for patients who don't know their mutation, there are many of patients out there who don't know their mutation. For sporadic cases, can we develop gene therapy approaches? That's what we're trying to work on, because we want to find a protein that is so critically important for the health of upper motor neurons, regardless of which mutation they have. So we found a gene, the UCHL1 gene, is very important for the health of upper motor neurons, and we developed AAV-mediated gene delivery approaches to the diseased upper motor neurons, and we used SOD1 and TDP. Those are very unrelated and different underlying causes, and even in those two different spectrums of the cause, we have seen the same level of improvement. So now we have patent protection on this, and we are looking for drug companies to team up with, and hopefully we can move this into clinical trials. Next, please. All right, so now knowing the mutation is important because there could be AAV-mediated gene delivery approaches in therapies, either personalized medicine, or we can target genes and proteins that are critically important for the health of upper motor neurons broadly, and develop treatments more for the sporadic patients. These studies were not present 2-3 years ago. This is very new development, and I think this is where the field is going. Because the disease is so rare, it's very hard for us to do normal clinical trials with like 200 patients, 300 patients, phase 2, phase 3. I mean, that is very hard for the rare diseases. I think this personalized medicine approach is the way forward. Next slide, please. All right, so let's get back to our first slide. What were the roadblocks? The genetics and mechanism. I think we are beginning to understand the genetics of it, and then there are GWAS studies, whole genome sequencing, so we understand the genetics of the disease better, and we begin to understand the mechanism of the disease better, thanks to the genetics and thanks to some of the mouse models. And then potential solution could be that gene therapy approach for known mutations, or even for sporadic. So the gene therapy could be a potential solution. Next slide, please. All right, so one of the roadblocks, as you can remember, is how do we develop the pre-screening platforms? Because let's say people have some compounds, and some compounds go off the market, or their time has expired. Now they want to repurpose the drug. They want to know if it also works in a different patient population, but they need some pre-screening platforms. So now we step in, and our lab was actually the first lab to make a reporter line for upper motor neurons. So after we made this reporter line, NIH actually gave us $2.5 million to investigate this even more, because this was the first time that it was a cell type specific reporter line in the motor cortex. So as I told you that UCHL1 was very important for the upper motor neurons, so I took the UCHL1 promoter and I cloned the GFP gene under the control of the UCHL1 promoter, and by doing this, we created the transgenic line in which the upper motor neurons became fluorescent. So next slide, please. When we create this mouse model, the question is, which mouse model are we going to cross it with? Which one is the good animal model for HSP or PLS? We must thank Dr. Bass, and I don't know if he's in the room. Dr. Bass, are you here? But we must thank Dr. Bass for generating these mouse models. And thanks to him, we actually have very good spastin mouse models. And in collaboration with him, next slide, please. We crossed our mouse model with the spastin model to be able to make reporter lines of upper motor neurons in which the upper motor neurons are diseased, but they also express GFP. You can imagine the power in that, because the cells are in the culture, and when you add the drug, you can actually watch how the fluorescent neuron is behaving. So this creates a platform for drug screening, and we actually received a DoD grant to build this further, and we are now crossing them with the spastin mice to be able to have a drug discovery platform and help companies investigate whether their compound of interest would also have a utility within the context of upper motor neuron diseases. Next slide, please. So we are developing large-scale imaging and mathematical algorithms to be able to detect the branching arborization of these diseased neurons and their response to drug treatment. So that's why this is semi-high throughput, because that's what the drug companies require. So we are getting there, and I think that we will be able to build a platform for drug discovery. Next slide, please. All right, so now lack of preclinical screening, check. I think that we are building that, and that would open the doors for repurposing and also drug discovery. Next slide, please. All right, but when we talk about drug discovery, I don't want you to think that it's so easy. There are so many challenges associated with drug discovery. So, for example, which compound, which drug are we going to test? And we need to know the cellular problems, the mechanism of action. We need to identify the targets. Is there a good preclinical data? Whom are we going to include in the study, in the clinical trial? How will we know if the drug worked? What is the efficacy? And is it really doing what it is supposed to be doing? And how will we measure improvement in patient, like the pharmacokinetic biomarkers? This target engagement and pharmacokinetic biomarkers are really very tough questions, because if the companies don't feel comfortable that they can assess these quantitatively and reproducibly, they don't spend millions of dollars. They say, all right, do your homework, come back again. Because I need to know, how will I know that it is doing what it is supposed to be doing? So this is target engagement biomarkers. So there are many challenges for the drug discovery. But then, there are ways to overcome them. Next slide, please. So I want to tell you our collaboration with Dr. Silverman. And Dr. Silverman is the inventor of Lyrica. And he identified a compound which we call NU9. And because we know that the upper motor neurons degenerate because of protein aggregation, mitochondrial problems, ER stress, cytoarchitectural dynamic problems, so we identified the problems. We know the target. And now he had a compound which did not have toxicity, had good pharmacokinetic properties. We investigated whether his compound would also improve the health of diseased upper motor neurons using those targets that we identified. And we published this paper, and I'm sure, is there anyone in the room who hasn't heard about NU9? Can you raise your hand? Have you not heard about NU9? You haven't heard? You haven't heard? A couple people have not heard about NU9. So NU9 is the first compound that is reported and published to improve the health of diseased upper motor neurons. And it made a big splash, it made a big news, because so far there was no compound, no drug that improved the health of upper motor neurons. And people were always working on the spinal component of motor neuron diseases. So NU9 was discovered as the first compound to improve the health of the upper motor neurons. So we think that's very important for the upper motor neuron field. And next slide, please. We realized that the upper motor neurons suffer from multiple problems. Protein aggregation, mitochondrial problems, problems with endoplasmic reticulum ER, that's where the proteins are made, and problems with their axon transport, problems with their apical dendrites. And with NU9 treatment, we were able to overcome all these 4 challenges at the same time. So to our knowledge, there is also no such compound that can overcome 4 different problems all at the same time. That's why we thought that it was very powerful, because compounds usually do just one thing. When you go for drug treatments or drug therapies, they say, what is the mechanism of action? Where does it bind to? They expect the drug to do one thing. But here, this compound seems to be doing 4 things, and all those 4 things are important for the health of the diseased upper motor neurons. Next slide, please. So then Dr. Silverman actually formed the company, Akava Therapeutics, and there may be a little insert in your bag about Akava Therapeutics. So the good news is, for the first time, now there is a drug company out there, and you can look at their logo, that they actually work on the upper motor neurons. Next slide, please. Yes, here in their logo, they say, our area of interest is the primary lateral sclerosis and hereditary spastic paraplegia. And here's the QR code, you can get more information. But previously, there was no such company. But at least there is one drug company right now who is focused on the upper motor neuron diseases, and who also has a compound that improves the health of the upper motor neurons within the context of misfolded SOD1 and TDP. So that's why, the next slide, please. The question is, would NU9 also improve the health of the diseased motor neurons in HSP and PLS, right? So I'm thankful to the Spastic Paraplegia Foundation for supporting our research, and I'm gonna show you 2 videos. And the first one is actually a corticospinal motor neuron taken from the spastin mice and no treatment, and the dots that you see are the mitochondria. Can you please play the video? And keep an eye on the dots. Is the video working? Can you click one more time? Oh, it's working. Okay, good. Keep an eye on the dots. You will see that most of them are not working. Maybe there are a couple that are making a little move. Because in the spastin mice, the movement of mitochondria is affected. The axonal transport is a major problem. And then this is a 5-minute video, it's a short video. But here, this is actually in the spastin mice, we edit NU9 to the tissue culture medium. Again, this is a 5-minute video. You can show it the video again. So there is improvement in the transport, do you see it? But of course, please take these data with a grain of salt because we have to repeat them multiple times, we have to quantify them blindly, this, that, you know. But these are some very encouraging studies that we have done in the lab and I wanted to share with you. Next slide, please. Yes, so there are some quantifications for the organelle transport, 18 hours in vitro or 4 days in vitro. With NU9 treatment, it seems to be that it is improving both in the spastin and in the G mice. And again, these need to be repeated multiple more times. Next slide, please. So for this figure, I really want to thank the Cure SPG4 Foundation. And they are 2 great mothers and trying to help their children. And they gave us support to investigate within the context of, again, SPG4 and NU9 in vivo. So we know that there are corticospinal tract defects within the spastin mice. And this area on the left is taken from the corticospinal tract. And you can see that these are coronal sections, so the axon bundles are coming towards you. That you know there is disintegration and they are disorderly, but with NU9 treatment, and we did 100 days, 100 milligram per kilogram, we did not see that anymore. So again, there seems to be an improvement, and we need to repeat those studies, but it was encouraging for us that it may be doing something. But again, it's very early and we have to repeat some more. Next slide, please. So the other thing is, when we have the spastin GFP mice, we can begin to understand what is the disease mechanism in those neurons. And the spastin mice crossed with the UCHL1 mice, the upper motor neurons are GFP. So because they are GFP, we can actually do fluorescence-activated cell sorting. That means we can isolate these motor neurons as a pure neuron population, and when we isolate them, we can do proteomics and metabolomics and lipidomics and all these omics studies and RNA-seq gene expression analysis to ask them specifically, what made you upset? Why are you showing signs of vulnerability while your neighbors are relatively healthy? What happened? And they begin to tell you. So next slide, please. Next slide, please. So with these proteomics and RNA-seq experiments, we begin to understand what is really happening in these disease neurons, and that also helps us develop the druggable targets and what is it that we can go after for the spastin mutation. Next slide, please. All right. So I think the problem with the drug discovery is that they're trying to solve all problems with one solution. They think we're going to find a drug, and that drug is going to cure everyone. So to me, this is waiting for Godot. Godot is not going to come. That's not going to happen because everybody is different. And we have to develop the cell-based and mechanism-focused drug discovery. We need to learn directly from those cells and ask those cells, why are you diseased? What happened? What made you upset? And then when they speak to us, this is my problem, that's what's happening, then we can say, oh, really? There's a drug out there FDA approved with the same mechanism. Or if that's your problem, let me just fix that problem. And it may be that it may be different problems for different people, and we may have personalized medicine approaches, and we may have a mechanism-focused drug discovery. So that's how we are moving forward. Next slide, please. All right, so then we again investigate the impact of NU9 on diseased upper motor neurons. This is something that we have to do because the timing is very important. If we can have a Clinical trial for upper motor neurons. Is it possible to include HSP-PLS patients? Can this be a basket trial? What is the biomarker for HSP-PLS patients? How will we know that the drug is engaged within the HSP and PLS community? What will be the outcome measures for them? So those are the critical questions that we have to answer. But I must say that, Dr. Mitsumoto especially is doing an amazing job with the PLS natural history study, getting ready for clinical trials, and also the Center of Excellences that the foundation has supported, and they are getting ready for the clinical trial readiness. They really want clinical trials to be initiated. And that's very important and timely because as our research evolves in the identification of drugs, and we are developing biomarkers and pharmacokinetic biomarkers, then we can actually begin to work together and make this a reality. And I think all stars are aligning with the research and with the clinics that within a short period of time, we may actually bring those two components together. Next slide, please. All right. So then, biomarkers, outcome measures, those are coming with numerous people actually are doing biomarker studies, and we have the DoD grant working together with our colleagues at Harvard and Johns Hopkins. And I also talked about the drug repurposing because now that we have the preclinical screening and maybe the identification of new drugs, and we might have identified a U9 as a new drug. And I think we have touched upon most of the problems, roadblocks, and the potential solutions. Next slide, please. This brings me to the neuron connectivity. And you can see the artwork here. That's one of my photo arts. And I think this brings the idea of the connectome. And even though each one of us seem to be different, we are all connected in a different way. And neurons in our brain are also connected together to function. So if we want effective treatment strategies and effective functional treatment strategies, we need to connect. And that's why I generated this artwork, especially for this event, because I think the essence is that we connect. And once we connect, all problems will be resolved. Because every problem has its own solution within the connectome. So next slide, please. And I think now we are beginning to understand the underlying causes of the disease. We are developing preclinical drug discovery platforms using upper motor neurons and using their health as an outcome measure. We are developing gene therapy approaches. NU9 is a new compound which improves the health of upper motor neurons. There's a drug company out there focusing on upper motor neuron diseases, and if you want to learn more about the company, please come find me. I am the head of the scientific advisory board, and we are identifying new biomarkers and inclusion criteria. So we did not have a company before, we didn't have a compound before, we didn't have gene therapies before, we didn't understand the disease mechanisms before. The science is now exponentially growing. And what we have come to know within the past 2-3 years is more than what we have come to know in the past 50 years. So it is exponentially growing, and that's the time that we need the momentum. That's the time that we need this connectome. That's the time that each one of you become the spokesperson of the upper motor neurons. Because upper motor neurons are the spokesperson of the brain, but now it's our time to be the spokesperson for the upper motor neurons. And next slide, please. And I think there's a bright future ahead once we do this. And you can find my email there, my Twitter, Facebook, social media, internet, this, all my connections. Please, let's get connected. And there was a hat here, yes. So please wear that hat, take a photograph, share it with your friends, and let the voice and the awareness spread, because there are many people who are in this room, but there are many, many more people who are not in this room. And I think we need to connect everyone, and then we need to get the word out to as many people as possible. People need to understand that the brain matters, the upper motor neurons matter, and that if we work together, and we can actually bring a change to the field. So next slide, please. Thank you again for your attention, and I'm ready to take any questions.
Okay, guys, if you want to ask a question, first of all, we've got a couple of microphones out here in the middle of the room. So if you can make your way over here to ask them, or you can raise your hand and I can find you. Oh, I love this part. Crickets. This is great. No, there's one. And of course it's way over there. Okay, I'm on my way. Hey, by the way, I slimmed down during the presentation, and I can cover the distance a little bit faster.
So by the way, this artwork will be on auction tomorrow night at dinner. So please be generous and let the artwork help the science move forward.
Okay, here's our first question.
Thank you. So in looking at this flyer that Akava Therapeutics, is that the name of the company?
Akava,
That they're developing AKV9, is that the same as
Yes,
MU9?
it is the same. Yes.
That's, okay.
Yes, because when the company licensed it, they changed the name to AKV9, but we as researchers, we still work on the science aspect of NU9. We don't work with AKV9, we work with NU9, but the company is doing the toxicology studies, getting ready for clinical trials, but we still need to understand the mechanism of action, we need to understand the biomarkers, like the science portion, we still continue doing in our lab.
Okay, I got another question right over here.
I was late, so you may have already said this, but my son just got married, and he and his wife are concerned if their child will have this. My understanding when I was diagnosed in 2017 was that both parents, or two people had to have a gene. Is that correct?
It depends on the gene, because some genes are dominant. If you have one copy, that you may have the disease. But if it's autosomal recessive, then two parents must have the. It depends on the gene. You remember the slides that I showed you? Not all genes have the same penetrance, and it depends on the gene.
Okay, I'm going to take one more from this side of the room, and then I'm going way over there. Oh, we got that. Norma, you got one over there? Here we go.
Yes, Dr. Buss.
Dr. Ozdinler?
Right here. Oh, really? Okay.
Question, has phase 1 trial started with the NU9?
No, unfortunately not. So let me tell you what happened. After we made our discovery, the company licensed it, and we prepared our IND application to FDA. It was more than 3,600 page document, and I signed it, and it was like 1,200 pages that I prepared. And then they told us that it usually takes about 5-6 months for FDA to review, and about 80% of the cases, they get rejection. We got approval in 2 months. Can you imagine? So now we got FDA approval to start phase 1. And I thought, when we have FDA approval to start phase 1, everyone is going to pour money on us and we'll just do it. And that didn't happen. So we're still trying to raise funds, and phase 1 studies became even more expensive. The only limiting factor right now is the finance. We have the FDA approval, we're ready to go phase 1, and it's tough. Because, scientifically, we have done everything that we could. And it's now, the funny thing is, my father was a businessman, and I was like the rebel artist and scientist, and he would tell me, at the end of the day, everything comes down to money. And I was saying, no, Dad, it's the science, it's the humanity, it's the arts, blah, blah, blah. And he says, everything comes down. And now, he's right. Everything comes down to money, and it's not my domain. And I hope that there will be people who makes a donation to move this forward, and we're raising funds.
Estimation on how long phase 1 will last?
Yes, phase one will be on. Our scientific advisory board is actually, if you check the website, you will see Dr. Mitsumoto is also there, and I'm very, very happy. We decided to include phase 1 only with healthy volunteers for a very good reason, because if we included ALS patients in the phase 1 clinical trial, then this was supposed to be only ALS clinical trial. But we said, let's have our clinical trial with healthy volunteers so that we have the clinical trial as a baseline, phase 1 as a baseline. Then if you want to go HSP route, PLS route, ALS route, Alzheimer's route, FTD ALS route, other neurodegenerative diseases, you will have a baseline for healthy volunteers and we can expand. So that's why our phase 1 clinical trials, we even know the site, we even know the trial design, everything is known. It's going to be healthy volunteers and single ascending dose, multiple ascending dose, and it's going to be 6 months study. And we hope with the recruitment, analysis, that, it's going to be 9 months to a year for the reports to be written and so forth. So that's why that 1 year is critically important for us to understand the biomarker, the studies for other diseases, so that when we get the results from phase 1, we can then begin to formulate how we're going to do phase 2.
Last question. How much money do you need to go forward?
It all changes with the economy. I don't know. Today it was last 3 million. Now it's 5 million. I think the earlier the better. I don't know that. I'm not a money person. I don't know, but we all know that we need support. So let me go back to Dr. Boss, please.
Here we go.
Hi, Hande.
Yes.
I have a technical question.
Sure.
With the work that you did with the SPG4 mouse,
Yes.
how old was the mouse to see that much degeneration? And when you introduced the NU9, was it after the degeneration had occurred or prior to? In other words, does the drug prevent the degeneration or does it reverse it?
Yeah, so this is a very good question. And by the way, if you don't know Dr. Boss, please know him. He is an amazing scientist, and he generated one of the best models for the HSP. So I'm very thankful to him, and I'm also very thankful that he shared his very precious mouse models with us. And we do our work in collaboration with him. So the question that he asks is about the timing. When do we apply a NU9? That's very important. For tissue culture experiments, we need to put them in culture. And for those culture experiments, they need to be early postnatal, which means like they are P1, P2, P3. So they're like little itsy bitsy. They don't have any signs of disease. But we study the cell, the neuron. And imagine that even at that age, P2, P3, there are problems in their axon transport as early as that. So we put them in culture, we let them grow for 6, 7, 8 hours, and then we add NU9 and different doses, 10 nanomolar, 100 nanomolar, 500 nanomolar, and things like that. And then we let them grow for about, 6, 8, that was the bar graphs, different times, and then we make the movies. So that's for the tissue culture, they are very early. For the postnatal, that when we applied NU9 100 days, 100 milligram per kilogram, that we started when they were P90. And I think P90 is about the time, based on your papers, that they are pre-symptomatic. It's not very, very early in the disease, but it's not also very late in the disease. And we give them more than 60 days. And we can discuss.
So the images that you showed comparing the dramatic degeneration without the drug and the vastly different effect with the drug,
yes,
that was when the animal was what, about a year old? Or
I don't know, it's not 3-year-old, but that was towards the end stage. I think, yeah, you're right, because 180, 200 said they're about a year old.
Thank you.
So, but they were aged mice. They were not like early age mice, because we wanted to see first at the very, very dramatic phase. If we see anything, then we can go back. But if we don't see anything, even at the very end stage, then we say, you know what, this is not doing anything. So that's why we wanted to look first at the stage where the difference is very dramatic.
Hande, one last question.
Sure.
Were you able, in your histology, to see any other neuronal cell types? In other words, does that drastic degeneration that you see, is that only in the corticospinal motor neurons, or is that in other sorts of neurons as well?
That's also another very good question. So we have cellular molecular markers to distinguish corticospinal motor neurons, like let's say CTIP2, FESF, there are some markers that are expressed in corticospinal motor neurons. But because this circuitry, the connectome, is so complex in the motor cortex that these upper motor neurons receive input from many neurons. And there are thalamocortical neurons, callosal projection neurons, interneurons, and so forth. There are also molecular markers that are expressed specifically in those cells. So initially, we did our analysis on the upper motor neurons, but we have not done detailed analysis on other cell types. And what I haven't shown here, maybe next time I will talk about it, we also begin to do electrophysiological analysis to look for the functional connectivity of upper motor neurons. And I think that's very important because it seems as if the interneuron population has an impact on the upper motor neuron function because we see cortical hyperexcitation early and then followed by hypoexcitation. And that means that the interneuron circuitry may be involved. And we looked at astrogliosis, macrogliosis, and large pyramidal neurons, and Kalbinden neurons, reticulin maybe for the interneurons, but not extensive studies on their loss or on their degeneration.
We've got a question right down here in the front, straight in front of you.
Sure. Yes.
Thank you for your presentation. I do have a question. I'm not exactly sure how to ask it, but I'm going to give it a shot. So for people that have had the disease for a while and have potentially significant, neuron damage, say this compound works and it helps, fixing the damage and like the neuron grows again. Is there, do we really understand the mechanism enough or how these neurons are being damaged enough in each type of mutation to try and understand if there is say scar tissue that could get in the way of say we fix the neuron, there could be other things that could be potentially getting in the way of these neurons making the connections that they need to again. Speaking very loosely and in a basic way, but the question is around that other step outside of fixing the neuron itself, like the ability for the neuron to do what it's supposed to do. Could there be other things that could be getting in the way, such as scar tissue and things of that nature for people that are fairly advanced in their disease?
So if I understand your question correctly, and that's a very good question actually. So we answer that in two ways. When we define a problem, we ask this question: is it intrinsic or is it extrinsic? For intrinsic, we mean the problems that occur inside the neuron. That happens with their gene expression, with the way that they make the protein. We know what's happening inside their kitchen, in the cell, intrinsic. And there are extrinsic factors that are not dependent on them, but on their neighborhood, on their environment, on their, the interneurons not inhibiting them, and inhibitor neuron not inhibiting them, then they are excited. Or let's say they depend the axon, right? But then axon is attract. How about other axons? How about the highway? If the highway is full of pots, it doesn't matter how good your car is, the highway is broken, right? So what is the extrinsic factors? So to understand this in extrinsic factors is very hard because there are multiple players in that, right? So in our lab, for the extrinsic factors, we focus mainly on the neuronal circuitry in the electrical sense, inhibitor neurons, excitatory neurons, and how they communicate with each other using electrophysiology. I did not show it, maybe I should have. And then the other extrinsic factor that we look for are the neuroimmune modulations, because there are those cells, astrocytes, macroglia, that have an impact on the motor neuron function. And we have reporter lines of MCP1 and CCR2, and we double cross them. We have triple transgenic mice to begin to understand how these neuroimmune cells contribute from the periphery to the central, to the function of the neuron. So we don't really focus on the track the highway if it is if there are any potholes on the highway, but we are mainly considering the neighborhood in the cortex, how that is telling them you can do this, go for it, I'm here with you. Or they say, oh, you can't do that. I know you can't do that. Then those are these communications that we have to improve. So we're looking for the intrinsic factors as well.
Okay. I'm sorry, we're going to move on to another question over here, and then we're going to come here. We're going to finish up because we've got to keep moving. Okay, great question. Go ahead.
Thank you for your presentation. Since my working diagnosis a year ago, I've come to realize that we are all part of this very large, complex jigsaw puzzle of pieces that we're trying to put together. So my question goes to the management of the data that underpins all of this. Because I'm actually a data scientist by practice, so that's where I'm coming from. So when I got my diagnosis, I did research on the online databases, and I found one other person has the exact same missense mutation in the exact same codon that I have. So my question is this, I don't know if you can speak to this. How do people reach out to one another? I found one other human being who has the exact same codon missense that I do. I don't know how to find this person, or alternatively, is there a body that's managing these connections and finding the patterns to connect people who have similar mutations? What is happening out there to pull the pieces of this jigsaw puzzle together?
Yeah, so we scientists, we don't have access to human data, but when we work with human samples, we don't even know what's their name, where they come from. To us, it's just a number. But that's why I think foundations are very important, because people become a member to the foundation, and then foundations may say, oh, I have this, and then foundations may say, may I share your information with the other person? Maybe they will call you. Or maybe if this data was connected at an NIH site or in a governmental site, maybe you can connect those people, they would know, but it's not kosher for patients to find other patients and it's basically that data needs to be as sealed as possible for personal security.
Wonderful. We got one last question before we go.
And a simple question. What is the best way to contribute directly to your continuing NU9 research?
Come talk to me afterwards. And please, yes, definitely. I think these are very exciting times, and time is ticking, and I have never been more excited about research because we are at the cusp of something. And in U9, with whatever experiment we do, there's not a single experiment so far that NU9 actually failed us. And Dr. Silverman is working with another colleague within the context of Alzheimer's and A-beta oligomerization. And it seems NU9 also reduces A-beta oligomerization. This may actually be a compound that reduces misfolded SOD1, TDP, and now A-beta. I mean, there may be actually an upstream mindset or big mind that controls protein accumulations, and NU9 could be one of those, and we don't know. And I am so excited. We are working day and night on NU9, and your help would be very much appreciated.
Thank you very much, Dr. Ozdinler.
Thank you so much.
Great
Thank
questions.
you.
Great, everybody. Thank you very much.
And Dr. Ozdinler will be around all weekend. So please feel free to approach her and, have conversations throughout the whole weekend with her. And I think it's one of those things that's very important that you are a part of the foundation and understanding what is it that we're trying to do, and that is connect you guys and to connect like numbers and similarities. As you guys see from the name tags that you have, those kind of denote what gene mutation. So that's what we're here to help you guys do. So thank you all for your time.
All right. Okay, guys, just a simple couple of notes here. We're moving right along really well. Remember, lunch will be here in a little while in this room. That's always going to be a good thing. I also want to talk about some of our sponsors. We've had a lot of people that help us bring this event to you and get all it's such a big program. We need a lot of sponsorship to help us. And we really want to say thank you to Chris Brocchini. She has been a huge supporter of the foundation for many, many, many years. And so we're really excited that she is still supporting us today. We'd like to say thank you to Matt Harden. We also like to say thank you to the Collin Foundation, the Your Medical Store, which you guys are really going to dig. Saturday evening when we start doing auctioning and a bunch of other kind of fun stuff with that. You're going to like those guys. Cadence is here, and they're an exhibitor out front. We also have some anonymous donors who are anonymous, but we still love them, and they help us out, and we're really grateful for their efforts in taking care of us. Chick-fil-A, you guys saw those little cards in the front of the pouches of your thing? By the way, if you drop any of those, you're probably not going to get them back, because I know there's a lot of people that are going to like to get a handful of those guys, okay? We want to say thank you to Mary Kay Robinson, who helped putting together all the potato pants. She sewed those and made those for us for this event. That's really, really nice. So I just wanted to give a quick little shout out. I'll do some more a little bit later. We're taking a couple seconds. Remember, like Norma said, the doctors and the presenters are here in the building. They are amongst you. So feel free to talk with them over the next 36 hours or so. They're here. They like to talk. They like to tell you about what they're working on. It's really neat to have this really close partnership with the people providing the science to you. Outside of that, we are just going to keep moving right along. Coming up next, here we are. Dr. Fakhari is our next presenter, and here he comes.
Hello again to our virtual listeners. We're glad that you're with us. And during this presentation over the next couple of days, I'm going to have a guest, Mr. James McLaren, who many of you know, Linda LaFontaine, who's very involved with the foundation. James is her husband. And he's going to work with us in between discussions to share a little bit of his insight on the research and information that we're hearing so that we might better understand that at home. So, James, any thoughts from what we've just heard?
So I just wanted to talk a little bit about a couple of concepts the doctor spoke of and make sure everybody kind of understands what's going on. So when we talk about dominant and recessive genes, everybody inherits two genes from each parent that kind of do the same thing. How does that work? Well, sometimes one of those genes is dominant over the other and the other one does nothing. Sometimes that doesn't occur. So for example, if you have brown eyes, you could have inherited one brown eye gene from one parent, one blue eye gene from the other parent. If you have blue eyes, the only way you can have blue eyes is if you have both copies of the blue eye gene. That's what she's talking about. Proteins, the genes are maps that make proteins. The proteins are like little machines inside the cell that do stuff, that act on various different molecules. And they kind of flex, they kind of move when they do that. They wear out, they get broken down with time. And there are proteins in the cell whose only job is to wipe out proteins on a regular basis, essentially as they get worn out. And that seems to be a big factor in HSP in particular, is these proteins that break down degraded proteins themselves are the ones that are defective. A protein landscape, like the genes make the proteins, each protein on its own doesn't necessarily do something. Sometimes they build a bigger conglomeration with other proteins and they sort of act together. And that's important because we have these many different mutations that seem to cause essentially the same disease in HSP. So it makes it hard to find a way to treat it, basically. And so Dr. Oz wants to understand how these things interact with each other so we can see what the problem is, basically. And when we talk about a druggable target, that's what kind of ties into that protein landscape. We want to figure out exactly what these proteins interact with so that we can find a drug that kind of fixes the problem. Just quickly, maybe a mouse model and GFP. Mouse model is a mouse that exhibits the same mutation or a similar mutation GFP is actually a molecule that causes a cell to glow in the dark, basically, so that you can see it. And it's a way to pick out a cell that has picked up a mutation versus another one that hasn't.
James, we're gonna pause now and head back to the stage. Dr. Fakhari is going to be talking to us, particularly about the work with the Spastic Paraplegia Foundation Centers of Excellence over the past year. James will be back with us, and we'll pick this discussion up again later. We'll turn to Dr. Fakhari and hear what he has to say.
There's still a huge gap between how much we know and how much we can translate into actual therapies, and this is for a number of reasons. So first of all, I think for one reason, we've learned a lot about rare diseases through genetics, and I think that's part of why this blue curve is really taking off. But at the same time, we're really limited by a couple of things. For many rare diseases, we lack appropriate disease models, we lack reagents, and on the clinical side, we often lack deep phenotypic and natural history data, and that has really held us back in many ways. But going forward, I think what we are starting to see is that there's change happening, and one driver of this change is the fact that we can diagnose genetic rare diseases earlier and earlier, and this is in part because next generation sequencing-based testing, so the latest generation of genetic testing, is becoming cheaper and cheaper, and therefore much more available today and tomorrow than even just a year or two ago. Next slide, please. So the fundamental challenge that we face as a community, as scientists who work on rare diseases, is that we can identify the unmet clinical need, but there's a huge gap between the unmet clinical need and clinical care. And just to give you an idea of what the process of developing a therapy looks like, I'll show you this little schematic here, which, if you click one more time, shows you basically how we go from identifying a disease, starting all the way on the left with the phenotype, to getting a therapy approved and getting it to market. And I'm listing here the highlights of like every step of this pathway. And at every step, there's a unique challenge. So when we think about creating therapies, we have to think about where are the gaps in this pipeline? How do we bridge those gaps? And this is for every individual type of hereditary spastic paraplegia, there might be unique challenges, but this is the framework that we have to think in. And this is what our research is geared towards. So next slide, please. So our group really has taken on two areas. One, developing mechanism-based treatments. We define those as disease-modifying therapies that are targeted against a specific molecular structure implicated in disease pathogenesis. And we're modality agnostic. We work with small molecules, we work with gene therapy, we would work on ASOs or RNA interference if the right case arises. At the same time, we have to be ready for these therapies, and that's something that we look to establish through a program for clinical trial readiness. We define clinical trial readiness as a state of having validated clinical research tools to the point where you can design efficient clinical trials. And I'll come back to that at the end, why it's so important to be efficient in the clinical trials that we start. Next slide, please. So I'll show you some of the efforts towards clinical trial readiness. Next slide, please. One of the key pillars of our program is a large natural history study. This is something that started very small. It started in a clinic room with a single family 5 years ago. And we have since built this platform where we meet with families with children affected by hereditary spastic paraplegia and conduct a detailed assessment of clinical factors and biomarkers. And if you could play the videos, please, there are 4 videos. Just to illustrate you, one of the challenges that we face is that although we think of hereditary spastic paraplegia as one group of diseases, the symptoms across the age span and across different subtypes can look vastly different. So we had to adapt our study to that. You see at this point, this is data from the end of last year, we've seen just over 500 families in our program, and we've seen many, at this point over 100, for a second, third, or fourth visit. And this body of data really is informative because it helps us design clinical trials in the most efficient way for a given disease and a given age group. Next slide, please. Now, what have we learned? We can just click through the animations here and have all of them shown. So we've learned a number of things. We were able to make some contributions to the common autosomal dominant forms of hereditary spastic paraplegia. Here we really took on the challenge of interpreting de novo variants, and you'll hear more about this from my colleague, Dr. Luca Schierbaum, tomorrow when she's going to speak about the genetics of hereditary spastic paraplegia. Much of our work has focused on autosomal recessive types, which are more common in children, and we've made a number of contributions here. One challenge that we came across, and I think we've heard about this a little bit already, is that if you take 100 children with suspected hereditary spastic paraplegia and you apply the most advanced clinical testing you have available, you find a diagnosis in about 60 to 70%. So that means that 30 to 40 of these 100 children do not receive a genetic diagnosis despite the most advanced genetic testing that we have. So to close that gap, we launched an additional program called the HSP Genomic Sequencing Initiative, where we provide these families not just exome sequencing, which is now becoming standard of care, but genome sequencing and long-read sequencing, so the next generation of genetic testing to close that diagnostic gap, and you'll hear more about this tomorrow. Next slide, please. Beyond this, however, this is all important work. We all want to understand, the clinical phenotypes, the symptoms better, but at the end of the day, we want to develop therapies. And to me, this is a perfect platform for developing therapies because although hereditary spastic paraplegia is different between different subtypes and different age groups, there are some universal features, and we can leverage these clinical universal features, for instance, progressive lower limb spasticity, and really test different types of modalities using this platform. And if you look at HSP, you have all these different mechanisms represented, and you can start developing therapies based on the mechanism represented. And I'll show you an example of that later on. Next slide, please. So last year, this started at this meeting, actually, and we got together and we decided that beyond individual centers conducting natural history studies and trying to establish clinical trial readiness, we needed a network across the country because no single site can accommodate all the patients, can really do this efficiently. And to take us to the next level, we decided to start this clinical research network that we call the Spastic Paraplegia Centers of Excellence Research Network. And at this first round, there are 10 centers who are part of this and have agreed to become part of this clinical trial readiness platform, which will advance clinical trials, but will also advance clinical care. That's the hope over time. You see, we have some geographic representation in many parts of the country, not in all parts of the country, and that's something that we'll work towards in the second round. Next slide, please. So we set out to establish this network to really facilitate and achieve a number of things. First, we wanted to build key infrastructure, and this was much of what we did the first year. We wanted to define clinical outcome measures and biomarkers across the age span, so from young children all the way to adults. We wanted to help create a state-of-the-art genomics platform for cases that do not receive a genetic diagnosis. And then really the next step is to facilitate the testing of therapies in clinical trials. And of course, this extends beyond the United States. Hopefully we can sync up and collaborate with other consortia around the world because this is essential for rare diseases. Next slide, please. So we had, and we can click through all the animations, we had 3 aims here. So much of the first year was spent on aim 1 and aim 2 to organize the consortium, develop a central research protocol. We have enrolled the first patients at the Boston site, and the goal is to enroll 100 individuals in this pilot study with SPG4 and SPG5A in a shared clinical trial protocol. Next slide, please. This is just to show you, a few weeks ago we had the opportunity to enroll our first patient. This was a young boy with SPG4 who came to see us from Canada, and who's the first patient in this research network. So now I take a step back and I'll show you what we've done for one specific type of childhood onset hereditary spastic paraplegia, and this is AP4-related HSP. Next slide, please. This is a group of 4 different types of HSP, SPG47, SPG50, 51, and 52. Next slide, please. We can play these videos. This is just to illustrate that although these are 4 different types of HSP based on the gene mutation, these children have the same symptoms. So if we click through the videos, you see that despite the fact that they have different mutations and different genes, they all share similarities. They all present with delayed development, initially with low muscle tone, and then over time with progression to spastic paraplegia. Next slide, please. So this was work that we conducted now a number of years ago, looking at the first 150 patients that we met from around the world. And we identified something that is very common, and I'm sure many in the room can relate to, which is that despite the fact that symptoms started in this specific type of hereditary spastic paraplegia, the first year of life, the average age at a genetic diagnosis was 10 years. So there's a huge diagnostic delay. This has changed in recent years. Most of the children that we diagnose now are younger. They're 2, 3, 4, or 5. But I still think that this is a problem around the country, and I think something that we look to work on in the next years. Next slide, please. The hallmark feature of this disease, not surprisingly, is progressive spasticity. There's a sequence from neonatal hypotonia, as I said, low muscle tone initially, then to spasticity. And unfortunately, from this cross-sectional data, we know that this is a progressive presentation, and a subset of children and young adults with this condition do have involvement of their arms, for example. Next slide, please. We can play through all these videos. I'm just trying to make the point here that although we've had a chance to meet so many families from around the world with this rare condition, it really gave us a couple of challenges. And mainly the main challenge, I think, is that the symptoms do have a spectrum. There's a spectrum from mild to severe, and there's also the factor of time and development. So studying this in children is much more challenging because this disease, unfortunately, unfolds at a time where these children are developing and learning new things, and so it makes it very hard to measure things. Next slide, please. One approach that we've taken is to do a very detailed characterization, not only of clinical factors, but also MRI images. We identified a signature of this disease that can now help diagnose this condition even without genetic testing. Next slide, please. And these are the most common symptoms. So even from this first cross-sectional analysis, we were able to learn about the disease spectrum and define the disease spectrum. But that's just the first step. This allowed us to then design longitudinal natural history studies to look at the changes over time. Next slide, please. And one thing that we've learned, and I think this is something that is pertinent to all childhood onset forms of hereditary spastic paraplegia, is that this is likely both a neurodevelopmental and a progressive neurological disorder. There are clear signs that there's involvement of early brain development, but then there's a second phase of the disease where we see progressive disability occurring, progressive symptoms occurring. Next slide, please. We're starting to understand the natural history. These are just some examples. You don't have to understand the details of this graph. The point is here, we're measuring development. And what you can see here basically is that development over time plateaus in these children as they sort of reach their capacity in different domains of development. Next slide, please. We see that motor disability, this is the spastic paraplegia rating scale scores, increases with age as a surrogate for disease duration. Next slide, please. And we find that, and this is something that I think is very important and something where we rely very much on the patient community. Through patient-reported outcome measures or caregiver-reported outcome measures, we're able to establish which symptoms are actually relevant to families, which symptoms are actually relevant to patients. And that's important not just for us as clinicians, but it's also important for conversations with the FDA because, of course, anything that we propose as an outcome measure has to be relevant to the patient and the family. So we've taken this very seriously, and in SP-CERN we have a whole battery of questionnaires that are targeted at understanding quality of life and daily activities. Next slide, please. Of course, all these clinical outcome measures are great, but what we really need is a biomarker that we can measure, something as simple as a blood test. And we've looked at a number of things. This is an example of a marker called neurofilament light chain, again in patients with AP4-related HSP. And we found that on a cohort level, the levels were increased, but we don't know yet if they're increased in a longitudinal fashion, if they're true disease marker over time. Next slide, please. Okay. So I've shown you a couple of examples of how we understand hereditary spastic paraplegia and build tools to conduct clinical trials. And now I want to switch gears and talk about therapies. And next slide, please. This is a very busy slide, but I want you to take away basically one thing, which is that there are disease-modifying treatments. And those are the types of treatments that get to the problem, the molecular problem in the cell. There are symptomatic treatments. Those are the types of treatments that we already have available that treat the symptom, but do not affect the underlying molecular problem. And it's very important to think in these two categories. And really what we want to see happening in the next year is that we shift from treating the symptoms to treating the underlying disease process. And this is not for HSP alone. This is for all sort of movement disorders that we see in our program. There are a lot of efforts in that direction, and that make me very optimistic that we'll see many of these disease-modifying therapies come to HSP. Next slide, please. So if you want to think about this systematically, you go back to the drawing board and you ask yourself, what's the molecular mechanism at play in a given form of HSP? And depending on these, and this is obviously a simple sketch and a simple representation in a review paper, but based on these four big categories, you can start thinking about what treatment modality is the right one for this type of HSP. Is it a loss of function? Is it a gain of function? Is it haploinsufficiency? Is there a specific type of variant that could be amenable to antisense oligonucleotides, for example? Next slide, please. So we can just go through these. I'm just, in preparation for this talk, I looked on clinicaltrials.gov to see how many clinical trials are happening in North America for hereditary spastic paraplegia. Not just symptomatic treatment, but disease-modifying. There's one for SPG50 right now, so we need to change that. On an optimistic note, I think there are a lot of preclinical efforts that will hopefully come to a clinical stage, and I'm listing a couple here. So focusing on small molecule drugs, there's specific small molecules that are coming towards specific types of HSP. There's a number of gene therapy programs that I know of because we're involved in it, but there might be others that are at a preclinical stage. And now really is the time to think about how do we get from preclinical to clinical for these specific types of therapies. Next slide. Next slide. So I'll come back to AP4 HSP because this is an example of how we go from preclinical to clinical. When we started working on this some 6 years ago, we knew what the gene was, but we had very little understanding of what was happening downstream of the gene mutation. And next slide, please. We worked very hard to understand the biology to the degree where we could use the biology to develop therapies. And this is just a summary slide that shows you that work over the last 6 years has delineated some of the downstream pathways of AP4 deficiency. Next slide, please. Without going into very much detail about the biology, one thing that we leveraged here was the fact that AP4 deficiency leads to a mislocalization of other proteins. And one of those proteins is called ATG9A. And without knowing anything about the biology of this protein or this disease, you can tell that the cell on the left looks different from the cell on the right. You see a lot more dark signal around that light nucleus on the right. And this is a difference that we saw in every model of AP4 deficiency in every patient-derived cell. And again, knowing that this is an immediate downstream effect of AP4 deficiency, we were able to leverage this cellular phenotype to develop high-throughput screens. You'll see that on the next slide. So we can click through this. Basically, we had this phenotype and we said, okay, how can we leverage this for therapy development? We set out to do small molecule screens. We've done a number of functional genomic screens that I won't talk about today. But really, this is to get to a therapeutic target based on a simple cellular phenotype. Next slide, please. This slide shows you basically the workflow, summarizing several years of assay development, really. But basically, now that we understand this mislocalized protein and can measure it on the level of individual cells in formats that are amenable for high throughput screening, we can start screening for molecules. This is very much what Big Pharma does. They basically develop assays and then run these large unbiased screens to find molecules that can correct the phenotype. And you see in this example 384-well plate, that you can identify outliers very quickly based on your pre-specified metrics, and then look more into the molecules that have provided a rescue. Next slide, please. This is again summarizing years of work, but basically in one of those screens we teamed up with a pharma company called Astellas. They provided us with a library of about 29,000 small molecule compounds that we screened. In this primary screen we identified 442 that were active, that corrected the cellular phenotype. Next slide, please. I'll summarize, and this is published now, all the stages that we went through after: secondary screens, orthogonal screens, validation in patient-derived iPS cell-derived neurons, which are the closest surrogate to a neuronal model derived from a patient. And through this identified a set of lead compounds, and one compound in particular that we think has favorable properties. And this program is now at the stage where we've synthesized the compounds, analogs, we've tested them, and now we're testing them in vivo in a mouse model to see if we can correct phenotypes not just in a dish, but also in a mouse. Next slide, please. So I said in the beginning, our team is agnostic to what type of therapy we work on. We want to test whatever works the best for a given disease and a given patient. And as I've introduced earlier, this is a loss of function disease. So, from this it follows that the best and most proximal approach to treating this disease is to replace the gene that is defective or missing. And that's what gene therapy is. So some 6 years ago, we teamed up as part of a larger consortium to develop a gene therapy for APOE4-related HSP. Next slide, please. One of the programs that is now in a clinical trial with 4 children treated thus far is for SPG50. I won't go through all the data, but just to illustrate the concept. This was, 4.5 years ago, was just a sketch on paper. We knew, and this is mainly work done by Stephen Gray at UT Southwestern, we knew how a gene therapy could look like, building on experience from spinal muscular atrophy and other diseases, and designed this gene therapy vector specifically for SPG50. We and others tested this vector in patient-derived cells, found that the vector was able to correct the underlying phenotype in the cells, then moved, next slide, into mouse models, and were able to show that in the mouse model, which hadn't been created at the time, created in parallel, this gene therapy vector corrected some of the phenotypes. This gave us enough confidence to move into IND-enabling studies. Next slide. And this involved toxicology studies, as is commonly required for gene therapies in mice and rats, and in this case also non-human primates, which allowed us to identify the safe dose and helped us design the clinical trial. Next slide. And we can click through all of this. This is just an example. I'm not going into the sort of the details of the science here, but just to show you that you can go from concept to proof of concept to a clinical trial, even for an ultra-rare disease, if you have the right collaboration, the right support. So all of this was made possible by incredible fundraising and philanthropy by patient advocacy organizations. It was supported by the platform that we had built for natural history and biomarkers. It was supported by the therapeutic entities that were developed at the University of Texas Southwestern and the University of Sheffield for a second program for SPG47. Then we had enough funds to really conduct proof of concept studies in different laboratories independent of each other. They gave us confidence to move into the very expensive parts of this, which are toxicology studies and manufacturing. And then for SPG50, this is in a phase 1, phase 2 study, 4 children treated thus far. We're planning a phase 3 study. So if you remember the cartoon I showed in the beginning, there's a part that goes towards approval and market access. You need a phase 3 study to get there. So we're designing this, and this is unique. I think it's never been done for a disease so rare. And at the same time, we're learning from this first clinical trial, and we look to repeat it for the next disease, which is SPG47, and hopefully for others in the future. So just to show you that you can go from concept to proof of concept to a clinical trial. Next slide, please. So with this, I'm at the end. We can click through all of these. I hope I was able to show you that there are unique challenges, but also opportunities for creating therapies in rare diseases, including childhood onset HSP. I think at the foundation, there's access to genetic testing and longitudinal natural history studies to really establish clinical trial readiness. I've showed you AP4-related HSP as an example of both a neurodevelopmental and neurodegenerative disease. I've shown you how we develop small molecule treatments through high throughput screens in a lab. And I've shown you an example of a gene therapy that made it all the way from concept to a clinical trial. Next slide, please. So with this, at the end, I have to thank the many people involved in this work, but most importantly, patients, their parents, their families, and advocates who support all of this research and come and see us in Boston, support our research studies, and all the funding agencies, including the Spastic Paraplegia Foundation, which have made this possible, which have made the Spastic Paraplegia Centers of Excellence Research Network possible, and hopefully we'll see a lot more of these therapies coming to a clinical stage. Next slide, please. So these are the studies that are open for enrollment right now. If you're interested, please reach out to us. We're very happy to tell you more about the work that is ongoing. So thank you very much for your attention.
Hi, Dr. Darius. Thank you for your presentation.
Of course.
Can you tell those of us with children with HSP if your work with the AP4 work will carry over to the other subtypes?
Yeah. So in terms of gene therapies, those are disease specific. Those are specific to a specific gene, which is a major challenge because basically you have to develop a different vector for every disease. There are some elements that you can take over, but at the end of the day, you have to develop 80 or 90 different gene therapies for 80 or 90 different diseases, and that's a huge financial challenge. This was one of the reasons why we started the small molecule program, because we realized that even developing 4 gene therapies was going to be very challenging for financial reasons. As far as we can tell, the small molecule treatment that we found only works for AP4 deficiency. It does not carry over to other types because the molecular problem is different.
Mm-hmm. Good. Thank you.
Yeah.
Any other questions? Anybody? Here's one over here. I'm going to go over this way.
Very good talk. Appreciate all the information. This is kind of a multi-part question. I don't have HSP, but still some of the work that you guys do in motor neuron disease helps all of us. When you talk about gene therapy, the number to treat is so small. You mentioned like specifically like SBG50, you mentioned 4 patients. Yes. If you were trying to scale that to get an approval for gene therapy, I want to know how would you do that? And is there a pathway or conversations that you guys have had with the FDA to even do that? Not just for that, but it would be for any disease, if that makes sense.
Yes, absolutely.
And do you share your research with, like, do you have another Fakhari in Europe or Asia or somewhere to where you guys might be able to raise awareness or get more patients into clinical trials? Thank you.
Those are 2 very insightful, brilliant questions. So I'll start with the first one, scalability of this approach. Major challenge. Major challenge for any gene therapy in rare diseases. There's a unique circumstance here, and the FDA appreciates that in the conversations that we had with them and in designing the phase 3 trial, which is that typically in a phase 3 design, you need a placebo-controlled group, which is challenging for a number of reasons in rare diseases. In AP4-related HSP, we've been able to show that the natural history of all 4 subtypes is the same. So what we propose is, in a way, a platform approach where you have a central control group recruited, matched based on the patients that are in the treatment arm for those subtypes where a gene therapy is not accessible yet. that will serve as the control group for all the types where gene therapy will become accessible. So it's a long shot, but we're hoping to, even with a small number of patients in the phase 3 trial, to be able to show efficacy and then move to approval. As I said, this is the first time that anybody has tried this, but it's a unique circumstance for the reason I mentioned, that you can have a control group without the ethical or recruitment issues that you typically have in rare diseases. Second question about international collaboration, absolutely. This is one of the reasons why I decided to get organized in North America and in the US, because there are similar networks in parts of Europe, in Canada, in Japan, and Australia. So we've already had a lot of traction and conversation with those partners, including in Germany and South Korea. And really the goal is to synchronize this as much as possible, so that we're all conducting studies in the same way and can learn from it the same way. So absolutely, it's one of the big motivations to pursue this network. Yeah.
Okay, Dr. Fakhari, we have a question straight up center ice.
I'm at the opposite end of the people that you're working with in terms of the disease. And I'm also one of the 35% of people that do not have a gene associated.
Okay.
And I've gone through at least the NIH screening, and then I went through screening with Invitae last year. And all, I'm still, as my purple thing says, I'm unidentified. So, and I sort of feel a little lost because I'm unidentified.
Yes.
Yet I've not heard anything that makes it really important other than for the whole community to go further. And I'd like to get your thoughts about, because I don't think it's going to affect me, but will it affect my children, others, the whole research area? How far should the average person with unidentified HSP go to, in terms of all the different variations that are relatively expensive, but they are they important for the whole community to be able to have that information?
Thank you for that question. It's a brilliant question, one that is very, very relevant, I think. The short answer is yes, we should try. We should make every effort to identify a genetic diagnosis. The reason is that this is an entry point. It's an entry point for a lot of things. One, it's an entry point to a specific community. Two, it's an entry point into research. Three, it's an entry point, if it's a known gene, to specific counseling, anticipatory guidance, genetic counseling when it comes to the family and children. So absolutely. This is a rapidly moving field. The things that we talk about in terms of genetic testing today didn't even exist 2 years ago. So I'll encourage you to connect with our team. We can direct you to similar efforts for adult.
Because the reason I'm asking the question, there are companies out there,
Yeah.
and it's very hard for the consumer
Yes.
to be able to judge A, B, or C, which is the best way to approach that question.
Yeah, so I think this goes beyond clinical testing you would do with a company. So there are a number of research studies for HSP that are looking to apply research-based genomic testing, and we can certainly do this for any individual under the age of 30, but we have partners at the University of Miami who cover the whole age range. There's a program at the NIH, there's the Undiagnosed Disease Network at the NIH. So there are these 4 resources, at least from the top of my head, that I would recommend. And this goes beyond anything you would do with a company.
Right, okay, so maybe I'll send you an email. I think
Absolutely, and Dr. Schierbaum from our team will speak about what we do tomorrow, I think, so that'll be a good opportunity.
Great, great, thank you.
Thank you.
Dr. Fakhari, Norma's got a question in the far right corner.
Hi, thank you so much for all your information. I have a question, but it's not about the research. With working with children with SPG4, have you found that in almost all cases that children who are starting to show signs of the disease have delays or show developmental delay early on? Or can they be completely, I don't want to say the word normal, and then start showing signs? Do you always almost see the developmental delay?
That's a great question, and I would say both is possible. So the short answer is more often than not, we see at least subtle signs of motor delay. But we've also certainly made met families where there were no signs of early delays, early motor delays, and the onset was later. So I think there is a little bit of a spectrum. We have certainly learned that children who have de novo mutations, for reasons we don't understand, typically present earlier, but both scenarios are possible. In the familial form, the second scenario of maybe only mild delays or no delays is more common than in the de novo form. Yeah.
Hey, do we have any more questions? I see one. Norma, your side of the room is, come on, girl. All the way up in the front. Keep coming. Okay.
I have a couple of questions.
Yes.
I'm new to all this, so the part about where you said like the treatments are, treating the symptoms, and then you've got the underlying treatments. Underlying treatments, I'm assuming, is like the gene therapy.
Correct.
And the systematic treatments would be, like you've got baclofen, or you've got physical therapy, you've got different things to treat speech or the behaviors.
Exactly.
My question is, can you explain? And I have another question after this, but what gene therapy really is? Like, what does that actually mean for gene therapy? Because the other stuff I understand, the physical therapies and all those things, but what is gene therapy?
Very, very happy to. So when I use the term gene therapy, really what I refer to as gene replacement therapy. So very simply put, this is a way of replacing a normal copy of the mutated or missing gene through different so-called vectors. And most of these vectors are particles derived from viruses. So the best example I could point you to is the gene therapy for spinal muscular atrophy. But in essence, it's a technology where we use viral vectors to deliver a healthy copy of the gene.
Oh, okay. And then also, when you said, that I think it was the SPG, or the 4 is the one you're working on, it had the neurodevelopmental disorder and the neurodegenerative disorders are connected with that one. How do we find out for our specific, is there a way to find out if our children who have a different HSP have that? Do you know what I mean? Because I'm looking at the definitions. What does that really mean? And those words, what does that mean? How do we find out if those are the things that are connected with our gene?
Yeah, it's a great question. I think we learned about this through examining a larger number of patients. So there are specific things where we can definitively say this is neurodevelopmental. I'll give you an example. When we looked very carefully at the prenatal ultrasounds that were done as part of routine prenatal care, we identified that in a number of patients you could already see certain structures of the brain not having fully developed. And that's clearly developmental. It's not something degenerative. So that's an example of a symptom or a presentation that probably dates back to the very early brain development. And so the short answer is, I guess, is that you have to look at a lot of patients very carefully to identify which symptoms are related to changes in neurodevelopment and brain development, and which changes are truly a surrogate for a progressive process.
And that clinical trial that you're doing, the registry one, is that to connect people, like also who have the different HSPs? Will it connect people together?
So we
families?
always make that effort because we realize that many of the forms of childhood onset HSP are so rare, and it's a very, very lonely place to be in. So if there's mutual permission, we always try to connect families, yes.
Dr. Fakhari, right here.
Yeah.
Just a quick question about that study, the natural history study. It's for patients with a childhood onset. So if we have an adult child, my son is 26 now, but he did have a childhood onset. Do you still want those patients involved in the study?
Thank you for that question. We do enroll patients up to the age of 30.
Okay,
that has logistical reasons, but 30 is our cutoff.
Okay, thank you.
Okay, Dr. Fakhari, I got another one on your far right. You got other way. Oh, way over here.
Hi, good morning, Darius.
Good morning, Oscar.
I have a question about gene therapies. I've talked to some leading gene therapy companies here in the States, and we're interested in that too. And the data you show for AP4 is brilliant. But the animal studies are really, most of the animal studies are done in mice.
Yes.
And then the mouse brain is really different from the human ones.
Yes.
So a lot of companies have concern about distribution of the viruses. maybe you touched on it. Like, I don't know, for the clinical trials, what are they using as in terms of a capsid of the virus, or what's the volume of the virus they're using in the human trial? Are they doing intrathecal, or are they, what are the strategies that cover that kind of concern?
Yeah, you bring up a very, very good point, and I completely agree. I think biodistribution is a major challenge for central nervous system diseases and the current generation of vectors. I think the first step is to identify what is your target tissue. And without really having a lot of proof for that, for HSP we say you need at least the cortical spinal tracts, because we know that's the structure in the brain that, if affected, can give rise to spasticity. Okay, that's an assumption we make. We have no proof of that. So then you target your tissue, and then you have different capsids available. The capsid we're using here is AV9, and that's just simply for the reason that there's an established pathway with spinal muscular atrophy. The route of administration for SPG50, for the SPG50 trial, is lumbar intrathecal. So it's a single lumbar infusion. And building on the experience from another clinical trial, the giant axonal neuropathy trial at the NIH, we know that biodistribution, this is published now in the New England Journal earlier this year, we know that the distribution is pretty good in the spinal cord. It's good in certain parts of the brain, but we really don't get the deeper brain structures. And that's a major problem. So for the second trial, we're already switching the route of administration. So we go from lumbar intrathecal to intracisterna magna. Will this be the best way to do it? I don't know. And the challenging part, and you brought up the mouse model, is that really none of the preclinical models predict that. We have to do the clinical trial to learn that.
Right, thank you.
Hello, Dr. This is Shamal Shah. My daughter does have a de novo mutation. She's a 13-year-old right now. We had seen the symptoms since probably 3 years old she was, 2.5, 3. She's around 5.5 actually. It was Dr. Blackstone's research that found the mutation. She has SPG3A ATL1 mutation. My question is, hearing a lot around the gene therapy and what we are reading as well around that, one of the concerns we have had over the years has been just the scalability of the treatment path. Most of the readings has been around very targeted, approach towards it, given the spectrum that's very broad in this disease. What's your kind of thought around progress that we can expect over the next 5 to 10 years in this direction as well? Given the doors it opens up, but some of the scalability challenges that exist.
I think the scalability issue is real. It's certainly real also for SPG3A. I think we need to have some successes. This is what happened with spinal muscular atrophy. I think the same conversations were happening before the gene therapy was successful. And if we see a success, that will, incentivize and de-risk the development of these kinds of therapies. I think the challenges are real. Don't get me wrong. I think getting a gene therapy approved for a rare disease as rare as this is a huge challenge. But I think we have to, work in that direction, hopefully see some success, which will incentivize new programs.
Okay, everybody, that sounds really, really great. Great questions, great answers, of course. Great science, it's wonderful.
And Dr. Fakhari will be here, so you guys have a chance to talk with him and his staff. Where is Amy and Dr. Luca? They're back here, so there's plenty of time to talk with them, so make sure you do that.
Okay, everybody, this is your lucky day because we have a little bit of a break right now, so you guys can stretch. I always say we're going to give you a 5-minute potty break, which really translates into about 3 hours. We know how it works, at least at my house, that's how it goes. So we're going to give you a little bit of a break because we've got some exercising and some stretching and some really great stuff coming up at the top of the hour. So guys, take a stretch, get your muscles warmed up, and we'll see you back here in just a little while, okay? Thanks, everybody. And real quick, while you're out there, make sure you guys go to the merch table and buy those raffle tickets. There's a lot of great stuff out there. Go out there, check it out, open those wallets, have some fun. You never know. You never know. You can't win if you don't play.
Hello again. You've heard some great speakers this morning. You hear Dr. Ozdinler talk about how important the research is that she's doing, and particularly with NU9. She's talked about how time and money are so important, so contributions that go toward that work are extremely important toward moving toward clinical trials that can make a difference. And then Dr. Fakhari, with his discussion about all that he's working on, he's the leader of our Centers for Excellence and Research Network, so a lot of exciting things going on there, and we want you all to know all about those things. But with me right now is it has been so exciting. We just met Dr. Lauren Leiva this week. She is with Exercise, and she's going to tell you in just a moment what that's all about. But you're going to be seeing her in the next session kind of talking to our membership about how important it is to move, how important it is to be thinking correctly, and moving yourself in a direction that can keep you healthy and on the move as best possible. So, Lauren, I'm going to call you Lauren. You're my friend. It's great to meet you. Great to see you. Share with our visitors what you do and what you'll be doing with us here this weekend.
Thank you for having me. So I'm Dr. Lauren Leiva, owner and founder of the Exercise Center in Tampa, Florida, and maybe soon in your area. I heard what you said. Another speaker talked about time and money. And for me, I know time is the most valuable resource on this planet. I think even way more than money. My mother's no longer here, and she always told me you can't take it to the grave with you, right? So each moment on this earth is just a blessing for me. That's my perspective. And that's how I built my clinic, is to achieve results faster than traditional therapy. So it's a biohacking neuromuscular wellness clinic. And I think when you came in, you could see it's very different than most clinics. I was in an electric suit, shopping around for the best technology. And that is also what is a little different about my center. I think that many clinics will get into a perfect wheel that they really enjoy, they can implement really well. But just like our iPhones, everything is updating consistently. So my clinic is based on learning something new every day and being on the edge of medicine with modern technology, but not forgetting traditional medicine, ancient wisdom and practices, respecting everybody's values. So that's how I built my center.
Dr. Leiva, as we think about what you'll be sharing here with this group today, we're talking with folks at home who are with us by virtual feed. If you could tell them right now one thing that they ought to do today and make a daily commitment to doing to maintain their lives, their physical circumstances, and enjoy life to the very fullest, what one thing would you tell us all to do?
Well, because I'm a little bit of a joker. I know you met me. I'm gonna say a little bit of a joke while I tell you, but I would say squeeze your butt. So squeeze your butt and think about something you love. But when you squeeze your pelvic floor and your muscles, whether you are bed-bound, wheelchair-bound, or you walk, often we might sit for more than 20 minutes. And again, even more so, this population, if you are wheelchair-bound or bed-bound, sometimes you might not get a lot of activity down there. And even if you think you can't, I used to have a colostomy bag, and the doctors told me I will never go to the bathroom normal again. And I don't have a colostomy bag anymore. I do go to the bathroom normal, and I don't let other people define me. So that's what I would say most. Don't let others define you. And I know I didn't answer it the best, but I would say squeeze your butt and think of something you love.
Well, that's pretty simple, and that's really easy to remember.
Yeah.
And we want to thank our Florida group down here as we worked on this conference for the past year. They helped in so many ways, and as Norma reached out trying to find local businesses, local people that would have an interest in working with us and supporting the work in this conference, Dr. Leiva, you stepped up, and as soon as we met you and began to talk with you, we appreciate your genuine interest. We know you have a business, and we know you run that on a daily basis, but when we stepped into your clinic a couple of days ago, you're right, it was a very different kind of clinic. Seeing some of the machinery and some of the technology that was there to help analyze what might be wrong or systematically need to be improved within a person was very interesting. And so we appreciate with your busy life and busy schedule, you're taking the time to work with us in this foundation and in this conference. And we look forward to what you have to say to these folks out here. And I'm hoping as you talk to them in a few minutes that we see them moving a little bit, stretching a little bit. And thinking more about how we can be a little more mobile and a little more active, both mentally and physically.
And if you see their heads going up just an inch, you know what they're doing.
Well, thank you, Dr. Leiva. We appreciate you, and we look forward to working through this weekend with you. And folks, in just a few minutes, she'll be speaking to this group, and we'll learn a lot more. So thank you so much for being with us.
Thanks so much. And if you're online, you can follow me at theexercisecenter.com or Queen of Biohacking.
Great. Thank you again. And I want to take just a few minutes to thank all of our sponsors. And they're all in the program, and I know you guys can look at them, but I want to take a moment while we have a break just to mention them specifically. Chris Brocchini, who has been a major sponsor of this conference for a number of years, and she normally tries to be with us, but she couldn't be here. And we want to say thank you, Chris, and we miss you, and hope all is well with you. Matt Harden, Conlin Foundation, your medical store, Cadence, who's a shoe exhibitor outside, and it's exciting for those of us who have difficulty walking, seeing their product, and we'll be interviewing them so you can learn more about that a little bit later. Joshua Cochran, Toby, and Becky, the Cochran family. We've had a number of anonymous donors, the folks who are here, the bags that they're carrying, which are very nice bags, were contributed by an anonymous donor. And so we thank our anonymous donors. Mary Kay, Chick-fil-A, the Karen Gerlasio, Captain Cookie Beads, First Community Bank of the Heartland, the Tampa Bay Rays, U.S. Youth Soccer, Canine Companions, and Glow Natural Wellness are the sponsors who have helped us here put this conference on. And Dr. Lauren and Exercise, we appreciate what you guys are doing to help us with this conference as well. So we're going to take a break for a few moments. We'll be back with you for some exciting time with Dr. Lauren. So take a break, and we'll see you in just a few minutes.
Bye.
Just saying, okay, so you better be focused and paying attention, or we're not going to feed you, okay? There you go. That works every time. That gets everybody going.
Sorry, guys.
So we're getting ready to go here. Hey, I just wanted to remind you guys about some other little things we got going on. Remember to go out to the merchandise table. Lots of good stuff. Get those raffle tickets and all the other good stuff. Check out all the things we got for auction, and everything is going to be wonderful. So without further ado, here we go. Dr. Lauren Leiva. Here we go, everybody.
Hello, hello, thank you. I think I have 2 microphones going. Sounds good. How's everybody doing? Ooh, ooh. Can I get a ooh, ooh? Nice. Most conferences I go to, they'll have someone with like a cowbell, and they're like, if you're talking, go outside the room. If you want to pay attention, listen, right? So if you're talking, go outside the room, and if you want to pay attention, listen, right? So we get to move at the end of this talk, which will be great, and then we get lunch. So if you're hungry and you're looking for some of that, it's coming around the corner. If you guys are standing on the perimeter talking, hey guys back there, ooh, ooh. Hey ladies talking, hi, we're starting a talk. So I always say if you're talking, you can go outside. I'm Dr. Leiva, owner and founder of the Exercise Center in Tampa, Florida. I'm gonna go, woo, thank you. So my talk is mind over matter. And I know that some people's minds are just, it goes in 100 places, and that's why sometimes it's hard to keep quiet during, talks, but it's just respectful for other people who really want to hear. Oh wait, which way do I click it? This way? There we go. So who am I? I'm a mother to these beautiful children here. It was Father's Day, that's my husband. We've been married 17 years, and I am a survivor. I've survived a lot of things, and my main message to you guys is a lot of times I was defined by a diagnosis, and it really structured my whole entire life, what I could do, what I couldn't do, what I could eat, and we don't realize how that affects our spiritual world and all the decisions we make based on what we think we can or can't do. I have different receptors here. So I'm going to give you guys my card, and this talk is a little bit faster than what I'm used to, so you can hear it in the next slide. But again, I am a doctor of physical therapy. I also went to USF Medical School. My mom had a stroke and lung cancer. Thank you. So I left my clinicals to help take care of my mother. And then my son was born, and he was born 2 months early. He didn't walk or talk. He needed braces on his legs to stand up, but really he still chose to crawl. And we're gonna get into that later, but I just want you guys to know I can connect with all of you on different levels. We all have some type of thing we're working through, but I say our life brings us to our passion, our purpose. And sometimes it's very, very painful. But if we can take lessons from that, that's what we're all here for, right? So when I was younger, I was diagnosed with Crohn's disease. And I think that's the easiest way to diagnose something because you could put it on paper, there's symptoms. What I realized later on in adulthood is that there was a lot of abuse and a lot of issues getting stuck in my tissues. The things I was eating, the toxic environment, all of the vitamins, lack of vitamins, all of the medications I was being given, infusions, and everybody was practicing medicine on me. And when I went to medical school, that became a joke. So I'm writing another book, From Patient to Provider, but we're all here practicing medicine. Just act like the smartest person in the room. And that's when I realized, whoa, I have to get into the education centers to help change the way doctors are thinking, because my mother was actually killed in 2019 by medical malpractice. And to do no harm first is my passion. And every one of you is so important. I think anybody that is going to treat you, diagnose you, be part of your plan, should truly care about you. I mean, you should feel it when you walk in. I brought one of my past clients, who's now my friend, and my assistant, and I'm not telling her what to say, but I think she's here because she knows I love everybody, and I love you. Again, so practicing medicine, I had no insurance. I came from Holocaust survivors. I just was on free lunch. Everybody just clocked in and clocked out and kind of did what the doctor before did. Nobody really wanted to take on my case and do something new. It's really easy to just copy what the last person did. And that's kind of what's wrong in the medical field, especially if you don't have insurance. Prescribed meds. I was given Ascal, Flagyl, Remicade, Humira, 6-mercaptopurine. I lost all my teeth from intubation, but I was losing my dental, care along the way. My enamel, my hair was falling out. I got osteoporosis. There was nodules in my bloodstream, so everything caused something else. And again, I just felt lost in the system. And that's not even the normal stuff that we're born with, right? Kids, my brother had an emotional handicap, a mental handicap as well. And because I was smart and intelligent, my parents kind of said, you can make it, which is why I stored it all inside. And my brother was the one that always went to doctors and healthcare professionals. So it's important to also focus on all your children if you have a child that is affected, because sometimes the child that is not affected gets neglected. So that was kind of me. Again, they call it a golden child syndrome, I found out later. So you look good, you feel good, just keep moving. And again, if you don't have something that's going on the outside, people don't realize everything that's going on the inside. And anybody knows when you have a diagnosis or you're really battling things, sometimes we'll really make ourselves up on the outside. We'll do our hair, our makeup, we'll get really cool clothes, and maybe we'll go out for an hour and be amazing, but then we go home and we crash and we do nothing, and our family will be like, but you did that, but you were able to do that with your friends, why can't you do this? So we'll get to the spoon theory later, but I still wanted to be an athlete. I was an ice skater. I did modeling, but because I kept pushing myself during short periods of time, people thought, you have no diagnosis, no disease, no problems. And it's almost discouraging for someone who does have a diagnosis, because if I do something, nobody will help me later. So it's kind of a, twisted situation there. Even when I'm in the hospital, if you're a medical doctor here, well, if their oxygen level is not at this, they don't get an oxygen tank. I used to whisper to my patients, just hold your breath, right? Because we are waiting for people to get out of a safe window to help them. And for me, I want to optimize all of my patients' lives, whether it's through physical motion, spiritual practice, but mostly your mind. Your mental state is everything. Navigating rough waters. So having an invisible illness, I dated the same person from 13 years old till I was 20. Because I went to the bathroom a million times a day, and they saw me go to the bathroom on myself, and they saw me throw up, and they saw me do everything. So again, it can affect your relationships. And these are things we don't talk about at the doctor's office. Maybe some of you out here don't even like your spouse. No, I'm joking. But they're taking care of you, right? But this is the life of someone who has a chronic condition. You need people, my mom said before she died, which we'll get into that, accept help and also ask for help. Because I don't want to ask my husband for anything, because there's been so many periods where he has to do everything for me. But it's important to be able to ask for help and to be in a situation where you are allowed to be yourself. I'll just switch away from this for a second. They had a study come out. What is the strongest emotion? There's an emotion of love. There's an emotion of hate. And these are the two strongest feelings they say come out of a human being. You can measure someone's heart and emotions now, a bioenergy field. It's not hocus pocus, it's not just for yogis. But they found an emotion that is 4,000 times stronger than the feeling of love or hate. Can you imagine that? And do you know what that is? Can anybody guess? Or even try to guess if you want. If you want, I'll give you a free bottle of Qualia. Depression. Huh? Depression. Good one. You get it. But also I have I think about 20 bottles. The first 20 gets it for free. So you're one of them. Ooh, ooh. It's not depression. It is authenticity. So I was in a conference, and I was at a dinner. Does anybody know Joe Dispenza? Well, whatever, he's amazing, go look him up. I was at a dinner with him the other week, and we were talking about this, and someone said, no matter what setting you're in, whether I'm teaching a women's ceremony in the woods, where we're all being wild, or I'm in a event like this, I'm the same person at all times, and that I'm authentic. And then when I found out this study, to me, that's the best compliment ever, because I think in a world we become toxically positive, where we say, nothing's wrong, but sometimes things are wrong. But it's the ability to keep moving forward. Any movement is better, mind, body, or soul. So, anyways, I was secretly getting sicker. I had disability accommodations. I went to graduate school, doctorate school, all of that. Every time I always had to explain my disability accommodations, I always had to activate a protocol for someone to let me go to the bathroom, which made everybody in the room aware of my situation, so it was never really private. It made me feel isolated. So what happened after the diagnosis? I'm gonna try to go faster. Again, just what happens with all of the medicines, bloating, bone loss, pseudo-rheumatoid arthritis, pseudo-psoriasis, depression, insomnia, increased stress, malnutrition. There's a QR code in the corner of all of my slides, so if you guys want to scan it throughout, there's a lot of stuff on my desk, a brain tap. I'm going to go into it later that helped save my life, but everything is with this QR code, so that's why it's there. We are ready for the YouTube video. So I have a 4-minute YouTube video that's going to say with pictures everything I said, because the pictures speak a million words. Because you might think I'm so cute up here, I want to show you my story, because it was pretty terrible. Do I press a button? Where's my guy? He left? The guy? No, it's a YouTube video. He saved it, Harper. Harper's fired. Right? Where'd Harper go? It's on your tab. Go to Safari. He saved it. It's not there. Go to Safari or the internet. Is anybody here with the Spastic Foundation? Can you find Harper for my video? Oh, there she is. So that's good. They'll find it. He saved it on a window on the internet because he could not access it there. So maybe go to the history. I don't know where Harper is. But my video just shows my hospitalizations. My rib cage used to be attached to my pelvis. So I walk like this, holding my IV pole. And my hips were turned inwards, and I had pressure ulcers just from not having movement. I would fall in, yep, there we go. It was so hard for me to talk about. I would cry. So what happened? So you could just listen, and then we'll go fast. This part of my story I pre-recorded because it's an emotional journey. A lot of our paths are from pain to purpose, and mine was a very painful one. The hospital cut my intestine while I was pregnant. I ended up with an abscess that was 18 by 19 centimeters right on top of my uterus. They recommended that I have an abortion, but I explored other options, and my spirit knew that I was meant to have this child. I ended up on TPN, which is all of your nutrition through your bloodstream. This was my last hospitalization. You can imagine how hard that was during pregnancy. I was given tons of IVs, which made my face really inflamed. I had my son with me, currently now 15 years old, and he was a great support system to keep me motivated and inspired the whole way through. Motivation and support is so important in your times of need, especially if you're hospitalized. You can see bruises here from lack of mobility. Nurses and staff are supposed to move patients every 2 hours with range of motion exercises, and this is rarely done in the hospital. That's what we're gonna do soon, Not only did I have to replace all of my teeth, but I ended up cutting my hair, if not losing much of it, from just lack of nutrition. This was a very special day for me. I ended up getting out of bed, even though they said I wouldn't. I walked down the hallway with support from my dad, and again returning to having a support system, having an advocate. Even if you are not married or you don't have children, it's so important to make sure you have an advocate for when you're going to the hospital, because things can get lost in translation. You think you're the biggest whale in the sea, but there's many, many minnows, as it was explained to me. I guess it's... And a lot of your orders get lost, and just You ever been to the
translation
hospital
of the
and not
wrong
get
order.
medicine,
So
not
having
get what you
someone
need?
there checking on you is vital for survival in these critical situations. This picture is a very moving picture to me. It was very analogous to my son growing outside of the hospital while I was inside. And this too motivated me, and I knew I was going to get out of the hospital one day. I ended up being transferred through 3 different hospitals, and I ended up being saved at the Cleveland Clinic here. I would walk down this hallway every day to look at this structure being built. And when I was admitted, there was nothing there. And as I stayed, I believe I was there almost 4 months, I saw this high-rise be erected. And it was just very emotional because I knew that was my son growing on the outside of the hospital without me. Returning to my yoga practice consistently, staying mobile, trying to do what I can whenever I had the energy. And from that moment on, every day I was stronger. Every day I did just a little bit more. And the secret is having the discipline to strengthen your weakest links. Here's my family that was always there for me. Again, that support, knowing my child was safe. This was my stepmother who was taking care of my children. If it wasn't for her, I don't know where my mind would be. This was my stomach after I ended up with a colostomy bag. And that's my mother. She actually was killed by medical malpractice in 2019 from an overdose on blood thinner. So again, it's just very important to always have support, have an advocate, and really use all of your resources to get a second and a third opinion. My story to healing from these pictures and to where I am today, I ended up, opening my clinic and with all of the tools that I used. So here's a picture of both my children. They were both born early, and why I'm a physical therapist. Physical therapy modalities really took my kids from being quote unquote delayed to quote unquote advanced. So follow me for the rest of this story.
And you could,
And not only did my kids get healthy, but as you can see here in the next series of pictures, so did I. They say that you cannot.
You could switch it. It'll keep going. Thank you. Thank you. So all of my patients that are above the age of 65 tell me, honey, you should have people read your story. They'll respect you more. So thank you guys. That's why I put it there. I've just been through a lot, and I'm on a documentary coming out on Amazon, and the guys that were recording it said, you're an anomaly because you're exhibiting joy, and nobody would have ever known any of that. And that's one thing my mom did. She said, don't give it to anybody else. So I'm now going to tell you how I got better, and then we're going to move a little bit. But again, for me, I'm a yogi, mantras. Your mind is very important. I looked at that building, and I saw how much time went by. Time's the most valuable resource while you're in this human body, right? So that's what really motivated me. I tell all my patients, print out pictures, of a beach, of a vacation, whatever it is that motivates you, put it around your place. So they say, again, once you have an injury, you don't build back stronger. I even work with UFC fighters, NFL athletes, and volleyball players, all of that. So when they injure themselves, they're always told, you're never going to be back to where you are. And I'm like, we're going to be way stronger. We're going to prove them wrong. And 9 times out of 9, correct. So mantras, mind, body, spirit. I'm going to go fast. These are the things that are on my wall. So all my patients, I put them in timeout sometimes. I'm like, go read number 4. Go read number 2, whatever it might be. But my 9 months got me to those solutions. Meditation. You'll see at the table, I have a brain tap. And if I had one device in my clinic, it would be this device because, again, I was in a very negative space. And if we even go back before my original diagnosis, I had sexual abuse, physical abuse. I never talked to anybody about that. Did I really have Crohn's disease, or did I just store it all? So these are things doctors don't talk about. Often, who wants to go there? You're not a psychologist. It's out of your scope. But maybe if you fix that, you wouldn't have to keep cutting and prescribing. It's important. So I'm not into talking with a therapist as much as I talk on stage. That's why I made a video. Sometimes it makes me feel like a
abused dog that could be kicked in a cage, just reliving all of that. So this, to be able to put this device on my head and go to sleep every night is just so magical. Before this device, I would say I slept most 2 hours at a time. I would wake up. Sometimes I would go to the bathroom 500 times because one bathroom break took 500 times to get out of me. And then I just woke up so exhausted, coffee, Adderall, just stimulants to keep going. And then I was the first person in Florida with my marijuana card. So then I did a lot of, let's go back to sleep. And it was just, it's not good for you. When I started working with veterans, I was in so many talks about how sleep is the most important thing you could do for your recovery, your nervous system, for all parts of your body. It's when your body is healing. Just think of yourself as a battery. We have nerves, we're water, we're conductive beings. So we're receiving signals. We want to do motor outputs, right? We want our battery to work. And if we're not recovering, it's not going to happen, no matter what's going on in your life. So I work with, again, people who want to have an optimal function. This 20 to 30 minutes now has equivalency to about 4 hours of deep sleep. I also do it in the morning. And before I became a speaker in 2020, my mom used to always be my proofreader. It's funny how we create ourselves our own handicap. I can't write unless my mom proofreads. And I'm going to stutter and go to the bathroom 100 times. How could I ever be a yoga instructor? How could I ever speak on stage? There was a speaker series. So I listened to 13 meditations where Dr. Patrick Porter, the inventor, said, imagine your favorite person on stage and not just looking at them, imagine you're seeing from their eyes because we're all one. But anyways, that's another story. I don't want to scare you yet. So this has different lights that can stimulate the meridian pathways through your ears. If you know acupuncture or dry needling, there's meridian pathways. If you put in a needle, it can release tension in another part of your body. So this is actually stimulating all the meridians. You have light technology as well as 1 or 2 voices that can speak to you. So it entrains your brain. Is anybody in here who tried it? Okay, but is anybody in here who tried it? Right? I see you over there. What did you think? Just shout out. If you thought it was bad, just be quiet. No, for real. Thumbs up? Yeah. I won't give your situation out there, but it's pretty cool. Come stop by the booth and try it. When I talk to people, it's like I return back to whatever happened, and it's not fun. So in my talks, I give ways to do things naturally, but also with technology. And again, working with veterans and fighters, they're like, I'm not a yoga instructor. I'm not going to do a sound bowl. So then I just stick frequency on them, which is the same thing, different way, different strength. So grounding, I walk barefoot. I'm always barefoot look, I'm barefoot now. I hate shoes because the way they shift my spine, my back, and everything I've been through. So I'll go places all the time, and I love being around all these medical doctors. They're like, you can't ground through concrete. I said, yeah, but I still love my knees and my back and how I feel when I'm barefoot. So everybody's different, and I always say, do what you love. So hugging trees, not staring at the sun, but in sunrise and sunlight, you get some good energy minerals from the sun. There's people across the world that stand in the dirt. I have to figure out the name of this tribe. One of my veterans told me, because they said I was from this tribe. Because during sunset and sunrise, I have to be outside, and I just meditate and stare at the sun. I return to what I'm grateful for, because I'm in this body another day. Technology, an anti-aging bed. I sleep on a silver conductive cover that I plug in the wall. And it's only with the grounding because it takes away all the EMF that's in my body. If you come to my clinic, or you might see a little bed on my table, I don't think we brought the plug, but anyways, if you plug it in the wall, even on the sample, you touch it. Well, let's reverse. So you have 60 hertz going through your body, usually around the zip code that my office is in. So it's just running, it's there. We got signals running through our body. So when I read someone, if they have a higher signal, maybe they have lymphatic issues, maybe cancer, maybe something's going on. If they have a lower score, maybe they're barefoot, they're hydrated, who knows? But when you put your hand on the bed, the conductive cover, it's made out of silver. Mine also has tourmaline crystals and other technology, but you immediately go to neutral. Because one, Tampa is like one of the lightning capital of the world. So every time lightning strikes the ground, you get electron donation to the ground. And I don't want to go in too scientific ways, but just think of a negative symbol in the ground and maybe like toxicity or cancer or inflammation as positive sign. So a positive plus a negative, which is me being barefoot, but you just laying on the ground, you return to 0. My kid sleeps on one. He has one for his keyboard. Every room in my center, every bed my patients are on is on one. Building a yoga studio, the floor is grounded. So it's pretty awesome. There's the bed cover. So plugs into the wall. It doesn't plug into the electrical part, just the grounding. It has tourmaline crystals, which is natural infrared technology, and silver threading is also antimicrobial and conductive. Movement. So if you guys want to squeeze your butt, I talked about it over there on my little internet spiel for the SPF Foundation. But pelvic floor activation. Not only spiritually is it the root of your spine, but you have all the nerves that come out to go to your feet. If you're going to the bathroom still, like with control, and even if you're not, because if you saw my pictures, they disconnected me completely. They said I would never go to the bathroom again. I was on a podcast that was like a medical podcast, and the last word I said, oh, I remembered what I was gonna say. I can take a it. Anyways, just joking, love you guys. But seriously, you can make crazy magic happen if you let go of what people tell you can do. Okay, scanning your body in prayer is amazing. So exercise, yoga, cardio, resistance, dance, and the technology I use is a newbie machine. I just got vision body suits for my clinic, so they look like scuba diving suits with 120 pads on it to stimulate your muscles. And for example, the pads on my stomach from the newbie machine, if I have it on for 30 minutes, it could be like 70,000 sit-ups, right? So with my spine and the video you saw, if I ever did 20 sit-ups, I can have a cramping and my muscles will pull and some, asymmetrical movement. My body might start going. I actually can't feel the left side of my body right now, even this thing in my hand. All I feel is vibration. But I use my whole body to help my left side of my body. And the neurofeedback, I used to want to cut off my arm because it hurt so bad. And when I slept, I just put 100 pillows because I didn't know what to do. And I would put my hand on hot cars. And sometimes I would hold ice. I was just trying to figure it out. And then I couldn't feel my left breast or shoulder blade, but that was from all the PICC lines and the IVs. The electrical stimulation, there's frequency. One, you can regenerate nerves. You can regenerate blood flow. It's proven. So I was able to, with 500 hertz, direct current, release all the tension in my body. Do you mind if I share a quick story about you? Are you sure? Okay, she's gonna talk a little bit, but you haven't had your legs touch the bed for how long? Yeah, a long time. So Brandy has some contractures going on, and she used 2 walking poles. I'll show a video later or follow me on Instagram, but it was her last day of her 4 months of treatment. I'm like, I'm gonna get your legs straight. It's gonna touch the bed. And I remember it was the last time, and in my mind, I'm like, fudge. I wanted to say the other word. I'm a cursor. I was like, I don't know if I'm gonna do what I said, and it's rare do I ever promise anybody anything. But the last day, we got her legs straight, knees straight, touching the bed. It was amazing. And maybe it didn't stick, right? Because maybe, yeah, it did. Twerk, twerk, twerk. Okay, so that's awesome. I mean, this is my passion, to get someone's legs to sit. When I see my patients sit up for the first time when I go in the hospital, like, to bring back independence is everything. I work with people of all different backgrounds, Parkinson's, everything. Nutrition is everything. You are what you eat. You are what you drink. And again, I'm not into MLMs. So if you ever go to someone and they're like, use everything from my brand, and they're your neighbor, I would ignore them. Sorry, I'm joking. Not really. It's totally true. Go to your doctor, go to a nutritionist. Sometimes you can get into the wrong program. And with a lot of alternative medicine options, there's a lot of clinics opening without any type of clinical supervision. So even though these things are available, you want to make sure you can trust your provider and stay safe. Again, supplementation. I have a dumping syndrome. I don't know if anybody else has that, but boiled an egg, and it went right through me. They said, I'll be there 6 hours. I was there like 30 minutes. They're like, okay, bye. I'm like, told you. I love when people yell at me, go eat a cheeseburger. I'm like, it'll make me get skinnier. I'll just keep going. So over the counter, supplementation is good, but for me, whatever I take orally will come out the same way. So I love IV therapy. I love oxygen, hyperbaric oxygen therapy, oxygen to my mind, oxygen to the wounds, the nervous system, and nootropics. I have supplementation on my table out there. Nootropics is anything for your cognition, to focus you. I've gotten 95% of my patients off Adderall. And I like methylene blue. And then we have, again, I was the first person in Florida with my marijuana card, but CBD and different things are really good for sleep. Okay, routine lab work, blood test, cortisol test, urine test, fecal matter. I was at a mom's group, and they're all eating like bonbons and Cheez-Its and whatever, and they're like, what's going on that your kid can't have red dye and all of this sugar? And I said, well, nothing's going on, but I want to prevent it. And they said, oh, the holistic lifestyle is only for some people. It only works for some. And I went like this, because, you know, they didn't know what I do, but the holistic lifestyle works for everybody. It's just the life that you want to live. Most of my clients want to have the most optimal life, the most functional, independent life. Or you could listen, you know, to whatever you want to. So what is biohacking? That's what I am, a biohacker. So you unlock your limitless potential. So I take my patients out of rehab into performance training. Like, I want them to get stronger. I had you doing sit-ups, and my interns were sitting on her straight legs. So I said, you could straighten them, good. Now we're gonna do core work. And I think she got mad at me, but I'm not gonna make eye contact. So longevity, brain health, performance, sleep, again, everything I've already mentioned. The brain tap, boom, that's my fave. If you scan this bottom thing, you'll see it all. That's the newbie device. That's what it looks like when I'm working with people. And now it's awesome. They have technology with no cords. Because if you are having any balance issues and you see cords around you, you know it's going to cause you to move a little bit differently. So I'm so excited for no cords. Now, again, alternative medicine. I'm a heavy proponent of marijuana. You're in Florida, I have my card, so if you don't like it, tough. No, I'm joking. Amazon plant medicine. So I went to a shaman, and again, I believe in God. It didn't change my religion. I didn't come back a different religion. But it helped me realize the stuff that I kind of shared about my brother, or different things in life, or even the sexual abuse, or things that I dealt with that I never wanted to talk about. Because when you hide things, or you don't ever even want to look at them again, they're always going to be there. You kind of have to look at it to get over it. So, someone told me like a scar when my mom died. It's never going to go away, but you'll be able to look at it and keep moving along. So that's my goal. Oh, boom, plant medicine, awesome. It improves my appetite, helps me sleep, everything. This is part of the other plant medicine. So we can talk about that later. I'm speaking at a plant medicine conference on the 13th if anybody wants to come. But nootropics, I talked about that to help you accomplish your goals through cognition. How are we on time? Perfect, because I know we got to do movement. I wanted Brandy to come share. So the methylene blue, it will turn your tongue blue. So if you try it, we're all gonna know. But then you become into the Smurf Club. We can have something to talk about at dinner. Methylene blue is something they put in your IV to illuminate your mind, you know, in a brain scan. So it's pretty cool how it works. There's true blue, and then there's true canatine, which is also microdosing nicotine. Nicotine we hear about in cigarettes. Oh, it's terrible, but it's when it's mixed with all those carcinogens. Microdosing nicotine is actually a very good stimulant for your brain. Infrared sauna. So again, my body was stuck like this. You saw all of that. I go in the sauna every single day. I stretch, I do cupping. I would never suggest anybody else who's not used to it to go as much as me, but it was really good just to keep my body pliable, because I was getting so hardened and being pulled down. Everything hurt so much. Body work. massage, joint mobilization, lymphatic drainage, range of motion. Again, anything is better than nothing. And when you have someone else touch you, sometimes you get a 70% more benefit than when you touch yourself. You know what I'm saying? No, I'm joking. I have all these jokes, but you get it. Stay open to all modes of healing. So I'm into biohacking. You can come on up. Are you good? You need help? You got it? So stay open to all modes of healing. You know, we have a regenerative medicine. I've done stem cell on my back, my hips, my knees. I've done traditional medicine. If a kid's having an asthma attack, I'm not going to throw an orange at his face. You know, and then also having Western, Eastern medicine. And this is why I'm not a fan of like one specific company, because if they say we have all of the solutions, that's not true. There's so many solutions out there. And I had someone redo my slide, but my old slide said sometimes in life you feel like you're falling, but sometimes you're just falling into the exact right place that you should be. So to infinity and beyond, I'm gonna let me just go through, make sure, boom, stay there. And this is Brandy, my amazing client. I'll just share a little. She has CP. She came to me and just felt that she hasn't been doing what she should have been doing, and she needed to help open up range of motion, didn't want to have surgeries or anything for the hips or the knees. And I'll let you share from here.
Oh, yeah, look at you stepping out of the box. You want to clip me somewhere?
Look at me. Okay. I'm taking Harper's job.
All right, so you're going to...
I'm kidding, Harper.
Yeah, that's all right.
Go for it.
We love you, Harper.
Yeah.
All right. So...
And you can stand by here.
Okay, wherever. So first of all, I just want to say thank you for letting me up here with you just to share my story. I'm sitting here just looking at everybody, and just for the record, I know we're all here, we have a lot of things in common, and maybe obviously things that are not in common, but one thing I'm going to tell you is I'm really damn excited that there's a ramp on the stage. Because it doesn't mean that none of us want to rock out and have a good time, because constantly we're always trying to fight and make sure that we're heard, and we're often not. And guess what? We don't want to compare, we just want to be like everybody else. Just like Dr. Leiva said, we all have a story, no matter what it is. And so everything that she put on that slide is absolutely true, and let me tell you, every step we take is always a step in the right direction. And so I was talking to Molly. I see you over there.
Molly!
Yay!
That's our other homie.
That's right. So I have to tell you, being born with cerebral palsy, I don't even want to say my age, but I will. I'm definitely proud to be 50. But at 50, it's really ironic being up here on this stage to talk to you, because as of last week, my father is being looked at possibly having the particular conditions that are all here and being talked about. So the neurologist even looked at me and said, well, who told you you have cerebral palsy? So it could very well be that I have the hereditary disease that could be causing my spasticity. All in all, it doesn't really matter at the end of the day. We all want to probably wake up and say, forget it, and throw the crutch across the room and say, why? Why can I just not be stiff for 5 minutes? I just want to be able to dance and move my body and bang my head and jump around and be crazy and have a good time. But when I walked into Dr. Leiva's studio, I used to always tell her, oh, I fall for a living. It's a lot of fun. Don't worry if I fall. And she said, no, you're dancing with gravity. Well, dang it, that is absolutely correct. We all want to have a good time. We all want to move. And for those of you that had that mobility earlier on, and then now you're going backwards and you're feeling frustrated, don't give up. There's many times that my mother was told she'll sit in a wheelchair and drool for the rest of her life. So just get over it. Well, that didn't work, and my mother was a custard. So I totally get it. But she would go back into the doctor and be like, well, she's had a few surgeries and she's not in a wheelchair, so now what? So she passed away when I was 21 of a stroke at 45. So outliving your mother and then following your father, who's about to really understand what he is actually up against, and to be able to be an advocate and be someone with a disability to say, hey, it is okay. Even if you're walking and you're normal, quote unquote, right? What is normal? We all have something, just like you say all the time. But every time I walk in her studio, you have to leave it at the door. So you have to move, whether it's 1 inch or 2 feet or a marathon.
Or your pelvic floor.
That's right.
My husband's probably really happy.
Right?
Did I say that out loud?
Their bottom? Is everybody
Yeah.
squeezing their butt while we're talking?
So I just wanted to say thank you so much, and please continue to be motivated. I'm happy that you guys are here, because without community and without the love and being able to share and know that no matter what your story is, someone out there in the world, you're motivating them to do better, because you never know who you're going to impact. I mean, I walked up 4 flights of stairs to my apartment complex when I was 21, and a woman stopped me for 2 hours. I didn't go to my apartment because she said she cried every day watching me go up because her daughter got injured on a horse ride and couldn't walk anymore, and she didn't know what to do.
I have a quick story I want to just share, and I'm also going to put you on the spot and get you a chair to be a demonstrator. To
Good idea.
Do the
Perfect.
movement that I'm going to do. Will you pass me one, or are you going to hop down and hop up?
Thank you so much.
All my patients are like, what are you doing? Showing off. I'm like, I'm just helping you.
It's all good, man.
Right? So have a seat. One of my patients, no neuromuscular condition, was
You want the
crushed
mic?
between 2 18-wheelers in a car. So they had a million surgeries for their pelvis, all these toes. They were in a wheelchair for 2 years.
You want the mic?
After she saw me, sorry, did everybody hear me? I have a loud mouth. Anyway, but I had a patient that was crushed between 2 18-wheelers, her car, and her hips were into sand. So they had to rebuild it, and then she was in a wheelchair. For 2 years, she saw me, and when she walked in, she said, I don't know why I visualized myself dancing. I attract all the dancers. She said, I see myself just salsa dancing, and I'll speed up. The last day she was there, she was doing this when I got out of the bathroom. But she told me not only did I help her, but she was in Europe, and she walked up a flight of stairs, and this guy was like, come back down. Can you show this group of women how to walk up? And she called me. She's like, I'm thinking of every single step. And this guy told me I'm like a perfect example for a fitness group. And that made her so emotional. It was just a confirmation, or being able to swim on the bottom of a pool again. Like little things. I call them non-scale wins. But we're gonna, how much time do we have? Oh, okay, we gotta move, I promised. So we're gonna do a chair warm up, and if you wanna stand up, you can do it standing. But I wanted to start in a chair because all of us are sitting, and some of us can't get up. So, yet. So let me put this here. So the first thing you can do is squeeze your you know what. All right, boom. But I do this every morning. So if you just bring your hands up like this, right? And maybe one of my interns could take a quick video of the room. That'd be awesome. Say cheese. But so we're going like this, right? And I told you, my left arm, I wanted to cut it off. But even when you squeeze to your fullest ability, maybe 5 seconds, right? 5 seconds is good to connect with the brain. A lot of times we might stand up and our knees want to buckle. It's a good amount of time. Open your hands as big as you can. Try to take out any bends, even if you can't. And if your hands are twisted or big or whatever, don't worry about it. And then squeeze again. So in the beginning, I'll start with a faster pace, but then I just really try to get that range of motion. Your hands are important too. And then I'm gonna leave them open, flip flop. So it's like you're flopping a pancake. Keep your thumb and pinky wide. So now we're just kind of rotating around the elbow. We're scanning the body, right? You can go faster. And now I have a chicken arm. Right? So we can make it fun. I was with the kids earlier. If you have children, right? And sometimes I have patients who want to involve our children in our exercise, our therapy, so they don't knock us over all the time, right? So like we can do a chicken dance. Right? I have my patients hold their kids while they run them around the room and they try to stop their wheelchair. Okay, your hands hurt, watch, right? Hold them out. So if your shoulders hurt, you can bring your elbows in a little bit, but you kind of want to activate that. When do you ever do this, right? Let's see who's the last one standing. Just joking. Squeeze your shoulders back. Thumbs up, though. If your knuckles are forward, a lot of times we're like this, so thumbs up. Try to do that. So just opening the shoulders. Again, you saw those pictures of me. It was little things. Nobody will go for a walk with me because this is how I walk down the street. I'm like... But hey, time is the most valuable resource. I'm with my patients, I'm traveling, or I'm walking down the street like that, right? So from here, and now relax the arms. How good is gravity now, right? We're always fighting gravity, but it's holding us on the earth. So circle your shoulders. Good, breathe. Maybe sigh. Okay, you get 2 more tries to be weird. Ready? Inhale. And maybe sigh. Ah. 3rd chance, or you don't get to be weird at all right now. Ah, good job. All right, so now, again, I work with fighters. I actually paired up one of my patients with MS with one of my I say UCF, but it's UFC fighters. So, and they're using my clinic. So I love to empower my patients to go even beyond, like I said, therapy. When one of my patients saw the fighter, he said, and now try to reach like to the person to the left of you. If you know them, squeeze their shirt, tickle them. If they're cute, pretend to, don't do it, sexual harassment. Right? So just getting that twist in your spine, nice. And maybe I dream of Jeannie. So go here, your spine moves 6 ways. Everybody's like, what? I thought it just like, but up and down, you think of a cat-cow. So you could squeeze your butt, drop your forehead, open your spine, inhale, reach it back. Exhale. Inhale. Neutral. Now right and left. So it's like you're opening one rib cage then the other. I do this in the beginning so you know what I'm doing. Now we're gonna twist, elbow back, so you get that twist. So that's 6 ways, right? But maybe if you're here and you can touch the floor on the right side of your chair, do that. And maybe look at the ground, let your forehead get heavy. Take a breath while you're down, breathe in. Exhale, slowly sit up and then switch, right? Your butt, your back will thank you. Reach, reach, reach. Go again. And maybe if you love yourself, do it again 1 time and stay for 3 breaths. Maybe pick up your other arm if you're not gonna fall off the chair. Or put your other arm on the outside of your leg. When you lift up, maybe use your core, both arms up. Exhale to the other side. Inhale back up. Exhale to your heart. And I think I had 5 minutes, so I'm done, right? Look at him, he's so mean. I gotta get up and forget about your pelvic floor. See you. Love you guys. So come see me at the desk. I'm gonna be talking later to caregivers and follow me. Last chance, take a picture, take a picture. And if you follow my links, I'm up for Best of the Best Physical Therapist. You have to vote for me once a day till June 30th because they want me to harass you every day, not just once. Okay, come on. All right.
All righty, Dr. Leiva, thank you very much. Okay, everybody, you made it through the morning. Way to go, congratulations. Now we get a little bit of a break because it's lunchtime. Okay, so that's what's coming up next. So the staff here is going to come out and do the magic that they do, and you'll be here too, and they're magical. That's all I know. All of a sudden, people come through the door and food appears. It's a really great system. Anyway, everybody relax. We got 90 minutes, and then we're going to get back into the program, okay? So look at your program, and at 1:30, we're back in to show, okay? Everybody relax, take it easy, have some fun. Remember, meet these guys out there in the lobby. They're fun, it's good to be with people, see everybody out there, check on everything, have a great time. We'll see you after lunch.
Well, hello again. We got a lot of exciting things going on here at the annual conference. I hope you at home, as you were watching and listening to Dr. LeVay, I hope you were doing those exercises and hearing about how important it is to stay on the move one way or the other, no matter what you can do or can't. Be positive and stretch all you can stretch, exercise all that you can, so that we can be better tomorrow maybe than we were today, or at least as good and able to do things. So hope you will continue to do that. With me now, I'm excited to introduce to you one of our sponsors in this event. We appreciate their being here, being in the hallway with their exhibit and talking to folks and supporting what we're doing, particularly with our children here at this event, is Jen DiTilio with Top Soccer, the Florida Youth Soccer Association. So Jen, it's been good to talk to you out in the hallway and to learn what you guys are doing. Interesting to hear how you guys are raising dollars to provide programs for kids and youth and soccer. So share with us a little bit about your work and this association.
So about 2 years ago, I took over as the Top Soccer chairperson for Florida Youth Soccer Association, and I work with the National Committee for U.S. Youth Soccer, Top Soccer. So what Top Soccer stands for is the outreach program for soccer. So what we've done is we have created an atmosphere for kids and adults with intellectual and physical disabilities where they can participate in the game of soccer at any level. So we offer free in Florida. We don't charge. So a lot of our clubs in the state will offer this program to parents and their children so that they can get a break. Parents and caregivers need that break as much as the kids need to have that personal interaction with other children and with other people.
So you're here in Florida, and this is exciting to hear about a program that's trying to work with youth and kids who have varying degrees of disability and physical challenges. This group finds that extremely valuable and important, and you guys are doing that here in Florida. Let's talk for just a minute about how you operate. How do you raise your money to carry out these functions?
So we in Florida, we're very fortunate that our state association actually allows us to have some budget to do that. We offer free equipment to our clubs in the area and in the state that want to start this program. The only thing we request is that they are an affiliate of FYSA. So in order to get a program started, they just contact us. So if one of your people watching or anyone in your organization is in an area near a Florida Youth Soccer Club and they go to them and say, hey, we want you to start this program, all they have to do is make that call to us and we're going to help them. So it's all about need, right? It's about what's out there, and if you go to them and say, we would like to have this for our kids, then they can come to us and we can help with that.
Well, you hit on a point that I was about to ask you. We've got listeners from all over the country and people here from all over the country. It's great to hear this is happening in Florida and how you guys are serving the population that would have difficulty being involved in youth soccer otherwise. If I were in some other state in this country, for instance, I'm from Kentucky, how would I know if I go home and I want to check this out and see if this is available in my area? What's the easiest way to do that?
The easiest way is to go to USYS, their website, and look for Top Soccer. So it's usyouthsoccer.org, and you would be able to go there and find out and search the program finder. So you'll look for Top Soccer. Anywhere you put in Top Soccer in Google, you're going to pull up an organization within USYS.
So is there a national office, a national contact, and where is that?
That would be found on the USYS website. They have a committee across the state that can assist you with finding a program in your area or your state. There are 400 programs across the U.S. for Top Soccer.
And you shared with me, I mean, you guys operate basically as a nonprofit operates.
Correct.
With some government input, but you're raising a lot of the money to do what you do. And, you know, this is what this organization is about, is raising money for medical research. And some of the same kinds of things that we try to do, you guys try to do. Anything in particular you'd like to share? There may be folks even in your area watching who might be able to help what you're doing.
Yes, so Florida Youth Soccer has done a silent auction for Top Soccer for the last 3 years, and we are actually raising funds this year to give back to our clubs so that there is no cost, again, no cost involved. If you go to fysa.com, Top Soccer, you will find a link to our program there, as well as finding a link to our fundraiser. So our fundraiser this year has several autographed pieces. We have Guy Harvey. We have a painting from him. It's a replica. It's 133 of 200, so it's very limited edition. We also have autographed things from the Buccaneers, the Miami Dolphin, Jason Taylor from Miami Dolphins. A lot of people are Dolphin fans in the state. We have a lot of different things like that. We go out and reach out to get different things donated to us. It's very difficult to raise funds, as you know, so any little thing that we can do to raise that money, we're very happy to do so, to be able to offer the program for free.
Well, If you're listening out there and you know of youth or folks in your area that would benefit from a program like this, it's important to keep these youth active, working, using muscles, because the more we do that, the longer we're able to do that and the more functionality we have in all areas of our life. So if you know of folks in your area that might be interested in participating in that, check out what Jen has told you, the resources she's told you. And Jen, let me ask you this. If somebody goes on and they can't find anything, is there by chance an 800 number or a toll-free number that they could call you just to get a...
There's not.
Okay.
I'd be happy to share if someone's interested in needing information and they reach out to you guys. I can definitely get in touch with them. I really.
Okay. Well, in the event that you're listening out there in SPF land,
yes,
and you have a need, you can let Norma or me know, and we'll try to access that information for you.
We definitely want to help with that. Also, I wanted to say, if you have family members that are interested in helping with our program, we are looking for volunteers based. We are volunteer based, so we are looking for people to help. We use buddies with our athletes, so that's important too. So anyone that's interested in volunteering, we'll take you.
They'll take your volunteer time, as will we. They'll also take any contribution you might make, and it doesn't have to be a dollar contribution, as you were saying. People can donate items, boats, cars, tractors, whatever you might have. You can donate it, and you'll find some way to turn that into cash to you.
I will, yes, sir.
Well, Jen, thank you so much for being here with us and the service that you provide here and to youth that want to play soccer. Have I failed to mention anything else you'd like to share with us before we go?
No, I just want to thank you guys for this opportunity, and I want to thank Florida Youth Soccer for allowing us to have the funds to come here. We're glad to sponsor this. This is our first event doing this, so we're very excited.
Well, we're so pleased, and this is another compliment to the community here in Tampa, because as we planned this organizational conference here, and we reached out, almost everyone we reached out to said, absolutely, how can I help? And for you guys to come to the table and be a sponsor and be here to participate, we can't tell you how much the foundation appreciates that so much.
Thank you.
So again, this is Jan with the youth soccer folks. Check into that for the folks in your area. We're about to take a lunch break here, and I know you guys at home want to take a break and have some lunch. We'll be back with you at 1:30 Eastern time, so depending on your time zone, you can make that determination. We look forward to seeing you this afternoon. A lot of information. Some of it, as I said earlier, a little bit different than what we're used to hearing about. It won't necessarily be about medical research, but there will be ideas about how do we access more resources, how do we access more networks. Again, we'll be hearing about something called Rarebase, an organization that looks at medical compounds and whether they might help our conditions. And we'll be talking with Thom, Hart, and CPATH about how they might help us quickly and better access resources in Washington, NIH, and those kind of resources. So take a break. We're going to do so, have some lunch here, and we will see you back at 1:30 Eastern Time. See you then. Well, good afternoon. We hope you are back with us. We hope you've enjoyed your lunch. We've had a great lunch and some time of fellowship here, again, at the 2024 SPF Annual Conference here in Tampa, Florida. Right now, folks are enjoying some time together, getting to meet new friends, visit with some old ones, and I've got a new friend that I've just met here today for the first time. I want to introduce you to Johannes Sauer. He is with Cadence Shoe Company. And Johannes, just from looking at these shoes as you showed them to me earlier, those shoes are pretty amazing, and I look forward to trying a pair. So let me just ask you to share with our visitors at home, share with them about these shoes, because all of us, there are probably 125 of us here who've got one of these conditions. And probably all of us walk a little differently. How we get impacted and affected by the muscular and the neurological issues is different in everybody. But for me, I can tell you the way I walk, I'm a scooter.
Yep.
And what you're going to tell us about these shoes would help a guy who's a scooter. So share with us about yourself and about this company and about these shoes.
Will do. Well, first of all, Greg, thanks for having me down here at the conference. It's an honor to be here, and it's so fun to see everyone and have conversations just like this one here. My name's Johannes. I'm the CEO of Cadence. We're a brand new startup based in Santa Barbara, California. And we've made the world's first truly adaptive shoe. Shoes made for people that are experiencing walking difficulties. So think anyone that shuffles, hikes their hip up, throws their leg out to the side, really anything when you worry about tripping and falling, the shoe could be really good for you. And the way it works, it has two distinct areas, but before I get into that, I'll tell you a little story on how it started, because I think it's fascinating. My co-founder, Dr. Tyler Sasko, started this as his PhD project at MIT, and it was a big robot that was made for people post-stroke to rehab and to learn how to walk again. And it was 2 treadmills that would basically mimic removing the floor as you were walking. So what people found was it was a lot easier to walk, they would normalize their gait, they would pick up some speed. And then one of the study participants was like, man, I wish this wasn't $1 million and I could take it out of the lab. And, fast forward 10 years later, here we are with some really nice looking shoes that, help people to walk better.
That's a fascinating story. It's good to know how things can happen that next year or 3 years from now have us working in a very different direction that makes a lot of difference for a lot of people. And I think these shoes would. Again, we all walk differently. I'll tell this quick story. In our annual conference, I believe it was 2019 in San Antonio, a former board member, and she may be listening today, a young lady named Jean Chambers from the Pacific Northwest, shared with me as we sat there visiting, we were talking about walking problems. She had on a pair of shoes that she had resurfaced on the bottom all the way up to the toe with a slicker surface. And she said, here's how you do this, go to your shoe person. So we found our shoe person at home. I bought 3 pairs of shoes specifically for that, took it to him, and now those are the shoes I wear every Sunday when I go to church. Why? Because they help me get from point A to point B.
There you have it.
Now share with us, you mentioned that the shoes might be varied for different walking issues. Share that with us.
Yeah, so basically what the cobbler did for you is we've built this into the shoe. So for the viewers at home, you can probably see there's 2 on the outside of the shoe sole, on the top part, there's 2 distinct areas. Those are nylon pucks. And nylon has low friction properties, meaning it slides. And it's raised off the bottom of the shoe. And in the middle part of the shoe, there's rubber, which is sticky. So what the shoe basically allows you to do, think a door gap, low pile carpet, anything that would typically worry you for tripping and falling, you now slide over it. But you don't want to be always sliding, right? Because that would make you feel unsafe, unstable. And so as soon as you put weight in the front of the shoe, the middle part, the rubber part, comes down and you can stop and toe off. So in a nutshell, the shoe slides when you want it, but it's sticky when you need it.
And for some of us, we have a tendency to drag a toe when walking. Yep. Are there any options now, or are you guys planning anything in the future that might solve that on the end of the shoe?
Yeah, it's a fantastic question. So you can also see it's shaped like a rocker. So already you should have an easier time walking, but we are in the process of adding, you can basically buy a little part that will go on the tip of the shoe, and that will then have additional nylon for you where you don't have to worry about clipping your toe anymore.
Can you share with us two things, what the price of the shoe is, and if someone was interested, how would they look into getting a pair of your shoes?
Yeah, great question. So it's $198, and if you think about it, a pair of Nikes costs $150, right? But Nikes don't have any adaptive features unlike our shoes. And you can just go to cadence.com, that's spelled CANDESE.com, and get your pair there. The great thing about that is you can try the shoes in the comfort of your home, right? And you have a 30-day risk-free trial with that. So if the shoes don't work out for you, and listen, while I hope it works out for a lot of people, I realize it won't work for everyone. It's just the nature of who we work with and cater to and serve as customers. It's not going to work for everyone, we realize that, but we want you to try it. And so there's a risk-free trial with that, so go to our website and order it from there.
Well, when you talk about the price, I know $198 may sound like a lot, but if you've got a grandson or grandsons like I have who are athletic, and when they call Granddaddy or Gigi, can you get me this pair of shoes? And $198 sometimes would be a good deal on the shoes they want. So those shoes can be very expensive, and the idea that a shoe like this might make my getting from point A to point B easier, and more importantly, safer.
Yeah.
Because I decided a number of years ago, actually Norma twisted my arm. I didn't want to go to a walker or to a cane. Leave me alone, I can walk. And what she convinced me is, Greg, if you trip and fall one time and you break an arm or a leg or a hip, you may not recover like you need to recover. So everything we can do to be safe and careful in our mobility is extremely important. And I'm excited about these shoes. I can't wait to learn more about them for me. Johannes, anything else about these shoes or your company that I haven't asked you that you'd like to share?
Well, a couple of things. So I think what we haven't talked about is it's easy to get on, and I'm holding the microphones, it's hard to demo, but so it has a Velcro. While it looks like a lace sneaker, these are just for design, so it's really easy to get on and off with the Velcro. Beyond that, what our customers tell us, and of course taste is something that's, everybody has got a different opinion of what looks good, but I think you can't really tell this is a sneaker that's designed to help with walking. We made sure it's a product that looks good, and so we'll keep coming out with different colors, styles, and all of that. But rest assured that any product that we make is gonna look good and make you stand out less, and that's what we care about. We really want to make sure that you have a product that you are proud to wear.
And if I want to try those, I just want to emphasize again, I can order a pair, and I've got a 30-day time frame to determine if they work well enough for me to keep.
That's
and
right.
If not, I can send those back to you and get all my money back.
100%.
That you can't beat that deal.
No, it's as it should be, right? We want to make sure that the money that you spend on something works for you, and that everybody's happy with the product.
Well, Johannes, we want to thank you again so much for being here, for your company being one of our sponsors and present in our hallway and visiting with people. And it's been curious to watch as you do that. I haven't looked down the hallway many times that someone hasn't been there at the table with you. So a lot of folks are learning about these shoes, and that's good for them and good for you as well. And we appreciate your involvement, your support here with our conference. And we will continue to make people aware of this opportunity best we can. And you can rest assured when I get home, I'm going to be checking them out.
Awesome. Thank you so much for having me. It's an honor to be here. What a great conference. I'm so impressed. I'm seeing a lot of these days, and you guys have put together a fantastic conference, and all the people here are so lovely. So thanks for having us.
Thank you for being here with us, and we'll let you get back to your booth and maybe sell a pair of shoes or two.
Will do.
Awesome.
Right.
Thank you so much. Well, guys, again, there's a lot going on with us. We appreciate Johanna sitting in. If you could be here, we've got probably more vendors visiting with us in the hallway. They're sponsors. They've helped support financially the conference. They've got a number of products that we can look at that might make our lives a little easier, a little more mobile. So look at our program and look at those possibilities. And if you have a walk like I have, at home I'm still up on a cane all the time. I only get in a chair when I go out in public, and that's for safety purposes. But there are places I go that a shoe like that, if it could help me scoot along much more safely, would be very important for my health and my mobility. So you guys might take a chance to look at that and see if a pair of those shoes might help you. I'm trying to bring in here now, my next guest is going to be Kim Sanchez with Social Media Podcasts. One of the things our marketing committee's been working on these past few years is how to get the word out much better about the foundation, about the fundraising that's necessary for us to fund research, about social issues that our population needs to know about. A lot of what we do here at this meeting is get to know one another, share life stories, share experiences so we can make life better at home. We also try to do that all year long through things like SPF talks, through the email blasts that we do, through the synapse letters that we send you. But part of that marketing involves more of a social media context than ever before. And Kim is going to come talk to us about how she does that and how she can assist the foundation in doing that much better. She's on a little scooter as well, so give us just a moment to help get her in here, and we're going to introduce you to her. Come right on around here, dear. Would you prefer to stay in the scooter? I'm in the chair. Yep. Give Kim just a moment to transfer. We're all a little bit slow, but we'll make it. Come on, Kim.
Got it.
As I watch you transfer, that's about how I transfer, so we might make a good dance team, who knows?
Oh yeah, for sure.
And while you've been sitting there for a moment coming this way, I've been telling everyone about you.
Awesome.
And about the podcasting and the social media kinds of things that you do.
Yep.
And how you've agreed to help SPF and our membership learn how to do that better.
Yep.
In fact, if you were here with us, Kim's going to have a spot outside where any of our folks will stop by and visit with her. She may do a podcast right there, or at least show them how to do that.
I've done a few interviews.
So tell us
already.
about it all, Kim.
Just in general, I can tell you guys I have PLS. I was diagnosed about 2 years ago. I started off doing an Instagram where I wrote kind of things about my journey, just almost like a journaling, right? It was like very therapeutic to just share my experience. And it was actually my brother who said, you should think about doing TikTok or Instagram. And I thought, I'm not that kind of person, right? I'm not out there sharing my story kind of a person. But I did start doing videos, which I think, today kind of catches on more. People don't like to read as much as they like to listen to little sound bites. So about a year ago, I started doing a TikTok and Instagram where I shared my story. And it kind of started off around ALS Awareness Month, because PLS and ALS are similar, right, on the spectrum. And I started sharing my story during ALS awareness and just getting really vulnerable about the experience and the process of getting a diagnosis. Because I know for me, I felt very alone, right? It's a rare disease and it's hard to find people like you out there. And I remember I would Google all kinds of different things. After I got my PLS diagnosis, I would Google it, and nothing comes up. Or what you do see is kind of really dark stories, and it just made me sad. So I thought, well, let me share my story and my experience, the good, the bad, right? The days when I'm feeling good, the days when I'm having trouble. And I feel like it really resonated with people to kind of find someone like them who's sharing both sides of it, right? The good and the bad.
So you started doing that. How often are you doing something daily? And if so, if people wanted to follow you, tell them how to do that on social media.
Sure. Yeah. So I have Instagram and TikTok. It's My Spastic Life is the handle. It depends. Sometimes I do it. I used to do daily. I do Motivation Mondays now, where I kind of think about something that's motivational, how to keep a positive attitude. Sometimes I share just kind of like funny stuff, and sometimes I share, stories of experience, right? Like if I go to clinic, or I go to physical therapy, or if I have a new mobility aid, or I tried the Cadence shoes that you just talked about, right? So I share kind of product reviews, things like that. So, but at least twice, 2-3 times a week, I'll share on there.
Well, you want to tune in, I can tell you, because Norma and I, as we met Kim, and we began following what you do, and it made it so much important for us to move this process along with the foundation.
Yeah.
We appreciate your help in doing that. And although it might be difficult to get used to doing it, comfortable doing it, once you do it a few times, it gets much easier, doesn't it?
It does, and I always think about, so part of, is just really documenting the story. And I know someday, my kids can look back on it and see it too. That's another thing that's really important to me. But yeah, I mean, you're kind of just talking to yourself, right? It's like talking into the mirror and just, telling your story. And there are times when, and the worst thing is, you save it in drafts and you never post it, or, maybe you redo it. But yeah, the more you do it, the easier it gets. And the more you can kind of figure out the really, what's the point of your story. And usually the night before I do it, I kind of go to bed thinking about, what do I want to talk about tomorrow? Like, what's been on my mind? And, I think that helps too for me to process my thoughts, right? Like, what happened today? Like, thinking about this conference, right? I'm sure I'm going to have lots to say about it, but think about it the night before and then just kind of, talk about it the next day. But it does get easier. And I find I get at least, I would say weekly, between Instagram and TikTok, I get 2 to 4 people reaching out to me to say, you've helped me to not feel alone. And I think especially folks who are, maybe on the younger side, right, and not knowing where to go, who to talk to, I feel different. It helps them too to kind of see it's more relatable, right? And so they reach out and say, you've given me tips or tricks of what to do, what's my next step in diagnosis, how to find groups like on Facebook is probably the biggest one, is, support groups on Facebook, things like that, so that you can find other people like you.
Well, and that's the central purpose for doing it, as you said, so that others can share the experience, so others can share lives, so we can make each other's day better. There are going to be tough days for all of us, but there are going to be days that life is good, and we need to share both of those so that we have a camaraderie in working through this process. We appreciate your willingness to help us with that, and we realize that, as you said a moment ago, some of our younger folks may be more willing, more interested in doing this, but we want to encourage all of our membership, no matter your age.
Yeah.
Any of you who might be thinking about doing this, we want to help you get comfortable with it.
Right.
And as she helps us learn how to do it, she and Norma will work together, and we'll try to learn how to circulate those things, get you guys involved in the network, and create a discussion constantly among our membership and the people who suffer with PLS and HSP, so that we can make one another's lives better and hopefully make this journey toward a treatment or a cure an easier journey.
Absolutely. And I think, I've had my daughter on my podcast talking about what it's like to be a child with a parent who has a disability. I think, caregivers, I think it's we all have that unique experience. Also, just kind of in general, people don't know what to do, right, when they see us out in public, and should I help, should I not help? It really helps with that piece of it.
All right, thank you so much again, Kim. And you guys, she told you where to pick her up on TikTok and on Instagram, so you might do that the next day or two. And she may have some of our folks on there talking to you. So, Kim, thank you so much.
Thank you.
We're going to head toward our afternoon program with some speakers talking about how we enlarge this foundation's work. So, guys, thanks for joining us, and we will see you sometime later this afternoon.
Hey, everybody. We're getting close to showtime. Okay, so we got a few minutes, so we want to make sure everybody gets tucked in. Get ready for our afternoon set of presentations, okay? You got a couple of minutes to get in place. We're clearing everybody out of the lobby. Come on in, get settled in. Do that stretch again, or whatever that stretch was. Get yourself ready, because in a couple of minutes we're gonna start the afternoon presentation. So here we go. Just hang on. Yes, yeah, please. This is not the blame for it. Oh my God. I want to take a look and we'll see what's going on. All right. All right, I'm going to do an intro.
Perfect.
First for a couple of seconds,
Great.
and I'll bring
I'll
you
get
in.
out of your way.
You can just chill with me. And I lost my camera, guys. Hey guys, we're glad that you're back in the room. I'm going to do a couple of quick announcements before we get going. This is real easy stuff. First of all, did everybody enjoy lunch? Okay, that's fantastic, because now we're going to talk about dinner. You show you where my thoughts are at. Hey, we got written in the program we were going to do this really great thing out in the courtyard and do this kind of thing. Guess what? We ain't doing that. That's been changed up. Sorry, when we went to print with this, back in January, that was sort of the model. So here's what we're going to do tonight. When it is dinner time, after we get you through all your breakouts and everything else, we're going to have a little cash bar action out there in the lobby. Okay, going to do a cash bar at 6 o'clock. And then at 6:30, we're going to come back here and have dinner back in here. We're finding out, as much as we love you guys, and we do, we love you a lot, logistically, lifting and shifting and moving, it's getting really complicated. So we're going to keep you close. We're going to keep you inside because it's hot and it always rains in the evening here, especially where I'm at. I know that is truth. There won't rain anywhere else except where I'm at. So we want to keep you safe, comfortable, and dry, and then we'll get you dinner in here tonight, okay? Sorry about the confusion. When we print this stuff off, things change and the dynamic changes. So that's the latest update. But first of all, congratulations on being able to read the program. We're really happy that you guys are doing that. That means you're getting a lot of information because you're reading and you're keeping up with everything. Okay, well, it's that time. It's 1:30. We are ready to go with our afternoon presentation, and we're going to kick this off. This is going to be kind of interesting. We're going to have a live person here, and we've got somebody going to be zooming in too. Welcome to the world of technology. So we're going to have a really great time with this. Coming up right now is Thomas Hart from CPATH. Thomas.
Hi, everyone. Give me a second to adjust. Here we go. There. Hello, everybody. Thank you so much for being here and inviting me to speak. I actually am going to tag out a little bit, which is why we have Zoom running. So my boss, Dr. Collin Hovinga, will be joining me. We are part of Critical Path Institute. I'll spend a little time talking about what that is and why we want to be a part of this discussion. And then I'll hand it over to Collin, and he can take it from there when we get into the deep science. I'm just a lawyer by training, so I've had to learn a lot about some of the drug development pipeline jargon. We'll do our best to explain all of that jargon. Feel free to find me later and ask questions. I built a flashcard deck, so we can work on it together. So a little bit about Critical Path. It was created 15 years ago by the FDA. It's a nonprofit organization based at the University of Arizona in Tucson, but we work nationally and globally. I actually live here in Florida, so welcome to my state. And sorry about the rain. So first, we're going to cover a little bit about CPATH. Within CPATH, there are different consortia, divisions, and programs. The most recent one is called the Critical Path in Rare Neurodegenerative Diseases, or CPR&D. See what I mean about the alphabet soup? Within that, we work with all of these different rare neurodegenerative diseases. So I think the Act for ALS created CPR&D, so we started working with ALS and other related diseases. I talked with Jackie and some of your other board members about a year and a half ago. We began our engagement, and I'm so excited to see the way SPF has grown, the work that's being done, the Centers of Excellence program, and all of you, honestly. I'm so excited to be here. After we talk about that, we'll discuss HSP, PLS, sort of that pre-consortium model that we work with, and why it might be relevant to the foundation in your work. And then we'll have some time for Q&A. Now, hopefully, Collin will be able to hear your questions. This is going to be a little bit of a trial for us. And then he'll be able to answer. So first, our mission, CPATH leads collaborations that accelerate drug development, advancing better treatments for people worldwide. That's really the focus, right? That accelerate part of it, which is a little bit different from other organizations. Wow. We go right into the dense slides, I apologize. So the way that we build these public-private partnership models is first and foremost through engagement. We're really there to be a support. We're not in charge of anything. We just lead the convening and conversation space. So that includes building consensus actively, sharing risk and cost, identifying and accelerating the best science, and working across the scientific community with the broadest experience. That way you get the input, you get the different angles, and you learn what's working in other areas that are related. It also creates this iterative model. So CPATH has close working relationships with all the regulatory entities around the world. We've called out EMA, FDA, and PMDA. EMA is Europe, FDA is the United States, PMDA is Asia. I think it's the Pacific market. I think that's what PM is. We also work closely with Health Canada and some other organizations. And that allows us to, in that pre-competitive space, identify promising practices and potentiality, move it through the drug development pipeline quickly, developing tools to speed that process, bring it in front of the regulatory bodies that need to approve that treatment, intervention, and get it to market more quickly. Maybe. There we go. Okay, there, sorry. So lastly, these are some examples of the work we've done. I feel like it's important to know that this is a proven model. So some of the other areas that CPATH has worked in includes Alzheimer's, tuberculosis, type 1 diabetes, IBS, Friedreich's ataxia, Duchenne muscular dystrophy. I don't know if you saw the news, but just today, FDA expanded some approved access or expansion of drugs. Kidney transplants, Parkinson's, Huntington's disease. You can see the CPATH solution, and then next to it is the clinical impact. And this is where I'm going to hand it over to Collin because we've now touched on too many science words for this lawyer. So I will step back and hand it over. Collin, let me know if you can hear me. Maybe. Oh, Collin, you're still on mute. Sorry, everybody, we're live. Problem, troubleshooting. There we go.
Hello?
There we go.
I think we're having some AV issues. Can everybody hear me?
We can hear you.
Okay. I can't see the slides. Is there a way to see the slides? Or just tell me where we left off, Thom, and I can.
Sure thing. It is slide 6.
Slide 6? Okay.
Neutral convener.
Okay. I'll just say advance. And then, well, thank you. I'm Collin Hovinga. I'm the Vice President for Rare and Orphan Diseases at Critical Path Institute. I am apologetic that I can't be there today. I had a conflicting meeting that we get required to be there, and I would much rather be with you all than be at this meeting. It's a more stuffy meeting. I like working with families and clinicians and whatnot more so. So I lead the rare disease program at CPATH, and I think Thom gave you a little bit of a peek about what CPATH is. What I want to do is continue on and discuss this further, and then give you some examples of things that CPATH has done, but how we engage with communities. I mean, CPATH was obviously founded at some time ago, and we pride ourselves as being a neutral convener. I mean, we truly work through, working and gathering stakeholders. So we bring in, patient communities. Sorry, okay. We work through with the patient communities. We convene with, regulatory agencies around the world, and we work with, sponsors and clinicians and whatnot, really to really generate scientific questions and then solutions to advance drug development for specific diseases. And in our case, it's really the rare diseases that is our primary focus, and particularly rare neurodegenerative diseases is a larger initiative that we have at Critical Path Institute. So the first step we really focus on is really under defining what the unmet need and translating that into what a regulator, such as an FDA or EMA person would try to understand, because, the regulatory agencies are really a bit of the policymakers that really align with deciding what drugs are safe and effective. Once we define the unmet need, we work through and focus on development of basically tools that would, overcome those bottlenecks, such as, biomarkers, clinical outcome assessments, which are, ones that are relevant to, endpoints within clinical trials. Those are important to proving a drug's, is effective. We have a data management and data science team. We'll talk a little bit about that in a bit. Which is, data is important, particularly in rare disease, because every data point is really precious. We have a modeling and AI group that focuses on, really innovative trial design methodologies so that we use fewer patients for shorter durations to produce the same benefit, but also tools that can help with informing decision-making so that, we're making sure that we don't, expose patients to trials that would be burdensome or more problematic. And then a regulatory science group. I think one of the things I struggled with when I was a clinician and a researcher is that when I would design experiments, I really didn't understand how a regulator approach the same science. And so it really helped me understand how I needed to design my study better so that whatever I was doing had application to advance drug development or to generate something that could be usable to advance a treatment. And so one of our core areas is in that space. What we do is we're in our role, we are a nonprofit. Once we commit any of our resources, we make them publicly available. So we want to make sure that we de-risk as many areas as we can, but also then disseminate those learnings, those experiences, and those tools, so that those communities can move forward and overcome those bottlenecks. Next slide. Now, the Act for ALS was passed, and it really was a pretty monumental legislation. For us, we were chosen by NIH and FDA to stand up a public-private partnership, which really a public-private partnership is something that generates with various communities, just as our model. And immediately what we did is we reorged as an organization, and we partly did this because we wanted to have shared learnings amongst rare neurodegenerative diseases, because in many instances there's some of the same questions, but they're applied to their unique circumstances. So a lot of institutional memory within the organization that we wanted to capitalize on. We also expanded, and we'll talk a little bit about ALS, but I want to introduce it in a bit of a different context to understand what we do at CPATH and sort of the steps of a pre-consortium. And then what we've been working on is trying to develop new areas as we move forward, so that we can impact better communities. And ones that we've been targeted recently has been, we have an effort in PSP, and one we're trying to forge right now in frontotemporal dementia. But one of the key things, and Thom is an example of why this, our commitment to this, is that within R&D, Thom was the first director of outreach. And what we really wanted to have, within this initiative, of this voice of the community to be even more amplified, more real time, more frequent and often, because we felt that the keys of success were really imminent there, and that's sort of the goal of this initiative, is to integrate and translate the stakeholders, but the louder voice in the room is we want the patient community. Next slide. Now, what I wanted to explain real quickly, and I mentioned data competencies, and the reason I say it's important is that one of the core, the important parts of our role is that we really work, we have a huge platform that the FDA supported that would allow us to take in data from all data types. I mean, it's to curate it, to make it, what I mean by that is basically improve it and put it into a common language, which is called a common data model, so that it can be used by stakeholders in sort of the same data language, that are around the world. And we actually have data from around the world. And one of the things that's unique about it is that it's been made also available to regulators. So like if someone say in EMA, they can pull up the platform and they can look at it and work with sponsors to interpret the data. I just had an email before I jumped on this call from CBER, which is the gene therapy parts of the FDA, about working through them about talking about creating some statistical models for trial designs in gene therapy. So they're using this platform as a way to advance drug development along with us. And so, what's nice about this is that it's also shareable to community. When we get data where we openly share it based on the permissions, whoever owns the original data, we follow those permissions, but we try to incubate that. Next slide. Now, why is this important? I mean, using the example of tools, I just want to give some examples of some tools in this next section and sort of understand what a drug development tool is and why it's important. One of the things that's really challenging when you work in rare diseases is oftentimes there's not been an approved therapy. And there's a lack of a regulator, making decisions about particular aspects of the trial design or how you judge a drug's effectiveness, or even really understanding well the natural history of disease and how disease changes over time. And so one of the things that sponsors have to do is they work with regulators to devise a study, but what's important to that is really not just the duration and the dose and all the other pieces out of it, is the statistical methodology behind the trial. And so how many patients should be involved in the trial, for how long, and, there are subgroups of the study that need to be considered. So one of the things that we commonly do as a drug development tool is create simulation tools. So we take all the data that we have, and we take all the different variables that are listed on that slide, and we synthesize statistical models. We can apply artificial intelligence methodologies, natural language processing, whatever that's needed to really optimize the model. And once that's produced, is that we have that model undergo regulatory endorsement. What that means is that anyone who wants to use that tool, the FDA or EMA will acknowledge that that's acceptable. So if a sponsor, a sponsor does not have to go in, or an investigator doesn't have to go into the Food and Drug Administration and say, this is my statistical plan, I have to, defend it and everything. It's been endorsed, you've got a green ticket to get into the fast lane. You can immediately use it for your purposes for the study. Next slide. Another, this is just what a pictorial example of what a GUI would look like, or a global interface is just to give you an example. As you take all of the different variables relevant to that disease state, this is one that's undergoing fit for purpose determination under Duchenne muscular dystrophy. But this is what it would look like. And it's a very user interface. Most people could navigate this, right? It basically uses an interface that you can do to calculate, and you can predict the type of statistics that you want at the end. We've had sponsors use these for various things, even decisions about how long studies, whether or not you should go into a specific space or whatnot, all those type of things. Next slide. One of the things that's often happens in rare diseases is that there's a lack of clinically meaningful endpoints that have regulatory rigor, which means that are accepted by a regulator in a consistent manner. We don't oftentimes understand how much they vary. We don't understand how much how much variation that there is, how they change over time, et cetera. The simulation tool does that. But oftentimes what happens is that when you're working in rare diseases, that there aren't, I'll back up. One of the things that the FDA tries to determine is what concepts are important to patients, what things help you feels, functions, and survives, what things are from that perspective. And so normally what we do is we have measurements that assess those things, right? The survives thing is pretty easy, right? Survival is pretty, black and white, but the other parts really get complicated. And so oftentimes scales that we have are developed for clinical purposes. Those sometimes aren't the same as they're not fit for use for a clinical trial. One of the things that we do as an organization is we begin to, especially if there's frustration with particular clinical outcome assessments, one of the things we do is we actually work on developing new ones. And so, like right now, we're in a process, this is from the guideline from the Food and Drug Administration, but it involves a series of steps that, some of them are literature review, some of them are, interacting with the patient community, but really understanding what are the things that we need to measure and how to measure them. And, then we work with the academic community and NIH, for example, to develop and execute on them so that there are tools that will more adequately capture, the variables that are important to patients. And so and in this instance, we got to get them fit for purpose under the qualification program or the fit for purpose program at the FDA, using that as an example. Another example of drug development tool, one of the problems that happens in various diseases, we don't have the same language. We don't understand how biomarkers change. We don't understand how the clinical symptoms match towards biomarkers or imaging or other things. And so one of the things that we oftentimes do is to create a framework so people can understand the time course of those things, because it helps you identify, where should I put my clinical trial? What most likely benefit am I going to see changed? Maybe it's early in the disease, maybe it's later. Really trying to work through what population I really want to study my treatment in. The other part of it is that as sponsors work with patient communities, it's trying to understand how many patients are there using the same terminology. How do we find these people, et cetera? I mean, so that people have, and we've actually created disease staging programs for alpha-synuclein like Alzheimer's and Huntington's, and there are others that we have active programs in. Next slide. Other one solutions, data standards. Oftentimes when we go into new spaces, there's not a common dictionary that's used for understanding, and so people really aren't comparing apples to apples. And so, we've worked to create data dictionaries that will help the community be able to actually do a better job of taking the data that they have and studying it. We try to make that publicly available. The other pieces that, like right now we're going under we've heard from clinical trial folks and sponsors that there are some diseases where how they do the exams and the assessments for those areas are pretty heterogenic. In fact, even within clinical trials in different parts of the world, they use a different scale, which was a bit surprising. What we try to do is create standards and even ways to analyze the data in those instances so that there's uniformity. Why that's important is that the more variability that you have in an assessment, the more likely that your drug is going to fail in a clinical trial. And we want to minimize that, so the probability of success of that treatment and the number of individuals in a clinical trial doesn't have to be as large. We want to try to reduce that. We do biomarker work, largely trying to look at qualifications or letters of support for biomarkers. I mean, I think Thom, alluded to in his section. I mean, CPATH's leaderboard for endorsements is really high, and then biomarkers are one of the key areas. And what that means is that the FDA will accept that biomarker as a specific tool that can be used in a clinical trial, versus for all sponsors, not just the one that's particular to a specific drug. We use methodological support. Lots of times right now, we're working with NIH to design natural history studies so that the natural history studies are better aligned with what a regulatory agency could use. So we're not just studying it in a way that gives you a paper, it evolves the paper beyond, and so it can be used for maybe things like historical control groups, so to minimize placebo, or it embeds exploratory endpoints that could be adapted for like new further biomarker development and things like that, so that we're actually using that natural history study in ways that it was not even anticipated when it was being thought to originate. We oftentimes work with different groups to try to advise them on fit for purpose things for data, the data that they have, and also, as I alluded to, things like synthetic control groups in the hopes of reducing the number of patients that get randomized to like control arms in clinical studies. Next slide. Now, I want to talk about, those are some examples of like drug development tools. Thom talked about what CPATH does. What I want to talk about is like how we work with communities. And I'm going to come back to that neutral convener role. Like whenever I go into a space, and I've come to a lot of spaces in my career, and one of the things that's nice is you're not biased. You come into this world with, pretty much new eyes. And you convene and you work with that community to understand what do they feel is important. Like, what information do you need to advance things? And you take into the different lenses from all of those stakeholders we talked about earlier, and you synthesize that into something that is an action plan and a strategic strategy. And so like a pre-consortium model, one of the things that really, starts out is I need to understand where I'm going to go. And I need to design a series of projects that will get me there. And, and whatever that is it can be a lot of options. There's, depending on the disease state and where it's at in its evolution of moving treatments through the clinical trial infrastructure and all the way to approval, it can take on various forms. We come into it with no preconceived expectations implications of what that's going to look like. But what the idea is, our sort of the underlying backbone is that everybody gets a say, and that we want to come up with something that produces, because that's our mission. It's really around, trying to make it so encourage. And I think with areas like this community, one of the things that's key is that we want to de-risk the space. We want to make it attractive. So all these tools are available so sponsors can come and try to explore and begin to work through things. So obviously there's a list there of things that we do as part of that activity. But one of the things that we do work with closely is we get feedback from the regulators who oftentimes, like I said, they're the decision makers, and they're like sort of the buck stops to some degree. One of the things that also we do is that, as part of like the pinnacle of it, is a research roundtable, that focuses on trying to bring the community together and ultimately a project plan and to launch a consortia that will have multiple work streams that produce whatever tools and solutions that are needed for that space. And then give you an idea of what that would look like and sort of the timeframes that we're talking, within like now. I mean, this is going to be hypothetical as well. The stakeholder engagement would happen over a couple months. Then we'll be beginning to break down all the pieces of that space. So it's basically inventorying everything you possibly can to understand as much as you can about the condition, where all the data lives, what all the trials are, even for ALS, for example, we ran around and we found where all the biospecimens were. I mean, we literally surveyed around the globe what type, iPSC, brain, longitudinal, plasma, can you share, not share? I mean, very comprehensive approach, even talking about things of how to connect the pieces together so that for researchers that are starting to develop this space, they could be able to move quickly towards that. That's not a CPATH thing to go analyze that, but it's to make sure that the community had, this is where we go, this is what we do, this is how you make it happen. And with the idea that we can feed back information on where are the gaps in information that we're seeing. The next part of it is really talking about, creating a more permanent structure. It's fundraising around that structure with that community. It's engaging sponsors. It's, working collaboratively for grants, et cetera, to execute. And those are roughly the timeframes. Typically, consortia will have multiple work streams. They're more complicated and involved. But the corner thing is that the community that engage with them continues on in the process. It's not like you're just one and done and you're off to the curb. It's that community continues to nurture the same initiative forward so that they actually involved in generating the solutions. Now, next slide. This slide really gives you a high-level summary, and you can have these slides. We're good with sharing them because they're all small print. But the pre-consortia for ALS that we just have worked through. I mean, as you can see here, this is actually simplified, but it's these are six streams. We launched a patient, a PWLE group. We have embedded them in all the work streams. We landscape data. I mean, there are over 70 international data sets with data. We're mapping clinical trials. We're actually working on curating those right now and putting them into regulatory formats and quality, working with the quality for them. Grab data dictionaries, really all with the goal of harmonization. We've already transferred data into the warehouse. We probably are at 3 transfers in right now. We have about 18 other agreements in place to launch that. We have biomarkers, we work with the community to identify the biomarkers that should be prioritized and ones that were particularly most meaningful or interesting to academics and sponsors. And then what we did is we targeted data sets as priorities for ones to go after because we wanted to understand what would be ready for use now, but then what could we feed back to that community, particularly in this case, NIH, of where you need to fund research and where you can fill in the holes so that we can make them usable by the broader community. The clinical outcome assessments, one of the things we've been working on a lot right now is actually, making recommendations to the Food and Drug Administration. We're actually working very closely with the Food and Drug Administration on this project because they hear the frustrations with the primary scale used for ALS, and they've just said, we want you to boil the ocean. And we took that to heart and actually executed a project to look at that. And, I walked you through the method there in that slide. We're actually launching an AI modeling group because we really want to align with the global community in this space. There's a lot of people starting to work in this space, but what we want to do is try to encourage the people that are the experts to get in the room with regulators to okay, let's do this. Let's make sure these tools are things that we can move things forward with that they'll accept, right? So you're not, it's not just for that specific, paper or publication, but a regulator is going to take this and say, you know what, I'm good with this. You can use this, a sponsor can use this, we're good with. And then DHTs. One of the things that was very vocal with the patient communities that we interviewed were they really wanted digital health tools because they wanted to have more power of their disease. They really didn't want to travel. They wanted to have, more flexibility in how their condition was being assessed for clinical trials. And so immediately we targeted data we worked with the different researchers to really start understanding how they were studying these things, how their data, and we're working through right now in conversations with the FDA to how to best use this data. And really, what I've told that community is I want an early submission this year, and I want to start the process. I want to get that moving because that was the thing that they told me that was most important to them. So now that's a big beast. That's consortia, would then launch. It would follow up with those work streams. That consortia is fairly large. We have other consortia that have 1 or 2 work streams. And like the ataxia programs, for example, have 1 or 2 streams. ALS is much bigger. A task force, think of it as a single work stream. It has a bit of it's more of a condensed, it's like a mini consortium. So it focuses on a specific solution that community feels important. And, the idea behind that is to really, focus attention. It produces a single solution. Oftentimes it will involve data. Some of them actually, like we're working with rare epilepsy community right now because they're interested in clinical outcome assessments or endpoints that don't necessarily relate to seizures because there are gene therapy studies right now where people are more concerned about like neurodevelopment or mobility, things like that you wouldn't have normally thought in some of these progressive epilepsies that could be impacted. And then the idea is that they stand up and they terminate, within a shorter period of time, like a year and a half, and then they're done. They've completed their submission. They've gone through endorsement, submissions and whatnot, and then they're completed. Next slide. Now, this gives you an idea of what that looks like. This is a PSP task force right now we have going on. They alluded to earlier. These are data that we curated, we stored, made it all regulatory compliant, and we're creating a clinical trial simulation tool for that community. I had a call this week and we thought, okay, we're talking about this and we're made progress. We mapped all the data. The academics, researchers told us we're missing data. We'd love to get our hands on it. And so we went after, I'm going after those now. But the other piece of it is we got to talking because there were a lot of people doing disease modeling in this space. And we said, well, we don't want to just stop here. Like, can we look at biomarkers? And we started talking about MRI studies. And so one of the things that we've started to look at now is like, okay, let's do this. But then let's blow up this as a side sort of topic in looking at identification of a biomarker. And so we're actually going to be working in that space. And so the next slide, that gives you an idea of how we work through. Is the task force started as really looking at a simulation tool, but it's also going to evolve into something that will look after a biomarker. And, what I'd like to do in that space is that, for those of you who have been in drug development, and there may be some researchers that have discussed this, the qualification program is fairly complicated. It's very long, it's very protracted, it's very challenging to get through. So what I had advocated for within the group is that, I would hope that we would go for letter of support. And why that is that a letter of support for the regulatory agency means that I can go after accelerated approval with that being supportive evidence, which means I get closer to the finish line faster. And so, we're working with that community to now look at the data and the imaging and the methodologies that they're using so that we can, put that on the list for them to work with us to pursue. Next slide. The next slide is just sort of our contact information. I know I've talked a lot. I hopefully gave enough time for some questions so that if the group wants to address some questions that they might have, I'm happy to do that.
Thank you, Collin. If we can turn the Zoom volume down for questions, then I'll repeat them to him, and then we'll turn it up for him to answer, if that makes sense. So quickly, congratulations, you survived that. It's a lot, and I actually think it's important to understand that it's been a lot of hard work in the community that's gotten to the point where you can sit in a room and have a pretty dense scientific discussion about the drug development pipeline. That's exciting. So congratulations that we've gotten there. Now for questions, I'm sure that there are a lot, or there may just be like glazed looks. Don't worry, we'll make the deck available. I'm gonna be around. So if, as you get a chance to digest a little bit, feel free to come and find me. Also, our contact information is up there, and I sincerely welcome any questions or outreach later on. Excellent. Oh, we have a question.
Here's our first question.
Thank you for the presentation, and all the work that you guys are doing seems very important. The question that I have is, what does the intake process look like? How do you identify, say, a specific thing that you want to take on and connect all of these different pieces together?
Great question. Collin, did you hear that? So the question was, what does the intake process look like, and how do we go about identifying targets for gathering information?
Sorry, the intake, could you please clarify the intake process for which part, I guess? That would be helpful. Is it for data? Is it for a pre-consortia or task force, or which part is?
I was thinking more in terms along the lines of taking on a new project, like taking on a new, say, if we're looking at HSP, a specific mutation, and connecting all of the different pieces together with the research that's out there, the patients, the biomarkers, and the whole end-to-end system.
So disease states.
Disease states, it really depends on the communities that we work with. I mean, we try to find out, where there's sufficient, interest and data and stakeholders that are willing to work together. And that's the biggest part. I mean, the only other piece is really important is just our sustainability piece. For initiatives under R&D, part of the funds would most likely come through that act, but we would also, because it's a PPP, the community. The other part of this is that what I've been working on, and I think I mentioned this to the board, I know the program officer for HSP and SP, and we're talking, and one of the things that we were hoping was to draw attention more to this space. He's oversees the portfolio, and I told him that I was giving this presentation. He said, well, I was going to talk to our board, our oversight about HSP and SP, and try to see about convening about some of the same issues. And so so there's those opportunities also as well, and how those all kind of connect. We learned a lot with ALS under this, because it was just this bigger strategy between all these different governmental agencies and whatnot. What I told him is that my hope would be is can we do something that's at least even a smaller version of ALS, because ALS has got a lot of stakeholders, and try to maximize that. But honestly, it's the community that really guides us there. I mean, I'm across rare diseases. We've got a lot of different diseases we're working in. To me, it's still not enough, because there's a ton of rare diseases. What we're trying to do is be as impactful as we can, and where we feel that the community will work with us to move things forward. As you can imagine, there are communities that, and this may be offensive. I'm a small town kid from Iowa, so I'll say it. But they're siloed. There's a lot of it's hard where people won't share, or they're and those are spaces where I'm very vocal usually about it, and I'll say it's like we can't move our mission forward if we can't work together. And so those are usually a no's for me, and it's because they don't want to share, they don't want to collaborate, and that's unfortunate.
Okay, we've got one more question for you before
to
we
give you
go.
more practical steps is that we sit down with those communities and we define what they need, and we fill in the pieces that, and we work with them. So like, for example, when we do, I usually, Thom, you can chime in here too, like with ALS. We have like every other week meetings with ALS community. We have a lot of feedback about how things are going, where things are going, and so it's not like, you just talk to somebody and then they just go off and do stuff. It's longitudinal and interactive.
One more question. So with CPATH.
What's the question, Thom?
With CPATH, I was curious if there's any red tape or anything that might slow down existing processes that we have in place, like with our CERN.
Eventually, we're going to figure this out. So the question was about red tape with CPATH, and if our process would slow down what the foundation is doing.
I guess I'd have to think about what that really means. So, like, to me, I was vocal about it a bit yesterday. It's really getting to an executable plan and with clear objectives. CPATH is not a huge nonprofit. It's not like, we probably have about 170 employees. We're not an NIH or FDA or, a pharma company or, a March of Dimes or, whatever. We're pretty aligned. Usually the things that slow us down are the same things that slow everybody down. It's the contracting process with different individuals. I can tell you we're pretty liberal on things because we still act like a smaller nonprofit. I don't think that's the issue, what I would say. In fact, that would frustrate me to no end. This isn't recorded, but that's been one of my frustrations with ALS is that we're sandwiched between two large regulatory agencies and government agencies. It's been slower than I would have liked. That's why you see like all those working streams literally took us probably, we knew where we were going probably within 6 months. The data stuff right now, it's not us, it's the trying to get through institutions right now. And Thom can attest to this. The reason I threw out that many DCAs agreements to get data in was because I want to get moving. And but since the prioritization and strategic, I've been very strategic about trying to get things that will get me quicker wins fast as possible that address the needs that were vocalized to us. So I don't think that is, and back to the accountability piece. Part of the reasons I'm very passionate about getting on calls with the community is that I need you to keep me in line. If we're not meeting what you all need, it's important to hear that, because our mission is really about producing things that will benefit the community. CPATH started as the FDA said, well, we can't work on this stuff, but we need an entity that can. And so that's hence our relationship with them. And so the bureaucracy for me most likely ends up is that you're working with same things as sponsors will have to work with, or same thing as an academic researcher filing an IND would have to work with. Those types of things.
Awesome. We're at time, but
Don't worry, the answer will be 15 minutes. So you can talk with Thom a little bit right afterwards, and he'll give you all the answers.
You're on
That'll
mute.
take.
I can't hear.
I'll talk to you out in the lobby about that one. Okay.
Awesome. Thank you, Collin.
Thank you.
Thanks, guys.
Thanks, everyone.
Thank you for having us. And if you have questions offline, please feel free to engage. We're happy to meet offline or do whatever you all need, okay?
Perfect.
Okay.
Thank you, sir.
Take care.
Thanks, Thom. Okay, real quick, once again, dinner in here tonight, 6:30. Little cash bar in the lobby at 6 o'clock, okay? No matter what the program says, this is what it really is, okay? Got that? Just one more time for that. Okay, we're rolling right along because we're moving really good. We're right on time. Next is Natalie Downs with Rarebase, from Rarebase. And she's coming, we're setting up her presentation as she's easily sliding up here to the stage. So she's gonna come on up and we're gonna get her going. Look at that, it's almost ready. There you go.
How do I operate this?
I don't have any idea. I think you hit the either the go button or the laser. You can laser somebody, or you hit the right button, probably to advance, I would assume.
Okay.
So, if not, you just tell the man.
Okay, I will.
Tell Harper, he'll take care of you. All righty, guys, here we go.
Can you hear me? There we go. Hi. Welcome to the Drug Discovery Hour. It seems like this is such great timing and a great talk to follow. What I'm going to be talking about is a little bit upstream of the regulatory drug discovery and drug pipeline that we just heard all about. My name is Natalie Downs. I am a genetic counselor by training, and I work as a senior project manager at Transcripta Bio. It is formerly known as Rarebase. It's on my tag. It's in your program, but we changed the name about maybe 6 to 8 weeks ago because we started really focusing in the rare disease space, but we've since then scaled our platform, and now we're able to really interrogate all genetically defined diseases based on the transcriptome, which I'm going to talk a little bit about in our next slide. So I'm very grateful to be here to speak with you all. I'm also here a Tampa local, and I wanted to welcome everybody to the Tampa Bay. Hope you're enjoying it. So let's talk about the transcriptome and what we do at Transcripta Bio. The real foundation of biology is called the central dogma. It's the idea that your genes are held in your DNA. And DNA and genes are read by creating an intermediary step called the messenger RNA. And it's from the messenger RNA that your cells read and translate it into protein. So the genes to RNA to protein is the central dogma. And your genes make up your genome, but your RNA, as it's being expressed, makes up your transcriptome. And that is highly variable. It varies by cell, it varies by what the cell needs in that moment, day to day, minute to minute. And that is translated into protein, which provide all the functions the cell needs to do its jobs. So we are at Transcripta, really instead of looking at the DNA like you would do in a genetic test at your doctor's office, we look at the next step over, the RNA, and evaluate what genes are being transcribed in that cell in that moment to try and look at the different patterns of gene expression. So we do that through a tool called RNA-seq. And RNA-seq, what makes us a little bit different at Transcripta Bio is we perform this robust RNA-seq on the whole transcriptome in human-derived cells. We've started in human-derived neurons made from human stem cells, iPSCs, and we can culture these human neurons in a dish, extract their RNA, synthesize it and sequence it, so read what messenger RNA is being transcribed in those neurons in that moment, and then look at comparative levels of gene expression. We use those comparative levels of gene expression, oh, there's my clicks, to try and look for opportunities to counterbalance any sort of genetic changes that drive disease. So for example, in conditions, autosomal dominant inherited conditions, where there may not be enough functional protein, one copy of the gene is mutated, we can look for opportunities to try and drive and increase the gene expression of the wild type copy. The converse is also possible, that we can look for opportunities in genetic disorders caused by a gain-of-function mutation to try and decrease the expression of that gene in that cell type of interest. For other situations where there may be a complete loss of function in that gene or a missense change that alters the protein function, you wouldn't really want to mess with increasing or decreasing its expression, we can look for alternative ways to kind of work around that gene. So looking at the whole transcriptome to evaluate compounds or methods to change the pathway that that gene is in to try to mitigate the effects of that gene. We do this using our TranscriptoBio FunctionSeq platform, which is what we call our RNA-seq pipeline, where we, like I mentioned, can use RNA-seq to evaluate the whole transcriptome, the expression of 20,000 plus genes at once in that cell. And that includes the 3,900 or so genes that are currently known to be the root cause for genetic Mendelian disorders. We do high throughput drug screens where we put thousands of drugs, one at a time, but all at the same time in an array, on these cell models, these human-derived neurons, and we look to see how those small molecule drugs are changing gene expression. We can take that change in gene expression using the transcriptome and look at what drugs are doing to dial different genes up and what drugs are dialing certain genes down, and evaluate those patterns or genes of interest. If there's one gene we can look for, what drugs show an efficacious response in the direction you'd want to go to mitigate or counterbalance that genetic disease. We've been using this massive amounts of data that we've generated. So we started back in 2020, I believe, and we've been amassing tons and tons of data through these high throughput drug screens. Like I said, in glutamatergic neurons is where we started, in order to amass a ton of data and look for repurposing drug opportunities, where we're looking for FDA-approved drugs that we could use to repurpose. They're approved for other indications, but maybe we find they have a beneficial effect on a gene of interest, and could that be repositioned or repurposed for that condition instead? So using all of that data that we've amassed in this effort, we're also able to now incorporate AI and machine learning models to learn what small molecule structures are doing to gene expression. And using AI, we can start making predictions about what new molecules could be designed to derive the same or desired effects on gene expression. Those new molecule structures can then be synthesized and put back into our wet lab in Palo Alto, California, and screened in the cells again and close the feedback loop. So we're able to continually iterate on our AI pipeline using real-world massive amounts of genetic expression data. We call this our conductor AI. So like I mentioned briefly, we've started over the last handful of years in glutamatergic neurons. These are in the central nervous system. There's many different neuron types in your brain. This is just one of them. We started with glutamatergic neurons over the last few years because when we started with just a singular rare disease focus, the majority of rare diseases are, especially in the pediatric space, are neurodevelopmental. So we wanted to start in the CNS space because that had the biggest population of potential patients to get going. In glutamatergic neurons, we screened over 3,800 compounds at a single concentration. These included both FDA-approved drugs and experimental drugs that had not yet been approved. And then from those top hits in our single concentration screen, we selected about 400 compounds to validate further in our neurons, where we tested those 400-ish compounds at 10 different concentrations and looked to see if they had a reproducible effect in what we call a drug-like response. With increasing drug concentration, are we also seeing an increase in gene expression like we would expect to see? So we've had great results there. We're very happy. And then from there, we decided the next thing we should really try is motor neurons. So in the last couple of years, we've been onboarding motor neurons, getting that assay set up for a pilot test of about 70 FDA-approved drugs, as well as experimental compounds, to try and understand how transferable our findings from glutamatergic neurons were to motor neurons, as well as get those assays ready to go, with the hopes that one of our next big screens would be testing hundreds or thousands more FDA-approved and experimental compounds in motor neurons to look for new hits. As I mentioned in the last slide, in addition to our motor neuron pilot, we've also been developing AI-enabled screening, so taking the lessons and the data that we've learned so far and training our AI models with it, and using that to predict new structures and new small molecules that we've then put back into the screening pipeline. We've done one screen so far in glutamatergic neurons using brand new molecules we had synthesized based on AI predictions. And so far, that has had very promising results. We're very happy. And we are just gearing up to do our second round of screening with AI predicted molecules, hopefully in the next few weeks. And then coming up next for us, we would really like to move into new cell lines and expand our screening and expand our data into new cell types, new disease types. We're exploring cardiology, ophthalmology, dermatology, and ideally expanding our screen that we've already piloted in motor neurons. So far in the last 2 years since completing the screen and really digging through the data, we've had a number of different successes that we're very happy about. It's early, but we've got about 5 programs so far where our partners, working with our partners, we've identified FDA-approved compounds that are repositioning opportunities. So these compounds were already approved for different indications, but showed some potential efficacy in our model neurons for the conditions listed here. And in all of these 5 cases, these compounds are being tried in an N-of-1 trial or an off-label prescription in a patient. So they're in the clinic getting tried right now. We've got very promising results. We're very optimistic for a number of them. And then down below, you'll see a number of late-stage asset opportunities that are at an earlier stage in discovery, where these are the experimental compounds that we've also been evaluating. They're not yet FDA approved. You can't go to your doctor and get a prescription for these. But they've been through the FDA approval pipeline and maybe past phase 1 or phase 2 for safety in humans, but it failed out due to efficacy in phase 2 or phase 3. They weren't effective for the condition they were tested for. So can we use our transcriptomic platform to test those compounds that are safe but weren't effective and find new things they might actually be effective for? And that's really something we're exploring and would probably fit very well into the drug development pipeline that was just discussed in the last speaker's presentation. Like I mentioned, the next steps for us really ideally would be branching into new cell models. We're very interested in the neuromuscular space and the unmet needs there. We've got the motor neuron pilot that went very well, and we'd like to expand on it. We've onboarded cardiomyocytes, retinal pigment epithelial cells for ophthalmology, and keratinocytes for dermatologic genetic disorders, with the idea that the more therapeutic areas we can build into, the richer our data set can be to find new repositioning opportunities, new drugs that we could find new indications for, but also to develop novel drugs and novel molecules to move into the drug discovery pipeline as efficiently and safely as possible. Now, I took the liberty of looking at our motor neuron pilot data. for some HSP genes. I know there's, over 80 HSP genes and many might be missing. This is just a top selection specifically of mostly autosomal dominantly inherited ones. Autosomal dominantly inherited conditions work very well for our platform because, well, one allele may be mutated, the other is usually a wild type, and we can target that for increasing expression of that gene to maybe try and mitigate the effect of having a decrease or a haploinsufficiency in the other allele. So on the rightmost column, you'll see that it's ordered by the expression of that gene in our motor neuron data set. In the unit here is TPM, which is transcripts per million. What our cutoff is to be able to really measure the RNA transcripts in our cells is 10 or above. So anything under 10 is just, it's too small to be detected, or more importantly, to detect subtle changes in expression for those genes. But I think 10 or above kind of meets our threshold. And then, as I mentioned, the autosomal dominant conditions are ideal for us, but I've highlighted here that, for example, SPG7, I know is common. It's an autosomal recessive condition, so both copies of the SPG7 gene will have a variant or mutation in it in order to have that condition. So that might be a target for the alternative pathways I discussed at the beginning, where we can look for a paralog or a gene that's similar that we could try to upregulate, or we could look at the pathway and see, can you upregulate something else and kind of work around and mitigate the effects of that mutation. Lastly, I have bolded some of the genes that just in our pilot study of about 70 compounds in motor neurons, we have at least one candidate compound that seems to upregulate these genes. So I'm hopeful, very optimistic, that if we test many, many, many more compounds, FDA approved or otherwise, in motor neurons specifically, we'll find even more candidates, hopefully for all of the hereditary spastic paraplegia genes. Now this kind of falls in line with a proposal that's currently in hand with the SPF board. They've been fantastic to get to know over the last 6 months or so, where we've been discussing how could we partner to get something like this off the ground and really get some more robust small molecule drug discovery data from motor neurons, a motor neuron screen. And in a successful partnership, we really look for opportunities to collaborate with experts. We have scientific advisory boards. I know SPF does as well. And the more that we can collaborate with the experts, even in this room, the better, to share ideas. We could the plan would be to form a joint steering committee to evaluate the data as it comes in at each milestone and decide which hits and which genes to move forward. That would be optimal for the community. It's important to have robust data sharing between the two, and anything that comes out of these findings we would hope would be opportunities for joint development, whether that means taking FDA-approved compounds into N-of-1 small clinical trials or off-label prescriptions with clinical partners, or unapproved late-stage assets that could be moved back into the FDA-approved development pipeline to get approval and get it into the clinic efficiently and safely. This is a little breakdown so far of the partnership proposal that we've shared with SPF. Like I mentioned, it would be very similar to our glutamatergic neuron screening, where we start by screening about 1,500 FDA-approved compounds in a single concentration in motor neurons, followed by selection with the Joint Steering Committee about what the top candidates, the best data, top hits, top genes would be based on that single screen. We can then move those into CRC screening, which is the concentration response curve. It's an industry standard assay to look for drug-like effect, where you test the drug at serially increasing concentrations and measure gene expression and how it responds. Using that, the next step and kind of final step would be testing the best hits for further validation studies. There's a little bit of room here, but it could be something like doing protein analysis. Does increasing gene expression translate to increased protein? Do we see functional effects on imaging or any other assays to measure how motor neurons are functioning related to that exposure to that small molecule? So this would be the proposals in hand. We're very excited to hopefully continue talking to your board, your team, and learning more about everything that you get to do. We hope to be a part of it. And with that, I thank you all very much for your attention, and I'm happy to take any questions.
Okay, I have a question back here.
Hi, I'm assuming that you guys have a proprietary AI. How are you preventing from hallucinations? What are you doing to mitigate that?
That's a very astute question. Yes, we've been designing our AI algorithms in-house. We've got a fantastic team working on it, including a whole battery of chemoinformatics folks. And we're doing this development based on our own real-world data. So we've spent since 2020, doing lab work and testing these drugs on cells in-house in the lab. And so we're just using our own proprietary data to train those algorithms. Those algorithms can then make predictions. And here's where we close the loop. We're able to put them back into our wet lab and test them again and see if they were right. So you don't really have to just trust it. We get to actually put them back on neurons in a dish and see if they were correct.
Any other questions? Oh, okay, I got one more right over here.
Great presentation, thank you. I need help understanding something, though. Sure. Going back a couple of slides, TPM, tell me the significance of the number.
Sure, no problem. TPM stands for transcripts per million. And I have a hard time with it too. My background's in genetics, and RNA is kind of a new area for me. But RNA is a variable thing. It changes depending on the needs of the cell at that time in response to its environment. It's just measuring how much that specific gene is being expressed. And we do that by trying to basically pull out the transcripts for that gene. So trying to kind of segregate the RNA for that specific gene and quantify it. TPM is one measure, but there's also other ways to use normalized measures. It's just the one that we use because we can set a nice cutoff of can we detect that gene at a robust enough level so that if there is any changes to it, that those are statistically significant and able to be detected. If the gene's like barely expressed and then you make a small change, you can't tell if it's just by chance, fluke, or is it a statistically significant difference?
Hey, Natalie, we got a question over here.
This was an excellent presentation. I missed maybe a little bit at the very beginning. So, the repurposing, finding the drugs that works on motor neurons is very important. And, there are many FDA-approved drugs out there, also new ones. So for your drug screening in motor neurons, so can you remind me again, how did you put those motor neurons in? Is it in culture? Is it in silico? Is it in vivo? What models do you use?
Yes, good question. So we did a very similar process to the glutamatergic neuron process. I believe that we started with human stem cells, and we can drive the human stem cells. They're usually, pre-differentiated to neurons, and we can drive human stem cells to become specifically glutamatergic neurons, or in this case, we were also able to drive them to become motor neurons.
Spinal motor neurons. Mm-hmm. Yeah, but this is the disease of the upper motor neurons. So that's usually the problem with the iPSC cells or, derived motor neurons. Then, first you have programmed cells, like they are engineered, these are pretty much made up cells. And now they're also not upper motor neurons. The only thing is that the species is human.
Yes.
But you may as well work with HeLa cells or other cells. So the glutamatergic neurons, you make them the brain glutamatergic neurons, right?
Yes.
But they may not really be the neurons that you want to study, because they may not be the bed cells, they may not be the corticospinal motor neurons, correct?
That's a good question for our cellular scientists. I'm not sure on the specifics, but that I do know that they're human-derived iPSC, glutamatergic, and motor neurons.
Okay, all right, thank you.
Yeah, thanks.
Okay, we got time for one more. All right, well, get ready now, because y'all are going to be stretching in a couple of seconds. Natalie, great job. Thank you very much. Okay, guys, you asked for it. Time to take a little break. We got mini stretching with Zachary Lipson. Here we go. Here he is. Here he comes. Welcome. You can do it from the podium, or you want this?
I'll take this.
Okay, good.
Good afternoon, everybody. How are we all doing? I know we've been sitting down. It's been a lot of science talk today, so we'll be taking a little break from that to bring you guys some stretching as well. As you guys can see, I almost just tripped. I have HSP as well. I have SPG4. I was born with SPG, or with hereditary spastic paraplegia. I understand some of us here might have developed HSP in our lifetimes outside of adulthood, but either way, I am here to help you. Consider me as your guide. So without further ado, let's get started. We've been doing a lot of sitting today, so everybody, let's just start with one big good morning stretch. Bring your arms all the way up. If I talk like this, can you guys hear me? Yes. Okay. Let's just take a couple deep breaths. Breathing in through your nose, out through your mouth, stretch it as high up as we can. Okay, you guys can bring your arms back down, and we're going to do some moves that a lot of you may hear people who do yoga are doing these, obviously not sitting down, but everything we do today can be done sitting down. If you'd like to sit up, please feel free. So we're going to start with a little bit of cat-cow next. So if you guys would like to put, is this on? Hello? Okay. So if you guys would like to put your hands on your knees, if you're sitting down or standing up too, we're going to be rounding our backs. So as you round your back, let's take a deep inhale. And then we are going to bring our back the other way, bring our stomach forward, and take a deep exhale. And this is called cat-cow. So as you inhale, we're going to round our back, and as you exhale, let's flatten our back out. I want you guys to do a couple on your own inhales and exhales, sitting or standing. Then we'll come back together in a couple breaths. Okay, everybody, let's return back. And from here, just a classic stretch, we're going to reach our left arm all the way across to the right side of our body. And let's switch sides right here. And we're going to go one more time through. So let's bring our left arm back across. And one more time with the right. Perfect. Great work, everybody. Okay, so from here, we're going to bring our arms all the way up around to the top as we take a deep inhale. And just exhale our arms straight down through the bottom. Let's inhale, going back up all the way around. And when we bring our arms down, you can follow along with me. We're just going to go straight down. You can bend forward in your chair. If you're standing with me, you can just bend straight forward. And from here, everybody, let's start to make our way back up. And we're going to bring our arms all the way back up to the sky. Beautiful. And one more time, let's exhale, bring our arms back down, and then we will finish again with our bodies. Okay, good job, everybody. Let's return back. We've been moving our bodies. We're going to get our arms moving a little bit. So if anybody has ever tried boxing before, we're just going to do a little bit of hand movements. So everybody, let's take our dominant hand. For me, my dominant hand is my right. We're going to consider our right hand. Sorry, I'm messing this up. Whichever hand is not dominant, we're going to consider 1. So for me, my right hand is dominant, so my 1 is going to be my left. And then when I say 2, that'll be our right hand. So we're just going to throw a classic. We're going to move our arms a little bit. 1 and 2. You can do this sitting down. You can do it standing. Whatever you'd like to do. We're just going to get our arms moving a little bit. Nice work, everybody. Nice. Hey, I'm seeing a lot of smiles today. Okay, so now we're going to incorporate some more. So that was 1 and 2. Now we're going to do 3 and 4. So when I say 3, you're going to bring the hand opposite dominant around the side to the front like that. Perfect. And then when I say 4, we'll do the other hand. So now that's going to be my right, and I'm just going to hook. Bring my arm straight out to the side. Perfect. So now let's just do it all together. 1, 2, 3, and 4. So it will look like this. Simple, right? Okay. Now we're going to throw all of those together. We're going to do it all together. 1, 2, 3, and 4. We're going to do it a couple times. It might look funny, but it doesn't matter. Nice work, everybody. Okay, and we have 2 more to add. After this, these are the last 2 we're adding. So our 5 will be the hand, number 5 will be our hand opposite from our dominant again. And from here, this is called an uppercut. So we're just going to bring our arm straight up to the sky. Yep, and then classic, 6 will be the other arm, just like that. Nice. So let's do them all together. 1, 2, 3, 4, 5, and 6. Just like so. Nice work, everybody. Wow, I'm seeing a lot of strong punchers out here. I don't want to mess with a lot of you guys. Just keep throwing them for a little bit longer. Okay, great work, everybody. Let's just shake out our hands. Okay. So let's bring our arms straight out to the side. And however far back you can bring your arms with our thumbs up, just slowly try to retract your shoulders. And if you can't retract them, if you're having trouble bringing them back, just keep them straight up. And let's bring them all the way out to the front, keeping them as extended as you can. And let's bring our arms all the way back out once more. And bring our arms in. Okay, so for those of us who can, we are going to just shake our legs, and if we can't shake our legs, we have our arms, just shake our arms. Okay. So we did this in the beginning, but we're going to do it one more time. This is called high mountain, so I'm just going to call it what it actually is. So we're going to inhale into our high mountain pose, which is bringing our arms straight up. Then we're going to exhale, just bring our arms all the way down. Let's inhale, bringing our arms all the way back up to the sky. And exhale all the way down, bringing our arms down to the ground and letting go of our back and top half if you can. And let's inhale one more time into high mountain pose, bringing our arms all the way up to the sky. And release. How are we all feeling so far? Good, good, good. How are we looking on time? Okay, we got 5 minutes. We're going to end off with some more stretches. Okay. Let's bring our arms straight out. You guys in the back can hear me right now? Okay, cool, just making sure. Bring our thumb straight up. Retract those shoulders as far as you can. How many people in here have swam before? Okay, for those of you who know what streamline is, we're going to bring our arms up into streamline, but if you don't, that's just where we're going to bring our arms straight up to our head, and try, if you can, keep your arms behind your ears. If not, just straight up like this is fine. Let's retract back down to the ground. And one more time, and in streamline. Okay, everybody, can retract back down. And I hope you all are feeling good. I know we've been doing a lot of sitting today, but I hope that woke you up. I saw a lot of smiles, so I hope you all had a great stretch. Thank you very much.
Okay, everybody. You guys did real good. We're really proud of you. You did good. You got some stretching. There's going to be another opportunity to stretch with Zachary after our next couple of presenters, okay? So you're going to have to do this one more time today, okay? A couple of quick notes while we've got a couple of minutes. When we do our breakout sessions later at 4:30, we're going to make sure we want the young adults group who want to get together. We're going to have them kind of meet in the lobby, or they can meet with Blair at the registration table, okay, for that breakout session for the young adults, okay? Hey, I got another note. Tomorrow at 5:15, Saturday at 5:15, we're going to be taking a picture with everybody who's got their 5K t-shirts on, okay, from all the 5Ks you've done over the years, okay? Wear one tomorrow, and at 5:15 we're going to get a group photo, okay? Because it's 5K season now. It's been open from March and going through October. So you got plenty of opportunities to do a fundraising 5K. So if you got a 5K shirt, wear it tomorrow, and we'll take a group picture probably out here in the courtyard area outside of the ballrooms. Okay, so that'll be 5:15 tomorrow. Not tonight, tomorrow, okay? So outside of that, we're moving right along. Things are going really, really good. We're getting ready to do our next presentation, and I'm waiting for him. There he is. Harper's in place. Ladies and gentlemen, we're bringing in, we're making a flip now on your program again. This time we're going to start with Dr. Craig Blackstone. Here he comes. Right now, coming up now. We flipped over. A little change on you. Keep you guessing. Here he comes. have this tool here. There you go. I think you just hit the right side, I believe, and it should advance. And you kind of have to aim it at the laptop back there in the right back corner with Blair.
Okay, sure.
Okay,
Thank you.
There you go. You're good to go.
Thanks. Okay. Well, it's a pleasure to be here, everyone. As many of you know, I've given presentations at these for many years. I think something like 15 years ago is when I first started coming to these meetings, and I know that many people are new. Oh, it's not on? Oh, sorry. Oh, sorry. Is this better up here? Okay, yeah, I think it was a little too low for me. Sorry about that. So I was just kind of saying that this is, I think, I've been here about 15 years or so to these meetings, and it's always a sort of a highlight of the year to be able to meet with all of you because, as you know, I've really focused on this disease ever since I first became a faculty member well over 20 years ago. And it's really remarkable to see the advances that have taken place in the HSP field to the point where I used to talk about basic science and a lot of the things that we were working on in the lab. And now we're really talking about therapies, how we're going to bring all these great insights that have been made over the past couple decades, both genetics, basic biology, model systems, how we ultimately bring that to the care of people like you that have this disorder. So it's really an exciting time. When I was putting my intro slide together here, just my title slide, I thought one of the things I noticed about this meeting and about HSP in general that you might not all realize is that many of the researchers, physicians, and so on that give talks here and work with HSP are from very different backgrounds. So you'll see I'm a movement disorders doctor. So the other kind of patients I see are Parkinson's, Huntington's, tremors, dystonia, things like that. Whereas Dr. Mitsumoto will come and he's primarily a neuromuscular doctor, sees HSP, but also ALS and other motor neuron diseases. And I know other people you've heard from, like Darius, have a training in pediatrics. So I think you'll see that a lot of us really come from different backgrounds to the HSP field. And I think that has the advantage of us all bringing the insights that we get from those other diseases that we study, to benefit people with HSP. Another thing I do is I oversee research for a very large department, and that has also given me a lot of more general perspective in terms of what's out there in terms of new therapeutic development, so we can take advantage of all the advances in stroke and epilepsy, brain tumors, Alzheimer's, Parkinson's, and all that, and see what we can learn from those disorders that might benefit people with HSP. So I will get on to my first slide. So I do have some disclosures, and again, these are companies that we often work for where we get our money. One advantage, even though I won't talk about anything related to companies today, is that it does give us the perspective of how to work with industry, how to work with biotech, and how to work with pharma. And many of us who train in basic science or in clinical medicine in the academic track really don't understand the business of medicine. And it's been very valuable for me to understand that because there are partners in bringing therapies to people like you. And to the extent that we get to work with them, I think has been very valuable for me. And I might share one or two general insights about what I think industry is looking for from people like us to be able to move therapies forward. Now, one of the, also I think, areas of diversity among all the different people that speak to you and that you hear from is that our institutions, though they're all academic, are very different. Some of them are small, some of them are large. They're in big cities, small towns. And I think that that's also a tremendous strength, that we all have different perspectives. So I was going to just spend one slide just talking about what I think my institution can bring that's relatively unique. And here's a picture of part of our hospital. So it's very large. We're the largest neurology department in the United States, and we're merging with another hospital where we'll be even much, much larger. And that has disadvantages. I haven't met many of the faculty that I work with ever in 3 or 4 years. But at the same time, we cover just about every disease, just about every type of trial is going on, and we can learn from one another tremendously. And this is just a part of our hospital. All those buildings are part of Mass General, and then we have a whole other section for research buildings and several $1 billion of new buildings on the bottom of the screen that are being built. The advantage of that is that we do have the ability to learn from lots of different areas of medicine. You'll see a bridge at the top there, and that bridge goes to Cambridge, Massachusetts, and that has the largest collection of biotech firms in the nation and probably the world. So we interact very closely with them, and we're hoping that that enables us to bring therapies to people like you much quicker. Also across the river, and this is all walking distance, is MIT. And MIT is an area, a university that we work with very extensively on new technologies, both in terms of research as well as in neuromodulation and assessments for clinical trials. And I'll mention some of those as well, because even though we're often using those in other diseases that are far more common, all these insights will ultimately get to benefit patients with HSP. So one of the things about therapy that I wanted to emphasize first is that a lot of the therapy that you get now is based on circuitry. So we understand, and I think you've probably seen this many times, that these upper motor neurons are involved in both PLS and HSP. And of course, one of the differences is that HSP has all these genes that get identified, though, as you've heard many times, a lot of people with HSP don't have a genetic diagnosis, and that's why there's so much emphasis on trying to, find additional genes, find additional types of mutations, to be able to give people an answer and move closer to therapies. PLS, of course, doesn't have as much of the length dependency, though it does have some, and of course, a lot of other upper motor neurons that are above the waist can be involved to a greater degree. Of course, there's overlap between these two in the cells that are involved, and we're hoping in terms of the basic mechanism so that a lot of the benefits that we see in research from HSP patients will hopefully benefit those with PLS and vice versa. Again, one of the, I think, as somebody who oversees a lot of different types of research, one of the really unique features of HSP is the number of genes that we have. And that can seem daunting at times, 90 or 100 genes. You have one of those 100, how are they going to treat this one disease? But as many of you know, we started to think about it in terms of different groupings. It's not 100 different diseases. There's a smaller number of them within various categories. And I'll show more on that later. But in terms of how we think about the circuitry, so you have these upper motor neurons that start in your cortex and go down to your spinal cord, and they synapse onto these lower motor neurons at different levels, and that controls our movement. A lot of our therapies, and this is not an exhaustive list, but I think there's something you'll notice. Obviously, number one is a very effective one, and that's what you were just doing. Stretching, physical activity is very important. But we also want to decrease the spasticity, relieve pain, treat other types of symptoms that can be associated with HSP. And again, there's just a list of different things. But what you'll notice is that in all cases, they don't affect the progression of the disease. They can alter how you feel and certainly can lead to improvement in your quality of life, but they're not what we're really looking for. We need to have therapies that stop it from progressing or even restore some of the function that's lost, or ideally prevent it in the first place. And we do these kind of meetings and trial designs for several other disorders like Parkinson's. So we're always talking about this for other diseases too. It's not unique to HSP, but I think it's important that we keep our eye on what that goal of really trying to prevent or prevent it completely, or restore function, or prevent progression, that all of those things are going to be targeted at cells rather than circuits. So if you think of circuits as the nerves synapse on each other and ultimately lead to movement, your body's made up of individual cells, and all these genes, of course, function within those cells. So we want to drill down to that cellular level, and that's where a lot of the therapies that you hear about are based. So this is an old picture, but I think in many ways, it's still how I think about it and how many people might think about it, is that we have all these genes and they sort of go into different categories. And those different categories means that these diseases might be similar, more clinically as well as in terms of basic mechanism. And our hope is that as we treat any of them within these categories, that other patients with different forms of HSP that fall in that same bins would benefit as well. And I'll show a specific example of that from our own work, but I think it is important to think about that, we have all these different genes and it seems daunting, but they really do seem to coalesce around a couple of common themes. And those themes are areas, of course, for therapeutic development. Now, one thing also, as you hear many times, is that even though it's the corticospinal tracts that are involved in this disease, that doesn't mean that's where the primary problem is. In many disorders, the problem can be in other cells in the brain that interact with those neurons. Like in the case of these, you'll see this up on the upper right-hand corner, you'll see oligodendrocytes, things like that. If those cells are affected and the myelin is disrupted, then the axons underneath that can be disrupted. So even though we think of it a disease of motor neurons, And in many cases, that's probably where the problem starts. It's not the only possibility. The other possibility is that it starts in other cells and that it impacts those neurons. And all those are important things for us to understand in terms of therapy. But I think that over the last couple decades, this idea of thinking about the HSPs in different subgroups, I think is very valuable scientifically, and I also think it's going to be very valuable as we ultimately move toward therapies. Now, this is one example, and I'll just zip through a couple of these just to throw them all up there. So you can see there's examples where we pick one part of a cell. In this case, it's the endoplasmic reticulum. So it's a very big organelle because if you have a corticospinal neuron that's 1 meter long, the organelle is that long too. So it actually goes the entire way down the axon. And if you disrupt that in any way, you disrupt the axons and you get symptoms of HSP. And we get that in a lot of the common forms of HSP as well as some rare ones. So if we imagine that spastin, elastin, and some of the really common HSP genes are involved in this process, we could have an impact on a very high percentage of people with HSP if we could unravel what the problem is here and come up with a way to treat it or mitigate it. So again, this is not a new idea. As you can see, the article that this is from 12 years ago. But in many ways, I think we still think about it this way, that this is an important pathway, and it's one of the primary ones that we focus on in terms of developing therapeutics for HSP. Oops, there was one more. So what I wanted to do is, before I launch into one specific example of how we can use different types of therapy for even a very rare disease like a specific form of HSP, I just wanted to summarize a little bit about what the field has. And thinking over the last couple decades, at the very beginning, we just had a couple genes. Then, of course, over the last 10 or so years, the number of genes has increased dramatically. So the genetics has advanced tremendously, and it's been a big part of the advance in HSP research. Another big part, of course, is when you have those genetic models, you're able to create genetic insights. You're able to create different models, and those models can span all different types. And you've heard about some of those today. You have these flies or worms. The reason we like those is they're cheap and they're fast, and you can generate them really quickly and make a lot of them for very little cost. Human iPSCs and organoid models you've heard about, those are fantastic because they're human. But as you also heard from some of the questions is they're not perfect. They're not perfectly representative of the neurons in your brain that are involved in this disease. But there's some similarities, and they're certainly a very valuable system, particularly since they're human, and many of our other model systems aren't. And organoids are where we allow them to grow together, sort of mimicking some of the connections that occur naturally in your brain. And of course, it's not perfect, but it's getting close. And I think it's important to remember with every model system that none of these models are HSP. You have HSP, but a mouse doesn't. It has some features of it that we can use experimentally, but only people have real HSP. Of course, we've heard about mouse and rat models, and I'm going to talk about mouse models. Many of you use rats, and rats are a good model in many ways for HSP because they're bigger. If we think, well, we want to get a long axon, well, why not use a bigger rodent than a mouse? One advantage of using mice, and it's why we've chosen to do that, is it's used for so many other diseases, and companies are so familiar with it. They're very used to mouse models and discussing and doing a lot of the pharmacokinetics and a lot of the toxicity studies. A lot of those have already been done in mice, so sometimes it's easier for us to move some of our ideas into therapeutics a little quicker in that system. But I always used to list just mouse models, but I think it's very important to list rat because I think it is, as you've heard from several other studies and a lot of the work that's funded by the SPF, is that rats are a very good model for HSP, and they have a lot of advantages. And hopefully we'll see more of those in the future. And again, I think that when we have these model systems, then we have these different types of readouts. So if you have a model, you have to see something that's abnormal. In a mouse, that could be that it walks funny. It could be that there's electrophysiologic abnormality. I'm not going to show a lot, but we're starting to use a lot more electrical recordings of the nerves, and that's proven to be very valuable because it's very quantitative, easy to measure. And we can, in cells, we can do the same thing. We can look at an abnormal organelle, or we can look at an axon lung that isn't growing properly or has all these blebs in it or something like that. So there's a lot of advantages to the model systems that have easy things to see, because then if we treat it and those things go away, we're getting closer to things that might be good therapies for people. So I think that readout is very important. And I think what the HSP field has really going for it in many ways is technology, the technologies of genetics, the technology of new advances in how we do clinical trials, how we measure symptoms in patients, how we even look at cells, and I'll show one or two examples of that because that is what my own work is about. But I think all those innovations, and they're not all specific to HSP, but they're all happening now, and I think the advantage of that is that allows us to rapidly deploy them, and a big advantage of a lot of the technology is that it allows us to do trials cheaper and faster, and that's always going to be good for industry because that's what they're looking for. Now, over the years, and this is not an exhaustive list, I have a couple therapeutic targets that people have tried to go after, and we're doing some of these trials now, and I'll explain a little bit in detail about what we're doing in some of our models, because we really are trying to design our trials in a way that we can partner with industry. And I'll mention a couple things that I think are important in that regard toward the end of my talk. But you can see there's all these different themes that have emerged from all the studies that people have done over the years. And these are things that we can now test in those model systems. We can test them very quickly in iPSC cells. We can, of course, do them in mice. If the mice are already there, we can do these trials. And I'm going to tell you a little bit about how we're doing that, and because there's always a couple pitfalls when you do those trials, especially with drugs that are new. If it's something that's very well known and being repurposed, sometimes there's a lot of knowledge about how to dose those and how often, and and what's dangerous, what's not, all those types of things. But for a new drug, there's a lot of work that has to go into understanding some safety aspects as well as dosing before we can get too far along in the development process and in terms of therapeutics for patients. So again, these are lots of targets, and you imagine we have all these models, all these targets, and people are sort of testing them in different times. So you'll see a lot of the research that you see published in the HSP field is toward this, and it's very important because they won't all work, but the ones that do hopefully can move forward. To human trials eventually. Now, what I wanted to spend a little bit of time on is our new therapies, because many of us that were trained years ago in medicine, we're very used to the traditional drug development process. And you heard a little bit about that. You have an assay in a cell, you do these massive screens with as many compounds as you can, and then you sort of whittle it down, you modify them, and eventually you get these drug candidates. Problem is that's very expensive, and for a rare disease, how do you de-risk that for industry? And I'm going to show you some ways that I think we can do that now. But in addition to the traditional drug development process, there's all these new ways we can approach things. We can do gene therapies. Many of you might have learned about that in some of the rarer forms of childhood HSP. They use these viral vectors to deliver into the brain, genes that are missing. So you imagine if you have an HSP, it's a recessive form, you're missing a protein. Well, you might not even need to understand that much about how it works if you just deliver it to the right cells, and it sort of corrects that cell, then that could be a really good benefit for people. The hard part is, of course, it's expensive and it's very cumbersome. It can only be done at certain centers and so on. We do a lot of these for different diseases. And again, you can see some dramatic improvements. So there's no doubt that gene therapy is one of the areas for the future. Another one that's quite a lot easier and can move even quicker is knocking down mutant proteins. So you can imagine if you have a dominant disorder and you have this mutant protein that you're trying to get rid of, if you can selectively get rid of that, then you're going to restore a lot of normal function. And those can be used, things called ASOs, antisense oligonucleotides, there's other ways to knock down genes as well. And these are widely used already in clinical medicine. We do quite a bit of it for other diseases, not just as research, but in clinical care. So that's another area that we can think of for several HSPs. And then a really neat way that we're doing right now in collaboration with some groups at MIT are repairing individual mutations. And imagine if you have a mutation and your amino acid is now changed from one residue to another. Well, what if we just go into the gene and just fix it? That sounds like the perfect solution. Of course, it's hard to get into all the cells, and we don't know if we're fixing it at the right time, maybe some of the damage is already done. But still, it's a very, very interesting approach, and it's one that we're taking. Now, you often hear the word CRISPR associated with this, but CRISPR isn't as useful for a lot of the human studies because it can have other effects too that are not intended. So there's all these new technologies called prime and base editing, and those are the ones we're using right now, and we're hoping to be able to have some advances in one of the forms of HSP. And I'll hopefully show that next year if we have any results. Now, one of the things that I think we've heard about with the formation of the network of HSP centers is trial design and readiness. And that's really a critical part of this because as we get these ideas coming through, the need to move quickly is very important. Companies don't want to wait, or even academics don't want to wait. And it's great to have all this infrastructure in place. Part of that is also making sure we're not just ready to do trials, but to do innovative trials. And there's a lot of innovation going on now in trial design. And one of the things, I think you heard a little bit about wearables, and one thing we're doing with some scientists at MIT is you don't even have to do wearables anymore. You can actually just put a wireless device in the room, and it can track you throughout the entire house. And it collects hundreds of thousands of data points, and then the machine learning can sort of give you a score based on how you did that day. And it can even follow one person in a crowded room throughout the house. It's almost scary in a way what they can follow because they can see how you're breathing, your sleep cycles, and we found it to be very useful for a disease like Parkinson's where your symptoms go up and down throughout the day. And I'm sure with HSP you'd think the same thing. You can imagine going to the doctor's office and having a bad day, and you're there for your study visit for 15 minutes, and it looked horrible, but a couple hours later you feel great, and how representative was that? So one of the ways that we're hoping to decrease the number of patients for trials and speed of trials up is to collect a lot more data at home. And again, wearables are a big part of that, but again, these wireless technologies are particularly good. We use them in Parkinson's in particular with sleep, because you can imagine it's hard to sleep with a whole bunch of wires hooked to you. So it's a lot easier if you're just sleeping your normal self and there's just a box in the corner that's recording everything. So we do have those. And again, they're a bit frightening when you see how much they can actually do. So the other thing is about innovations in drug development. I think we heard that already. One of the great things about AI is it can do drug screens. But there's also new technologies in a lot of companies coming out where you can just do de novo synthesis. You don't have to screen compounds at all. You can identify a target and build a drug right there. And that has the advantage that it takes out all that screening time, and it makes companies more willing to jump into diseases like HSP that are relatively rare. It essentially de-risks it. You don't have to do massive screens and pay all this money. You can just jump right to a de novo drug design, so you're willing to take on even rarer diseases like HSP. So I think all of those are real good things for our field in terms of the ability to move a lot of the advances that have already been made forward into therapies. I just wanted to sort of end my talk with one example that I'm working on, and I just thought it would be nice to sort of, because I know many of you think about these different mutations, and especially with SPG3A and 4, that can cause these really severe symptoms. And what does that do for us in terms of opportunities for therapy, and how do we use that information even in terms of the science that we're doing? And as many of you know, there's one with this elastin, which is SPG3A. There's certain mutations that give rise to very severe forms. And this is an example of one of those. Again, this is not new, it's from 2008. However, Darius and we worked with him and several other groups a couple of years ago to look at a whole spectrum of these SPG3A mutations. And it turned out that a lot of the kids, mostly kids with these really severe forms, had either the same mutation or one that's right next to it. So I think you can see in this diagram, it's hard to read the individual things, but there's a couple important themes from that paper. One is that almost all the mutations are missense, one amino acid swapped for another. So that tells us there's a bad gene that we need to get rid of because it's only on one, it's a dominant disorder. So if we can get rid of the bad one, and we think that one is enough, then we've made a significant inroad into therapy. So when we think about a disease like this, and when we see it in the context of all the other mutations, it gives us several therapeutic possibilities. First is it might just be a more severe form of the other one. Maybe the same drugs will work for both. So the typical drug discovery. maybe we can use an ASO and just remove the bad allele, or remove both of them and just get it down to a point where symptoms are better, and that's possible. Or there's the gene editing approach where we go in and fix it. So that's what we're doing. We've created a model that I'll describe that is sort of humanized. It's a mouse that has some human features, not a chimera, not like a mouse that looks like a human, but a mouse with some DNA that's human DNA. It's not as sort of Frankenstein-like as it sounds like it might be. So what we do is we pick the HSP proteins, obviously that are related to ER function in terms of our studies. And the reason we need to combine them is when we try to model a disease like HSP in a small mouse, we're missing one big feature, the length. How can we model a disease of 1 meter axons in a mouse that's only 10 centimeters, so much smaller? And one way is to combine different genes that work in the same pathway and get a more severe phenotype. And we've done this over the years, and you can see, here's an example. This is not a new paper, but I can show you the mouse at the bottom has two HSP genes mutated, and the one at the top is completely normal. The good thing about this is the mouse develops its abnormal phenotype very young, and therefore it's something where we can start screening right away rather than having to wait a year or two. So again, having a good mouse model, this is just some of the quantitative aspects. The only thing, you don't have to read this, the top is this males, the bottom are females. It's just a couple different assays. What that allows us to do is compare mutant to wild type animals. So wild type are the normal ones. And what this allows us to do also is we can start treating these animals, and we want to see the bad findings become closer to the normal. And one of the things we've decided to do, so this is a mouse model that we've already developed, but we're doing the exact same thing with that rare severe mutation in elastin. So this is a regular mutation. The new mouse is the same basic idea with a severe mutation. And it looks like it is more severe so far. Another thing we see is that when we actually look in the spinal cord, in the corticospinal axons, which are of course the ones involved in this disease, we see a huge abnormality in ER, which is kind of good because we said, well, it's an ER protein and ER morphology is disrupted. And in our mouse model, we see a huge effect. So the one on the left is a normal axon going kind of up and down, sort of, it looks like northeast to southwest. And then the other one is just going north to south on the right. And you can see that there's these little lines going horizontally that aren't present on the left. And it turns out that that's abnormal ER. And to do this, we had to use a technique called FibSem. And as I mentioned, one of the advantages we have in this field is technology. We have these new ways of looking at things. So we can actually use a very special form of electron microscopy. It's only available a few places, and we're able to get a very high resolution image of the ER. You can see in panel B there, the white is what the ER looks like when we do it this way, and the green is what it looks like under a microscope. So you can see the microscope misses all sorts of detail. So one of the important things is, as we move forward, is to make sure that we have the sensitivity to see the abnormalities that we need to. And everyone likes to hear about AI, and this is one way we use AI. We used this like 7 or 8 years ago. None of this is new. But this was back when we told AI what to do. Now, of course, anyone that works with electronic medical records knows that the computer tells us what to do now. But back then, we actually used to be able to train it, and it followed commands. So we actually asked it to go through all these massive data sets, find us the ER, and reconstruct it. And it was interesting because the abnormality was so great, the computer couldn't do it. So we had to keep going back and training it. And eventually, it got it and was able to reconstruct. So this is what they call segmentation. So the computer has to identify the ER. And it's a lot harder than it sounds because microtubules are the same diameter. So it's hard to tell those two apart. So people would go nuts trying to do this. But what we're able to do is we're able to create massive numbers of these data sets, and the computer can go do it for us while we sleep at night, and the next morning it's all done. So, and this is what it gives you. This is a completely 100% computer-generated figure. No human being did anything to this. Of course, now we get in trouble with ChatGPT if we do this, but 6 or so years ago when we first did it, it was considered an advancement. Now it's considered cheating if you do it now. Again, you can see the difference here. On the left is a normal axon, and on the right is the abnormal one with the mutations, and you can see there's all these like little ladder-like structures. So very easy to tell. And it's a great readout for us because what we're able to do now is we're able to not only look at the phenotype our mouse has, which are the abnormalities that we see when we examine them, but we also can look at their spinal cords. So when we treat them, do we make those ladders go away or not? And that'll be an important part of our therapy. Another new technology, and I just have 1 or 2 more minutes to go, is the ability to see individual proteins. And one of the challenges in biology has always been, we're trying to look at something with the tools that we have, and often they're not good enough. But we've been able to work with several groups. We're very fortunate to have several collaborators who are really the world leaders in this, and they're able to allow us to see an individual protein. And they're not only can we see it, we can see it move in real life. So this is an individual protein moving. So you can see there's all these different proteins moving on the ER. And they're moving so fast, we usually have to slow these down. And why this is relevant is here's an example with spastin. On the right is a mutant form, and on the left is a wild type form. And that's one spastin molecule moving. And you'll notice on the left it moves just fine. It's going along the ER. The one on the right is moving so slow you might not even know it's moving. And that's what the mutation does. And when we think about how all these different mutations might give different symptoms, one of the things we're looking at is not just thinking statically, but thinking dynamically. Maybe part of the problem is that the protein isn't moving properly. And that's going to be a new line of investigation. So you can see, look at the huge difference that is. So you can imagine when we start doing therapies, we can see if it's changed or not. And here's another one that we just published. This is an ALS. It's a related disorder, of course, not HSP, but some similarities. And we see the same thing. At the top is a normal ALS protein. It's called VAP-B. And at the bottom is a mutant form. And you'll notice the mutant form doesn't move as much. So we have the ability now to use technology not only to improve the trial designs, the things we do with patients, but also to improve our sort of trial design and analysis of cells. So this is a whole new, I think, technological advancement. And we've started using this, what we call it, sort of dynamic systems, a lot more in our HSP analysis. And we're doing it in HSP and ALS, and of course, a natural disorder to add in between is PLS. So we're starting to look at PLS models because we know that those have historically been much harder to make because there isn't as much genetic understanding. So we're starting with patient cells and really trying to see, are there abnormalities we can see in PLS, to really sort of jumpstart that field in terms of some of the models. But again, these are huge, huge effects. So we're really hopeful for the future that this will be part of the design of projects going forward. So again, when we think about kids like this, one of the things we're doing is we're creating this new model system, but one of the reasons we decided not to do it ourselves and to work with a lab called Jackson Labs, which is buildings and buildings full of mice. So that will give you nightmares tonight if you think about it too much, but there's like a whole city of buildings that's just filled with mice, and they're experts at it. And what they're doing is we decided to have them make our mouse and with a human portion around the mutation so that we can do gene editing, but we're also doing drug trials and we're having them do it in the same way they do it for industry. So we're going to learn a lot about what they think are good ways to analyze HSP models, and of course we'll share that with everybody because that might be a really good way for us to know what are the best types of measures in these model systems that allows us to interact more with companies that might be interested in developing some of these therapies. So with that, just wanted to emphasize that, we obviously, science is a team effort. My own group, because I do a lot of administrative work, is very small, but we benefit tremendously from collaborations throughout the world. Certainly I'd highlight Darius. We've worked with him for many years. He's our neighbor across town. David Liu's lab at MIT. He's the sort of the pioneer of prime and base editing, and Jackson Labs, of course, does all of our mouse work in collaboration with us. And finally, at the bottom there in bold, Jennifer Lippincott-Schwartz is really sort of one of the pioneers in cell biology in the entire world, and we've been very fortunate to work with her the past 15 years or so, working on models of disease and analyzing them. And again, it's been a tremendous benefit for us and hopefully for the field to have that opportunity. I think I have a few minutes left if anyone has any questions I can answer, and thank you.
Okay, we've got time for one. We're going to do one right here.
Thank you. So, you mentioned physical therapy. I've had a couple of physical therapists tell me that given my disability and the aspects of it, that there is no benefit to doing it. I've been told, well, stretch your legs, it'll feel good, but your muscles will contract back to what they were.
That certainly hasn't been my experience in the patients that I've seen. I think they've benefited from it tremendously. And you could ask the people, they would know around here in the room, they would know better. I think it really, because I think that, your body is plastic in the same way that if you're an athlete and you're stretching, that benefits your sport. I don't think it's any different for somebody with HSP. It's going to benefit you, and it's going to prevent you from being injured. It maybe, hopefully, will decrease things like pain. It might not slow the underlying disease, but the symptoms that you feel will be better. I think, again, there's lots of doctors here you could ask this to, but in my experience, it's better than the medicines that we give. It has fewer side effects, and I think it has a real measurable impact on people's lives. At least that's what I've seen in all the patients I've seen.
Thank you. Yeah,
Sure.
I just think they were thinking the spasticity and the fighting back, or that they're, I don't know, but
well, remember, they're not used to,
multiple physical therapists
they're
told me
not
that.
used to HSP. That's the thing, is a lot of people don't have experience with it, and
You're very right.
they'll think it's the same as a stroke or somebody with cerebral palsy. They'll sometimes lump you into those groups, and HSP is different. So I think it matters.
Can I ask one other question?
I'm going to have to give somebody else one more chance because we got to move on. Any other questions real quick? We got one more over here, and then we've got to move on. It's a big day. Here we go.
Dr. Blackstone, it's good to see you again.
See you.
I was in the undiagnosed disease program in 2012.
That's a while ago, yeah.
And I was tested extensively at the NIH Clinical Center. And you were on the team.
That's where I was before.
Do you remember Colonel Saburo?
Yeah, we used to see quite a few people with HSP around then because, again, that was at a point where it was really underdiagnosed and people weren't getting the testing. So people like you would come through the undiagnosed diseases program at NIH. And now, hopefully, those diagnoses, people can get that in their doctor's office. They don't have to come all the way to Bethesda.
Your clinical diagnosis was spastic paraplegia. Felt to be consistent with, consistent with hereditary spastic paraplegia of unknown type. So the question is, that was 12 years ago. How many new types have been identified since then?
Well, there's a lot of new types since then. I don't remember exactly how many are in that year, but certainly, around the late 2000s and into the past 2010, a lot of the new next generation sequencing techniques were coming, and insurance started paying more for some of these tests. So I think over those 5 or 6 years, there was a lot of new genes identified. Plus, people were looking more throughout the world, at other populations and so on. But it raises the point that there's still a lot of people out there, and I think the percentages differ from study to study. Let's say 30%, who we really believe have HSP that we don't have a genetic diagnosis for. So I think it does emphasize the importance of those continued genetic studies and really bringing centers together to find new genes, because I think that that's one of the key steps in terms of therapeutics, is knowing what type you have, because that might alter the direction that you go in terms of treatment. So I think it's very important that obviously this network, one of the real focuses it's going to have is trying to shrink the number of people who don't know what form they have, and so we can find more forms and give more people answers to what type they have.
Great. And we got to go. We got to move on. Thank you very much. Great questions, everybody. Dr. Blackstone. Okay, guys, we're trying to keep you on time and get you going. We got a lot more to do this afternoon. All right. We're going to get up here. We're switching everything. We're going to get you set here, Dr. Mitsumoto. There we go. There we go.
Short.
Good. I know. All righty, here we go. We're ready. This is the quickest change we've had all day. Dr. Mitsumoto, everybody.
Thank you very much. Thank you very much, Dr. Gladstone. Switching with me, I had unavoidable conference call. So I'm so happy to be here, and this foundation is an incredibly great organization, and also I'm grateful for your continued support for our study. I'd like to talk to PLS today, natural history study. Let's see. Oops. Okay. Just a minute. So before talking to PLS Natural History Studies, last year in St. Louis, I was at this meeting and sitting together one table with a patient with PLS, and they asked me about the startle reflex. Startle reflex is something, noise or someone talk to you, suddenly jump up. And that's called the startle reflex. And one of them asked me, do you know anything about the startle reflex in a patient with the PLS? I said, no, I never heard. And then he started asking around. Turns out most people who were there had a startle reflex. Some are a lot of startle reflex, some are some. So wow, I did not know. That was the beginning. I decided to ask our patient natural history studies, about 70 patients, and most of them had surprisingly startle reflex, which was not described in the textbook, and I have to admit I did not know. So I asked our coordinator to start this startle reflex, and then looking at ALS, which I am specializing in, and patient with ALS doesn't seem to have this startle reflex. However, a couple patients had startle reflex, which are predominantly upper motor neuron dominant ALS. So actually talking to us and suggesting the doctor to initiate some surprisingly new studies, and eventually we're going to write this up. Now, today we are talking about a little bit of history among ourselves and natural history studies, and another conference international conference will be held in this December. So in terms of our perspective histories, PLS already we heard many times, which has been rare disease, puzzling and obscure disease, just so rare, and considered to be an extreme end of the ALS spectrum, and neglected disease. We conducted this prospective 5-center PLS Cosmos studies sometime in 2009 in conjunction with NIH-funded ALS Cosmos studies. Cosmos means actually oxidative stress multi-center studies. We decided to develop PLS-specific functional rating scale, which we're going to talk about later. And also, we had the International Conference 2019 in Philadelphia, and conference supplement was published. PLS diagnostic criteria was also published after this conference. And PLS natural history studies initiated based on this conference. And now, next third PLS conference, international conference, will be held in December, as I told you. So the first disease-modifying clinical trials in PLS is not any longer unrealistic and far-fetched. But I heard Dr. Gladstone talk today just before me. I see for HSP clearly light in the tunnel. So I think we should be very hopeful of PLS. Now I'm talking about HSP, we are talking about the PLS. Phenotypic and molecular analysis of primary lateral sclerosis paper was published based on PLS Cosmos study. So this study taught us something very unique, very important. ALS FRS is everyone who does anything at ALS using this ALS FRS, which is the standard, most commonly used functional rating scale. When you look at ALS studies, usually runs about 1.0. Take a look at these red letters and run 0.9, 1.2 monthly, moving pretty fast. ALS moving fast. The functional rating scale was made based on ALS. So we used this ALS FRS for PLS studies. It moved monthly 0.05, which is more than 20 times less, actually, moving so slow. So we cannot use usual ALS-FRS for PLS. That's the reason we decided we need some PLS-specific functional rating scale. So comparing ALS-FRS revised, everyone using this for ALS, some people using for PLS, but we use the PLS-specific, more interim grades included and used. So based on this scale, we can show disease moving pretty well in PLS. So we have to now demonstrate it is also true in natural history condition. So also we made a consensus diagnostic criteria for PLS. There was no really consensus diagnostic criteria in the past. We published this based on this conference call. And natural history studies also planned. And in natural history in the past has been mostly retrospective at the best. And our previous application of an NIH application, we presented because we need a NIH grant which funds a lot of money, and yet we was not approved because we don't have any natural history for PLS. That's the beginning that we need to think we need such studies. Rare disease such as PLS requires a multi-site study because so rare, just we cannot do by one centers. We need more centers. United States, Canada may not be enough. We have to move to Europe, Asia, and Australia. Natural history is a prerequisite for designing future clinical trials in PLS. We need to determine the best outcome measures for PLS clinical trials. We should be able to validate the reliability of new diagnostic criteria. And also to improve understanding of PLS is very important to develop biomarker for the futures, a lot of purpose, a lot of objectives. So we got the multiple funding, of course, SPF and ALS Foundation, Mitsubishi Tanabe, so forth, and private donation from Mr. David Marin and the family. We got those funds to start, and we organized the 31 centers, all those specialized PLS specialized people, investigators. And then those happy face site for the United States and Canada. A couple of centers cannot continue, but about 29 centers continue to do this natural history studies. This is the organization. Columbia University is organizing centers. We have committees, executive committees, and EMG. I'm going to talk about all those committees organized studies. And standing committee and objectives, steering committees made of those people, and other committees I'm going to talk about. Any issues resolved needs to be resolved by steering committee. And inclusion criteria of the studies, people has to be older, 25 and older, and the diagnosis has to be established by new PLS diagnostic criteria. Symptom onset no more than 15 years prior to the baseline. And also ability to independently walk, because we have to measure the speed of walking. It doesn't matter whatever assistive device you use, not wheelchair, but we has to be able to walk. And in case where molecular testing has been done, there are no HSP or HSP-related mutations, and expected to have at least some bulbar symptoms because most PLS patients develop some bulbar, means speech and swallowing difficulties. And then if we include those people, the possibility of clinically HSP is becoming less and less. And upper motor neuron symptom signs in region other than legs, and try to exclude the patient with HSP. Normal brain scan, spinal neuroimaging has to be normal or consistent with the PLS. No active major neurological and other medical disease, and residing in reasonably commutable distance, and no history of ALS or PLS in the immediate family, and no family history of hereditary spastic paraplegia, and examination less than 12 months has to have EMG, but we modified because this excluded a lot of possible patients, so we allowed a little bit more easy way. So those are inclusion criteria. There are some exclusion criteria. Unwilling to unable to give informed consent, upper motor symptoms and signs only in legs, and unwilling to unable to visit the study site are clearly exclusion criteria. And clinically obvious cognitive impairment needs to be excluded, and participating in clinical treatment trials not a candidate for this study. So recruitment enrollment, and we decided to recruit 50 early PLS. Early PLS including less than 24 months after symptom, also including probable ALS, based on current criteria, consensus diagnostic criteria. 24 months to 4 years are probable PLS. Then what to do with 24 months? Upper motor neuron, less than 24 months after symptom onset, we decided to include. I strongly believe myself, disease has to be early. The time take more and more, neuron degenerate, axon degenerate. The best time is to treat is earliest time. So we need to include a patient with early stage of disease. But also 50 patients are definite ALS. That means 4 years more than, 4 years after symptom onset, but less than 15 years. So those are inclusion criteria, early and definite PLS. So a lot of studies, including baseline, 3 months, 6 months, 9 months, 12 months, and 24 months. A lot of needs to be done, including cognitive and also testing. But this study is very unique because we are using via phone, iPhone, whatever phone, using those phones to get the measurement and histories, status. But the baseline 6 months and 12 months has to visit the site because they have to check upper motor neuron signs by physician, and also timed up and go has to be done at the site. So this seems to be all the slides set. Sorry, I made a mistake. But anyway, we published paper, this natural history studies for protocol. So if anyone wants to see more detail, you can look at this paper. So characteristic of PLS natural history studies, this is a large multi-site study recruiting a participant with extreme rare disease. It is a study funded by multiple funding agencies, a donation, and Neurobank. Neurobank is based on Mass General, and they donated their time, equipment, everything for these studies. And the study is managed by steering committee and other committees. It is a study which most common data obtained by Columbia Study Coordinator Center by smartphone. The study is a prerequisite for the future clinical trials in PLS, and also it is incorporated with future genetic and biomarker studies, hopefully expanding our understanding of PLS and pathogenesis. So we explore biomarkers. We are looking at oxidative stress biomarker in the urine, lipidomic studies, microRNA changes. Those are planned and being, and just we got all the oxidative stress seems to be no difference compared to controls, which is very important negative studies. We also analyzed DNA exome sequence. Dr. Matt Harms and NIH Brian Traynor are involved with a genome sequence, not exome, genome sequence, looking at some underlying genetic cause of PLS. So biostatistic analysis will be done by Dr. Ken Chan, Columbia University, and looking at analysis characteristic natural history of all 2 patient cohort and providing information useful for next clinical trials. Primary outcome is PLS FRS, whether this is good enough to use future clinical trials, and longitudinal outcomes such as PATACA and NeuroQOL, finger tapping, slow vital capacities, and TUG, which is which I mentioned, walking distance 3 meters from sitting position, walk 3 meters, coming back to original chair and sit down, excuse me. And also we have site visit 3 and 6 visit with phone call from baseline. And electromyography is done at 12 months. So outcomes, I mentioned all those outcomes, including cognitive impairment, and pen upper motor unit score and timed up and go. TUGS test has to be done at the site. And EMG committee checking all EMG baseline done at doctor's office or at the site, and they approve patient can be enrolled in these studies. And also at the 12 months, all do exactly same EMG, same muscles, evaluate changes based on Gold Coast criteria, which is ALS. ALS criteria is very helpful to determine who has ALS, but PLS is very helpful based on this criteria who does not have this ALS sign. So EMG committees, as I said, looking at all those changes baseline and at the 12 months point. So this data will be so important how we define at the baseline as a PLS, and also 12 months how disease evolves, whether it remains to be a PLS or any sign of ALS. Parenthetically, I should mention, if PLS is becoming a little bit more ALS, upper motor neuron initiated disease, even becoming a little bit ALS, clinical features would not change, remains to be slow, remains to be clinically PLS. So diagnostic validation committee is these people all among our sites, and also publication committee looking at any papers they read, they comment to make sure our paper is within our guidelines, purpose, and objectives. So we had a serious concern about execution of PLS natural history studies. This is large multi-site studies. It takes an unbelievably long period of time to obtain IRB approval and set up a subcontract with each site. So multiple site, 31 site organization is very, very difficult. And furthermore, we had COVID. I'm sure everyone wants to forget about the COVID, but the COVID stopped our study tremendously. And the patient cannot come to the site for enrollment, and the people are not coming to the institutions. Even administrator, people, ourselves cannot come to so easily. So delayed just enormously. Yet we have a coordinator. We have to spend some certain money we have to spend. The study initiated 2020. The money is spent. Of course, study cost would not be spent because the patient is not there. But because we had a shortage of funds, and thanks to SPF, and then they supplemented some deficit, which was greatly helpful to carry on these studies. So we have had enough time budget to conduct the complete studies as the organization supported. And we had aimed 100 patients, but became so difficult that we stopped at the 72 patients because we at least reached 2/3 of objective. So currently we have now overall 72 patients, and average age 62 years. Male 40 and woman 32, and disease duration 5.5 average. And then early means less than 2 years, 5 patients, as you see, and probable, that means less than 2 years, but longer than 2 years was about 25. And the definite is longer than 4 years, 42 patients. Currently, those are total of 72 patients. We will finish entire studies end of October, and hopefully we can analyze some data based on Ken Chan, and we can report at the next meeting in December. So next major question is, we have already planned to obtain outcome data, PLS, which is the primary outcome, and other obtainable data via phone during a 24 months visit. Some patients have already completed the 24 months telephone visit, and probably more than 65 patients are still has not reached the 24 months. And the natural history study is the first and only study looking at the long-term natural history of PLS, at least 24 months. But this is incredibly important study, and our database and people suggested we should keep going to obtain long-term, at least 5 years long-term health condition, the PLS conditions, how they do long term. So based on those expressions, we aim to extend 5 years of follow-up studies, and probably I'm going to ask our colleague, Dr. Lee at Columbia, the successor of these studies at this point. So I'm going to switch a little bit. We are going to have third international PLS conference at International Continental Hotel, Intercontinental Hotel, Montreal, December 4th, 1 to 9 p.m. So we will have 31 speakers, organizers, and we have 12 members of program committee helping to establish this speakers and panelists and so forth. This is very unique about the several panels, only several panels, although Dr. Beatty, Peter Beatty is a keynote speaker and talking about neuroimaging in PLS. And probably anyone who is involved in PLS, you know, neuroimaging most rapidly progressing. So we are going to hear what Dr. Peter Beatty say at this meeting. But rest of the meeting are made of all panel discussion, unique because people can present only three slides, and 5 minutes talk, and maximum. And so everyone talks, and I also believe a really short presentation to the nutshell, really key point is important to spend more time to discussion. So we are talking about diagnostic validity of current criteria. We made the criteria based on consensus. How accurate, how reliable is very important. That's a panel discussion. ALS-FRS and other outcome measures are key for future clinical trials. We are going to discuss in panel discussion. PLS registries, biorepositories. Now, PLS is, I learned, United States, Canada is a vast, big countries. You think enough patient to do clinical trial, natural history studies, turns out to be very difficult. And some people are living very distant from centers. People cannot come. Even using a telephone is very difficult. We need a larger population. And we know about roughly 2,200 patients annually present in this United States, but not good enough, not large enough. We have to expand study population, patient population to Europe, Asia, and Australia, and perhaps, I don't know, South America or not, but I think we need to expand so patients are available for clinical trials for the future. So that's where we're going to discuss ALS registry and biorepositories. We are talking about biomarkers. We are developing biomarkers, not perfected yet, but clearly some future possibilities. And rest of the times we are talking about the clinical trial, how we design, how to develop, and also steps towards the next clinical trials. That's the major goal of this conference. So anyone who is interested in, please come to this meeting. We also make copy of this conference and record, so anyone can look at, participate later. You can look at this recorded conference. So again, already the conference, international conference supported by SPF, MNDA, British MNDA Association, Mitsubishi Tanabe, NEOS, and also Mr. Marin and the family supporting this conference. So I am quite excited about our conference to look over, make PLS more being aware, and also making progress in the future development. So thank you very much for your support, and I really appreciate it. And if you have any question, I'm happy to answer if you approve.
Yes, sir. We got a few minutes. Great job. Congratulations. Dr. Mitsumoto, Hank Chupi, who is watching on the live stream, asks, what is the difference between PLS and upper motor neuron ALS?
Well, ALS is a combination of upper and lower motor neurons. Upper motor neurons reside in the brain, send tracts to spinal cord to give a signal to lower motor neuron in the spinal cord. Based on that signal comes along the arms and legs, bodies, to the muscles, muscle contract. See, I want to move. So that's the disease, upper and lower motor neuron as ALS. PLS, upper motor neuron only. Somehow, lower motor neuron and spinal cord is spared. Why? They are resistant to the disease, or they have nothing to do with the PLS disease. Whatever the reason, spinal cord motor neurons are totally spared. Disease, upper motor neuron. So your question is, what is upper motor neuron ALS? Disease we call ALS is not just one single disease, uniform disease. Everyone agrees ALS is multiple disease, up and orange and other things. The disease we call ALS. Some diseases are predominantly lower motor neuron, a little bit, well, almost no upper motor neuron. Some diseases are predominantly upper motor neuron, very little lower motor neuron. That's what we call upper motor neuron dominant or lower motor neuron ALS. Upper dominant ALS, it happens to be better, clearly better, slow, going on years. It's very similar to PLS. So what's the difference, PLS versus upper motor neuron? Of course, we don't know. But if we know PLS, why lower motor neuron are spared, why upper motor neuron only affected, if we know that secret, we can do something, good things to slow down or help the disease. So we need to more understand the disease. Basic science is so important. But I am a clinician, so looking at the patient, how we can help. And of course, at the moment, we don't have any medication to treat. And Dr. Hande is showed this morning as a keynote speaker, and a possible medication presented. It is likely. But I think we need to understand how patient and moving the disease. So if medication is available, we can immediately try those medications based on natural history studies. Thank you for your question.
Great answer. Great question. Great everything. Sorry, I end up over here. Sorry about that. Here you go.
You might remember, Dr. Matsumoto, I was attending that medical conference when your whole study started, and there were genius PhDs in the room, all experts on PLS, and then during dinner or lunch, I don't remember, someone came up with the question is PLS a form of ALS or is it a unique disease? And everybody voted and spoke independently, the different scientists, and about half of them said it was a form of ALS, and about the other half said it's a unique disease. So after your whole study, have you come up with a better conclusion?
Of course, answer is not yet.
Okay.
but I think ALS, PLS is PLS is extreme end of the spectrum, ALS spectrum. So interestingly, if you look at autopsy, spinal cord, and so forth, and looking at more biomarkers which are available currently, and PLS has features of ALS. Looking at the cell, there's changes found in ALS there, yet not clinically obvious, clinically spared, but morphologically, pathologically, there. So I think disease is spectrum. And as I said, ALS is not single disease. PLS is probably not the single one disease, multiple disease in it. We are not good enough to separate out. Seems to be just we call ALS or PLS. So, looking at it, I know everything, but it's not true. Still so much more we know, we realized how little we know. So we have to do a lot of studies need. But as I said, for this foundation, I see the light in HSP, clearly light. So you have to go after it, go after that area. PLS is a little bit way behind, but still we are moving. So I believe we are right directions.
Thank you.
We've got time for one more. I'm going to get one over here because we've got to get you guys moving to your next stuff.
My doctor told me that the difference between ALS and PLS, we can have all the same symptoms, but the difference is ALS, your muscles are completely destroyed. With PLS, it's weakened to nothing.
That's correct. Big difference between both upper and lower motor neuron and a pure pure upper motor neuron. And, you know, lower motor neurons are involved. People develops respiratory problem and significant bulbar signs. And so combination of upper and lower motor makes the disease much, much, much worse. PLS is a pure upper motor neuron. Yes, without any doubt, people have tremendous impairment, difficulty walking, difficulty using arms and so forth, but much slower pace. So I published a paper a couple of years ago hastening the diagnosis of ALS. ALS is a dying disease. Most people after diagnosis dies within a few years. Well, some are longer, but that's true. But still chronic disease. Chronic disease, physicians are relaxed. Relaxed, making a diagnosis quicker. We don't have any better medications. So you see the doctor. If you call to see doctor, they don't see you next day, next week. It takes 2 months, 3 months sometimes, average. So you think disease would not progress too much in 2 or 3 months? That's not very true. That's not true at all. For chronic disease like ALS, disease is progressing. Time is essence. The data is shown, the clinical trials and medication, which works a little, but works medication, if you delay, benefit is much, much less. So we have to start the medication sooner. Now, PLS, even of course, HSP, diagnosis not made immediately, slow. And disease would not change. I think that's wrong. I bet the future, when I die sometime soon, and future, people realize chronic disease, ALS, PLS, HSP, Parkinson's disease, Alzheimer's disease, not like stroke. 90 minutes a key. Doesn't work that way, but still you have to make sooner diagnosis and treat faster. Best example of spinal muscular atrophy. So spinal muscular atrophy and SPA, and we used to call Wernicke-Hoffman disease. That disease, the baby die within 12 months, or at most a few years, baby. And now genetic disease treatment is available. If you start earlier, you can stop the progression. You can delay the progression. So the time is essence. And so make sure we have to tell the doctors who are young doctors proud, my first patient is waiting list 3 months, so much request coming. That's wrong. You have to see patient as fast as you can because you cannot deal with slow neurodegenerative disease and slowly. You have to move faster. So that's the reason we published the paper. And I think can be applied to PLS and HSP. Sooner is better. Sorry. Okay.
Okay, everybody, thank you very much. Dr. Mitsumoto. Okay, we got some stretching coming up real fast, but you got some complicated moves you need to make here after Zach gets done with you guys in a couple of minutes. Remember, we got the small breakout sessions coming up. The first one with the caregivers is actually going to be right down this hallway, just out in the round and down in this left-hand corner over here for the caregivers. For the women and the men, you've got to go all the way up to the front corner of the building on this floor, and there's an elevator to take you up to the second floor to some breakout rooms. It's going to be a little tight. It's going to be a little time consuming, okay? And there's a staircase for people who can use stairs, and there's an elevator that only goes up one floor, okay? And so we'll own it. When you get there, it'll be your elevator, okay? So that's you guys are, it's completely on the opposite end of the building, on the front corner, opposite of registration where you get your hotel keys and stuff. Upper right-hand corner over there. You got it? Okay, you know where you're going, that's what's going on, and then after, the only group staying in here is going to be the parents of childhood onset with Dr. Fakhari, okay? That's the group that's going to be staying in the room for their presentation, okay? So Zach, you got to keep them moving, you got to get them going. You're the man, here he comes.
Thank you for those of you who are going to stay with us. I hope not too many people are going to get up.
Come on up. You can just drop it in your pocket if you want to.
Yeah. Thanks. Okay, so there's something I just wanted to note before we start. This thought has been just moving through my head all day. And something I've kind of realized through, sorry, I'm a teacher and people moving is very distracting. No, it's okay, it's okay. So something I've come to realize with having HSP and kind of what I hear other people say is just because a cure doesn't exist does not make positive progress impossible. Positive progress is always possible. I mean, as you guys can see from the back of my shirt, it's something I thought of. The only disability is believing you have one. We all have the ability to positively progress, no matter what we hear anybody say. It is always possible. So before we just started, I wanted to let everybody know. So, oh, please applaud yourself. So let's just start with stretching out our hands. If you're sitting down, great. Let's just bring our arm right out and try to pull our palms down. And if you can't pull your palm, you can just grab a thumb or you can grab one finger, whatever works best for you. Because I've come to realize through boxing and Muay Thai, which I've started to do recently, let's switch sides. I feel like the story when I was growing up was that I have a degenerative disease and I can't really progress positively. Let's switch sides. But then I realized I can learn and I can grow. And boxing and Muay Thai kind of helped me see that. And let's switch sides one more time. Because every day I left, I was able to leave with new knowledge. I can guarantee you my hips have never felt better than when the days I do Muay Thai. Okay, let's bring our arms straight out in front. And we're just going to spin our hands. I saw this video on Instagram where it's impossible for somebody to move one hand forward and one hand backwards. If anybody can do that, very cool. All right, let's go backwards or switch the rotations, whichever one you guys have been doing. Doesn't matter which one you do. And let's switch sides. All right, everybody. So let's shake out our arms. Do what you gotta do here. Okay, so we're just gonna start with bumping our arms straight up in the air. We're not doing anything crazy. You can turn it into a dance if you'd like. Okay, now I see a couple new faces in the crowd, but before last time we did a little bit of boxing, I'm gonna do a little bit again because I did see a lot of smiling faces. So for those of you, everybody can sit like we did last time, but if you want to stand, totally feel free. So for those of you who are not here, just gonna start us again real quickly. I'm gonna say numbers 1 through 6. When I say 1, you're gonna throw your hand opposite from your dominant hand. So my dominant hand is my right. When I say 1, I'm gonna throw a left hand straight out. And when I say 2, I'm gonna throw my right hand out. So once again, let's just throw 1 and 2. Nice work, everybody, nice work. You might feel your shoulders popping. Let it, it's all positive. Okay, now for those of you who remember, we can go right in, but we're gonna do 1, 2, and 3. So our 3 was when we took our left hand, or the hand that's not your dominant hand, and we're going to bring it we're going to hook it to the side and just end right in front of us. And then 4, the next one, same thing with the other hand. So let's just do 1, 2, 3, and 4. So we're gonna do those all together. Let's do it. 1, 2, 3, 4. Just throw a couple, and then we're gonna add in 5 and 6 in a little bit. Nice work, everybody, nice work. Okay, so actually I'm gonna challenge you guys a little bit. We're gonna throw a little combination here. Let's do 1, 2, 4. Let's see if you guys can do this. So if you're doing it along with me, where you're throwing your left hand as 1, we're gonna go 1, 2, then we're bringing our right hand back, we'll go with the 4. So we're going 1, 2, and with that same hand you just brought out, we're bringing it right back to the side. These ones were the most difficult for me to learn, using the same hand. Nice work, everybody. Now let's throw a couple. 1, 2, 3. This one is going to be a little easier because it's just alternating the hands we did. So we're going 1, 2, and then 3 with the hook. 1, 2, 3. I want you guys to throw about 5 to 10 of these. Then we're going to reconvene. Nice work, everybody. Nice work. See, positive progress is possible. I don't know if not everybody who's doing this with us right now might not have HSP or PLS, but as you guys could see, over time, we can grow. It might hurt. It might be very painful because I can almost guarantee you it does hurt. It is possible. We're not done. Just wanted to get that out there. Okay, so now I'm gonna add in our 5 and our 6. So our 5 and 6 are uppercuts. So this is where we're just going to bring our hand like a shovel. From the bottom, bring our hand from the bottom and straight up almost to your opponent's chin. Okay, so we're gonna throw 1, 2, and then 5. So what it will look like is this. So we're throwing a jab, a cross, and then an uppercut with that left. And we're gonna throw a couple here. Nice work, everybody, nice work. Nice work, nice work. Okay, and I just want to get a little bit of leg movement in there. So for those of us who can, and if not, do what we're going to do with our legs with your hands. So what we did last time, let's just shake them out. Do what you can. If you're having trouble moving your legs, no worries. Just give those arms a whole big shake. Okay, and as we do a Muay Thai, thank you.
That was really great, everybody. Thank you. Remember, you've got other places to be right now, okay? So you got to get yourself to the other breakout sessions that you're supposed to be at. Women only, men only, none of that mixing stuff, okay? We got guards at the door to make sure that doesn't happen. And then the caregivers are just right around the corner, down in this corner over here to the left. So get to where you got to go, and we'll see you for another presentation or dinner later.
Well, good afternoon again, and you continue to hear a lot of great information. Dr. Mitsumoto, Dr. Blackstone sharing some of the more recent concerns and more recent research that everyone is working on, and that's all good to hear. With me now, though, another one of our sponsors, another one of our partners this weekend. I have with me Leah Fronerith with Canine Companions. And you guys know these animals have been so important, these beautiful dogs, companions, and help to many of our members in the past. We have at least 2, I think, who are here this weekend with this group. And so what Leah and Canine Companions do, not only here but all over the country, maybe all over the world, you can share, is so important for our community. And so Leah, thank you for being with us. Thank you for your Canine Companions, what you guys do. Let me just ask you to share what you guys do here locally, what maybe you do across the country, and how you see it kind of a ministry to and a help to this group.
Absolutely. So Canine Companions is a national nonprofit that trains service dogs, hearing dogs, and facility dogs for individuals and institutions to utilize and help individuals empower themselves and gain independence. Our dogs are found at 7 different regional sites across the country. Applicants can put in their application and tell Canine Companions what they're looking for in terms of a dog, what services, what tasks, what independence someone is seeking. And Canine Companions will work on finding the right match, finding the right dog, and place that dog free of charge for individuals with any kind of disability that we can serve.
Can you share some of the specific functions that one of these animals could do for someone that might have HSP or PLS that has mobility problems, getting in and out of beds, through doors? Share some of those kind of things that the animals can assist with.
Absolutely. So our Canine Companion dogs have several tasks, 45 plus, that they are trained with upon graduation. It can be taking and tugging an arm of a sleeve of a jacket off if that's a difficulty for someone. The dogs are trained to pull those things off, pull off of a sock. They're trained to retrieve items that are on the ground and hand them to their person or to the registrant at the cash register. They are trained to alert people in the household if there's a need from an individual that is matched with a dog. They are trained to sidle up next to their person to provide a little bit of support and leaning ability as well, to kind of really support. They are also, many of them, trained to pull the wheelchairs and assist in that kind of mobility if necessary as well.
You use the terms matched with the dog, so I think the question then would be, so an animal, when he or she is chosen to go to a specific person, they are specifically trained for the needs of that person?
Yes, so all of the dogs have a standard set of training commands placed into their brains and into their drive. But when it comes to the matching process, actually it's kind of a date. So the individual is called up and told that Canine Companions has 1, maybe 2, or 3 dogs that they think might work. And during the first couple of days of team training, that's what we call it, we match them and have them, so to speak, date for little rendezvous, little tasks, and see what kind of pairing presents. And the trainers and the professionals who know these dogs very well can watch that dog behavior, see the bonding begin, and choose the right dog for the life of that partnership.
So very specific thought process and training processes going into that for the benefit of the dog and the eventual owner.
Absolutely.
You might share with our audience who might be listening anywhere across the country, if I were in some other part of the country and I thought I had a need for a companion animal, and I'm just hearing this today, where would I go to start that process? Is there a website? Is there a phone number? Share with us how we might do that.
It's as easy as canine.org, and we should spell that out. It's CANINE.org. And on that website, you can look up the different types of dogs that the organization has in their training process. You can even learn about all of our research that we are doing to make sure that we're training the right healthy dogs for the life of their work and for the benefit of canines. But you can also see all of our regional sites and which site your city and your town would be filtered to. Because in the state of Florida, in the 8 most southern states, we go to the southeast region, but out in Texas, you go to the southwest region. There's also the northwest, northeast. We have them 7 different locations across the country.
So once an animal becomes a partner with a person, how long does that dog function as a service animal? Is there a certain length of time?
There is no specific certain time that a dog will work for someone. It is a conversation between the dog handler, the person that was matched with the dog and canine companions, and the veterinarian, of course, because dogs will age and dogs will tire depending on how hard they're working in their daily life, and perhaps whatever illnesses they may inadvertently or unfortunately come down with. We do a great job of genetic testing our dogs to match and breed the right healthy matches, so that we have the healthiest of dogs that are out there. But it's between 8 and 10 years of age, maybe longer. But once you're with canine companions, you've got us for the rest of your life. So there's a conversation that starts up when we're talking about retirement for your dog. Are you ready for us to start talking about the next dog? And should we send you our application for a successor dog, is what we call it.
So kind of an emotional process for that owner and for the dog as well. They've been together for a number of years, taking care of each other, kind of fallen in love as owner and pet, but a great service for the person who needs that help. We've had a number of our members, I know 2 of our board members who have had service animals, and we as a board, we fell in love with those animals as well because we appreciate the personality of the animal. As you watch that animal take care of the person they are trained to take care of, it's so impressive to watch, and you know the life of that person is enhanced greatly because of what that animal can do and will do for them.
Yes.
Leah, is there anything else that I should have asked you about this process that we've left out that you think we should communicate?
Well, I think it's kind of important, or just a little side note. I'm coming to you from Canine Companions, but I think I should share that I too have HSP. I have SPG3A, and while I come as a representative of Canine Companions who has a facility dog, and I work in a children's hospital with one of their wonderful dogs serving multitude of children, my goal when Brea and I retire from our work life is for me to get a service dog from Canine Companions myself, because I know what she has done for me inadvertently, even though she's not my service dog. I know that I need one for the rest of my life.
Well, we thank you for being here with us today in Canine Companions, and you just hit on another storyline here that I want to make sure our listeners and the folks who watch this in the future don't miss, and that is that as we work to put this together here in this area, we had no idea when we started, when we reached out to the Tampa community and asked for help with our children's program and help with what we're doing here. We had no idea we were going to meet a fellow HSPer who deals with HSP. And so we appreciate meeting you in that regard. We want to recruit you and your involvement and support with our foundation as well. But thank you for being with us.
Absolutely.
And thank you for sharing with us. And if you're listening and you think you might have a need or want to consider one of these beautiful animals to assist you, Miss Leah has told you how to do that, so get online and start that process. And if you hit a snag, we'll find someone to help you. So Leah, again, thank you so much.
My
we appreciate
pleasure.
your time.
Thank you.
Well, guys, we've had a great day. We're about to have another session in here in just a moment that you'll be tuned into as we talk about the issue with child onset issues with Dr. Fakhari and some of the parents in our group who are dealing with that. So take a break for just a moment or two, and we'll be back with that group to close out the day. When we finish that session, we'll be finished for the day, getting ready for dinner here, so we'll let you go. After that, we'll see you in the morning about, I think it's 8:25. You got the program, so we'll be with you about 5 minutes before that program begins, and we look forward to this last session and to tomorrow. So thank you for spending the day with us, and we'll see you in the morning.